Hello, welcome to this ninth class of the Protein Engineering course based on artificial intelligence. And in fact, this is the class where we are going to talk about this topic. Remember that this is just one of the 10 classes that make up this course, which is completely free and is found on the YouTube channel so you can delve deeper into each of these particular topics. And before starting the class, the announcement is that all the images, the quotes, as well as recommended literature to be able to delve deeper into each topic, is found in the notes section of each of the respective slides. So for more information about this course, please check its official website. Likewise, check out the associated repository where you will be able to find tools, databases, as well as other courses related to artificial intelligence, as well as protein science to be able to delve deeper into each topic. To be able to download the slides and adapt them to a course you are developing, or access the information, you can do so from the Google Drive or Zenodo platforms. And if you want to ask a question, you can do it here in the comments box in the video on YouTube, or you can ask it via one of my social networks, where you will find me as the user at my hangar. And finally, if you find this course productive for your professional development and you like to reward it financially, you can make a donation via PayPal in the following link. Well, let's see what topics we're going to cover in this specific class. The first is to understand in general terms what we are talking about when we say protein engineering, and in particular, protein engineering based on artificial intelligence, as well as discuss the advances that have been made within this area in general. Later, we will cover what are the main methods with which we can design proteins. The first two would be rational design or evolutionary design, which precisely take information from the physicochemistry of proteins, as well as the evolutionary information that we can extract at the level of their sequences or structures, to improve functions or design proteins from scratch. And the second part of this class, which is the one that carries the most weight in this sense, is two large areas that have emerged thanks to the use of algorithms based on artificial intelligence, which is the first module called representation learning, where the algorithms that perhaps stand out the most within this area are precisely the protein language models, where we will cover how they are really interpreting the proteins as well as some phenomena that we have also seen associated with their capabilities, such as scale laws, multimodality, among other aspects. And as a last topic, we will cover what is the use of generative artificial intelligence to be able to precisely synthesize both protein sequences and structures. The idea here is not to exhaustively address what rational, evolutionary design, representational learning, or generative guidance is, but to touch on what the main algorithms are within each class, and precisely how they have adapted to what would be the era of artificial intelligence that we currently live in. So let's get started. What are we talking about when we refer to protein design? And in particular, even when we talk about protein design, but based on artificial intelligence, which, as it is relatively simple to understand, is actually the use of artificial intelligence algorithms to create or optimize new proteins with desired functions. That is, they meet some requirements, for example, at the level of their structure, as well as being capable of carrying out some biochemical reaction that is of interest to us. And so, why do we precisely design proteins? The reality is that the topics on which this area can be expanded are really quite a few. It is at the biotechnological level, for example, of the enzymes that we use to be able to replicate genes and thus study them. But there are also many applications such as in the pharmaceutical, chemical, cosmetic, environmental, agricultural, or even food biotechnology industries. But among all these issues, perhaps the one that receives the most focus is precisely the pharmaceutical sector, because it is the one that usually attracts the most capital and investment rounds in the private sector. And this is precisely what we are seeing in this bar graph, where on the y-axis we see what the market value of different companies would be, and we can observe that there are some, for example, from the United States of America, such as Eli Lilly, Johnson & Johnson, Merck, among others, that really have great economic power, and that is precisely based on biotechnological development. He finished studying proteins, as well as the molecules with which they interact. But there will also be other companies from other countries, such as Denmark, Switzerland, among other countries. And they also have their respective companies such as Novartis, Roche, or AstraZeneca, who together represent a part of what the biotechnology market is. But in reality, they really extend to quite a few areas of research. And this is precisely what we are seeing in this new bar graph, where what we are seeing are the top therapeutic sectors where there is more capital investment. Be able to develop research in search of various treatments. And well, it is quite clear that perhaps, at least currently, what it means to find treatments to deal with cancer, as well as all the varieties where this type of pathology can occur, is the one that takes the largest share of investment from the private sector. But there is also investment in the development of treatments at the neurological, endocrine, autoimmune, pulmonary, among other areas related to human health in general. And it is surprising to see that it is precisely cancer that receives the majority of private investment in this sense, directed towards biotechnological development. And within these 10 main medications that contribute the most to economic production, curiously, we find adalimumab as number 1. Adalimumab, which is an antibody precisely responsible for inhibiting one of the most relevant transcription factors that we have, which is a protein called TNF, and which is responsible for triggering many immune responses and which can even be used as a treatment against diseases such as part of rheumatology, among other aspects. And well, it is not quite strange to turn to see that precisely these types of treatments are the ones that are being investigated by the companies that are developing this type of artificial intelligence algorithms, such as Isomorphic Labs, which is in fact the startup where AlphaFold 2 and AlphaFold 3 are being used by Google DeepMind. Be able to do research within the field of biotechnology. And here we even find some statement from the director of artificial intelligence of this company, which is Max Jetterberg, who properly recognizes that they precisely have this type of research programs focused on the development and search for treatments at the level of immunology and oncology. Therefore, it will be very likely that when you do your research in the literature or even in the private sector, you will find greater interest at least within this type of treatments. Also, when we observe, for example, what the business niche is at the level of what companies are doing throughout the world, what they are focusing on in their research, we find different specific topics, such as the identification of small molecules to precisely be able to generate these treatments. But there are also protein design companies, antibody design companies, even glues, vaccines, RNA therapies, and in total, in general terms, we can say that so far, although it is not an exhaustive search on the database that I took for this analysis, there would be a total of at least 114 companies distributed throughout the world that are precisely doing, well, this research within the world of protein science, and where perhaps precisely the United States of America is the one that has the majority of companies in this sense. But there will also be consortia located in the United Kingdom, Israel, India, Germany, Canada, among other countries that invest heavily in the science and development of their own countries. And this, regarding the approach to the economy from the United States to America, It is quite interesting to look at how large technology companies, namely Nvidia, Amazon, Microsoft, Google, Meta, even Netflix, have begun to increase their investment capital in science and technology development, not only in an algorithmic sense to optimize their platforms, but it has also begun to get involved in the biotechnology sector. So that when you look at each of these companies and do some research regarding their investments at the biotechnology level, you will already find several projects within each of them. And here what this graph precisely shows us is the increase in investment capital in science and development in general. Well, investment in general by these companies, where perhaps a relevant part of all this capital is also being invested in science and development for both its purposes as a platform and also now the biotechnology sector. What this has done is generate a landscape of companies that, in general terms, there will be some that do is train their own algorithms based on artificial intelligence and distribute their licenses. In order to be used, there will also be other companies that what they do is also train their algorithms, but make strategic collaborations with other large sectors, both private and academic, to precisely develop these treatments now within a more experimental margin in the laboratory. And finally, there is also another set of companies who are dedicated to developing practically all of their technology from scratch, maintaining all of its intellectual property in terms of experimental developments, as well as algorithmic ones, where we find different companies. For example, isomorphic labs would be precisely within this margin, where they are also investing in the experimental part, as well as in the training of algorithms similar to AlphaFold or even AlphaFold itself. But we will also find other companies that, for example, are represented by kite discovery, even by bots, which are algorithms similar to AlphaVault 3, to AlphaVault 2, but precisely what these types of companies or startups do is provide the computational resources to be able to execute these algorithms, as well as the graphic interfaces to be able to run them in a more friendly way to any user who does not really have a fairly extensive background in computing and can carry out their experiments in this sense. Even this has opened up a quite interesting dynamic in my opinion, which is providing software as a service, but curiously not only software, which we are already quite used to, but also bioinformatics software as a service. And this is something that, for example, we see in cloud platforms such as Google Colab, where we can access the computational resources that Google offers through its cloud and that we can use for free, or that we can contract their different plans, but also through other platforms such as Diffuse Bio, 300GS AI, NVIDIA BioNemo, Lightful, even REX, which what they do is practically take the most prominent protein design computer algorithms and generate a more user-friendly interface so that they can precisely carry out their bioinformatics analyses, obviously with a monthly, annual subscription, or depending on the analysis that you want to carry out, and thus make this entire search procedure more automatic through artificial intelligence algorithms, which has been relatively interesting in recent years. And when we look again at how an economic sector has emerged, well, almost out of nowhere because this is a fairly recent technology. We are talking about approximately 2022. And where just now, in a couple of years, these types of startups have been established to be able to precisely provide this type of user-friendly bioinformatics services. It is also interesting to look at what the labor sector would be like for people with this type of bioinformatics profiles, but also literate in the development of algorithms based on deep learning, where we can see different job vacancies in this type of startups, such as Evolutionary Scale, Arcadia, Product Fluent, or even other more established companies such as GE, Zio Research, where we can even find salaries around $120,000 So it really seems to be an area that is being quite bustling or is having a great boom in this sense. And that is looking to capture jobs, talent at an international level. So it would perhaps be desirable to be able to train even more in this type of specific areas. And there are even quite interesting platforms such as Kaggle, which offers a series of computational resources to be able to execute processes in the cloud, as well as other learning resources. But perhaps Kaggle is more recognized for its international competences, which is precisely what we are observing here, where different companies or even the academic sector can launch challenges, and they can be of different kinds. They can be, for example, directed at image classification processes, segmentation, audio identification, or in this particular case, a challenge provided by Stanford University in collaboration with other companies to predict the three-dimensional structure of RNA molecules. And in Kaggle, what these types of competitions give us is a description of what is intended with this type of tasks, as well as the set of data with which we have to enter our algorithms. And there is even a section where you can discuss with other competitors within this competition in general. It would even be reviewing who are the best candidates to win the awards. Because even in this type of competition, precisely an economic remuneration is offered, which in this case was approximately $75,000, but which is being distributed among the main places. For example, the first place, whoever manages to precisely satisfy the requirements provided by the challenge in terms of the evaluation metrics, will win a prize of $45,000, and the rest of the $75,000 will be distributed between the second and third place. So if you know about artificial intelligence algorithms, both machine and deep learning, as well as the physical, chemical, and biological bases of various molecules, then you can enter this type of competition and see if the algorithms that you can train bear fruit. When we also turn to see if there are already some treatments that have been designed using artificial intelligence algorithms, applied in what would be treatments already at the level of products that have reflected a benefit, we will find that so far, or at least as far as I know, there is only one great example, which would be this compound, which is valsartanib, a compound that had been identified as a good treatment against rheumatoid arthritis. But due to the conditions generated by the pandemic and the need for new treatments to deal with this disease, which was COVID, it was found through artificial intelligence algorithms that, in reality, this compound could really be useful to deal with COVID. And in particular here, what benevolent AI did. It was a type of computer-based analysis known as knowledge graphs, where what one does is integrate multiple channels of information and do an exhaustive search for the type of relationships that allow us to hypothesize that some compound could perhaps precisely be taken from another perspective to address a new disease, as was the case with COVID. And in particular, we are talking about what this network of interactions is made up of information at the level of the molecules that can be used, as well as the set of diseases that we are susceptible to, our proteins and their corresponding genes, as well as the metabolic pathways in which they are involved, or even the expression levels of each of the genes and proteins. By then, through a series of algorithms, precisely identify to the best candidates based on the literature, as well as on the information available in general, which led us precisely to observe that this compound could be useful in dealing with COVID. Something that was relatively interesting because this particular network was truly gigantic, with approximately 15 million connections between multiple nodes that are represented by a total of 39 types of information. So it would be a search that perhaps we as humans could not do, and that thanks to the synthesis capacity of artificial intelligence algorithms, we can do this type of analysis. But obviously, there are also applications that are in progress to be developed as a treatment for human health. In this case, we see other examples provided by this company called Dinotherapeutics, which has different viral capsids that are specialized to be able to deposit the treatments they contain inside in a specific cell type. And then we have here two variants of this type of capsids, such as DinoBCAP1, which precisely specializes in delivering its treatments to what would be the cells of the central nervous system. But there's also another variant, which is the DinoECAP1, which is responsible for delivering these treatments again, but to cells related to our eyes. So it would be quite interesting to follow this type of technologies a little more closely and see if they become validated through this biosafety validation path. This type of treatments by health and biotechnology safety agencies, such as the FDA in the United States of America. And even quite interestingly, when we look at other companies such as Generate Biomedicines, which is precisely the company that owns Chroma, which, as you remember, Chroma is an algorithm for generating protein structures similar to what AlphaFold 3 does. They also find different treatments that have been developed against different diseases. And even curiously, at least on the part of Generate Biomedicines, some treatments have even reached phase number 1, within the total of 3 phases usually proposed by the FDA, which would also be necessary to be able to precisely implement this type of treatment and make it safe for human health in general terms. So up to this point, precisely the idea of protein design is to have a design or capabilities that are completely programmable, and in that sense it is precisely what this perspective offers us, written by several pioneers in the world of protein design who cautiously but at the same time enthusiastically tell us that this type of programmable design, or in particular programmable catalysis, will be available in the future, and that it will not only be the dream that existed for a few decades. In fact, when we talk about protein design being programmable, we are talking about us describing with a computer, even at a text level, some properties, for example, structural or functional, that we want other proteins to have. To understand that, for example, we say that we want a protein composed only of beta sheets, because perhaps we find that this is a fairly stable folding for the use in which we want to carry out our design, and that in addition this protein composed of pure folded veins becomes capable of making some compound that becomes capable of carrying out some specific biochemical reaction through only the text-level instructions, or even little more that we give it. This has actually been like the dream, or one of the things that have been tried to achieve over several years of research, and that perhaps we are just beginning to see these small glimpses that this can be real through interfaces, for example, such as large language models, that through indications at the text level, or even the generative and conditioned capabilities of diffusion models, well, now we can design this type of proteins without perhaps needing to know super specific details of its associated physicochemistry. And in this sense, it is precisely the work of multiple colleagues throughout the world who are precisely in charge of investigating how to achieve this completely programmable protein design. What you can see here is a repository which I strongly recommend you review in case you want to learn more about it, where there are even a few hundred laboratories distributed throughout the world of scientists or proteinologists in general, who are precisely seeking to achieve this type of completely programmable engineering. And even within that section, a colleague named Sebastian also published some quite interesting information from laboratories of proteinologists, specifically that are precisely trying to address this type of problem. And here, this is on a personal basis. There is a list of several researchers whose work I personally admire a lot and follow on a recurring basis, so I would highly recommend it, as well as researching at least profiles such as Google Scholar, or even following them on different social networks such as Blue Sky or even Twitter, now X, in order to be aware of what their most recent publications would be. For example, we find people like Sergi or Mohammed, who precisely carry out analysis at the level of interpretability and explainability of this type of algorithms, or even developing a new code to be able to extend these generative capabilities of these models. We also find other profiles such as Nolia Ferris or Patrick Bryant, who stand out precisely for their training generative artificial intelligences, as for example in the case of Nolia Ferris, trained an algorithm of the first of its kind known as PROGPT-2, which we will review in later slides, or Patrick Bryant, who even what he does is take precisely this type of models such as AlphaFold 2, and now develop them in a generative facet to investigate what causes they would have learned during their training. We also find another set of researchers such as Anthony Jeter, Bill Romero, or even Francis Arnold, who in general terms what they do is bring precisely this type of algorithms based on artificial intelligence to directed evolution campaigns, something that we are also going to investigate in later slides. And what they do is generate a series of feedback based on what the experimental data are. And so our experiments come out, let's say, better and better. That is, we can optimize the function of proteins using screening based on bioinformatics algorithms. We also find other characters such as Kevin Yang, Pranam Chatterjee, Fajan Johan, or Martin Steninger, who develop quite a few algorithms within what is bioinformatics applied to what is protein science, which would also be quite interesting if they followed him. And in particular, Martin Steninger also developed, Martin Steninger's laboratory developed a very useful tool to be able to compare protein structures in a massive way called Foldseek, which in fact was also a watershed within the world of protein science in general, and which we will review in later slides. We also find other characters such as Bunny Berger, Jennifer Litz-Garten, or Po Sa Huang, who even make a little more theoretical developments regarding what this type of algorithms can do. We even have other pioneers, such as Deborah Marks or Bukarth Rost, who were already within these competitions such as CASB, seeking to make precisely this type of predictions based on co-evolutionary information before neural networks were as famous as they are now, and that they are currently trying to develop new language models, new diffusion models, and extending the theoretical capabilities that we currently have to precisely develop algorithms. And we also have young promises such as Brian High or Martin Pesa, who are again adapting these types of models such as language models to precisely be able to do evolutionary analysis, or even design proteins that bind to other proteins with a fairly considerable success rate compared to the success rates that we had with the algorithms that had a physico-chemical basis, something that we will also investigate in more detail in later slides. So in general terms, I would highly recommend reviewing the work of this entire series of researchers, and that, unfortunately, I am limited, both in time and space, to be able to talk about the work of many other colleagues, which in this case are only those who, in my personal opinion, I consider have had a great contribution within the world of protein engineering based on artificial intelligence. But perhaps here I am not necessarily covering the work of other colleagues who may be oriented more towards evolutionary biology, more towards cellular biology, among other aspects, but in this case they are colleagues who work precisely on the topic that we are investigating in particular in this session. In fact, together these types of colleagues, along with others, have signed what is this letter of responsibilities for protein designers in such a way that a series of criteria values are precisely set out. This in general guidelines on how to direct our work to precisely carry out responsible designs in terms of biosecurity that the development of this type of algorithms may imply, because as we can use them precisely for the development and characterization of treatments against diseases, it is also commendable that they can be applied in terribly erroneous ways to generate some harm. And in fact, this is even reflected in several algorithms, as could be the case of the SM3 algorithm, a protein language model, which even following what would be the guidelines proposed by this letter in general, because they precisely removed or weathered, let's say even so, they censored the capabilities of this generative model to be able to synthesize proteins related to toxins or viruses. Something that when one releases a model in an open source way, one has to be able to consider in sociological, ethical, even philosophical terms, what kind of implications its release has precisely for the entire community. Because practically anyone who has the money to pay for cloud resources or physical computing at home, in their laboratory, well, you can make both good and bad use of this type of algorithms. So it is also necessarily thinking about the types of things that can be done both for good and for bad in this sense. And really, when we look back at this dream of what protein engineering is, a completely programmable protein design, we can subtract it even back to the 80s with this type of magazines from the United States of America, where we find precisely approaches to what the design of molecules could represent, which in this case, with this cover, spoke of what the next industrial revolution would be through this type of research in biotechnology. And in this sense, in addition to the work of all our colleagues that we saw previously, one that perhaps takes many of the cakes from us, and that also represents a great intellectual admiration for many of us, would be the work carried out by David Baker, who even because of his work, and the fact that he managed to move in the right circles, he came to the establishment, to the creation of what would be the Institute for Protein Design, based in the United States of America, in Seattle in particular. And from what would be David Baker's laboratory, which, well, within this institute there are also other colleagues. Quite relevant algorithms have been born for what is protein science based on artificial intelligence, such as the Rosetta Fold algorithm, ProteinMPNN or Rosetta Fold diffusion, which are precisely algorithms that we are going to investigate in more detail in subsequent slides, but also the design of other algorithms that were watershed during the first years of research in this specific area, such as the Rosetta Suite, which, as you will remember in our classes on structure, function, and even evolution of proteins, this type of tools was developed based on physical, chemical, and biological bases of proteins in order to optimize the design of proteins. And that in a very intelligent and curious way, laboratories like David Baker's decided to now focus completely on the development of tools based on artificial intelligence, because they really demonstrated great performance in the tasks. They do, even over algorithms that have quite robust physico-chemical bases. In that sense, when we look at what would be a brief history of protein design, and when it was really born, in what year we can say that the first design was achieved, we are talking about approximately 1988 with this particular work, where perhaps it was not a very complicated protein. It was a protein composed of only 4 alpha helices. But at this time, we are talking about it was not even the 2000s. That is, access to massive sequencing platforms had not even been democratized, since it was possible for the first time to design a protein and through experiments corroborate that it would be expressed within cells. But this is precisely what allowed us to establish this new era of protein design, where, based on our knowledge, structured on what would be the physical, chemical, and evolutionary bases of proteins, we now had access to being able to design this type of biomolecules, and not only reuse them for other functions that interested us. And even perhaps one of the most notable advances in what would become the field of protein design is precisely this protein that was called DOP7, which in 2023 became the first de novo protein to be completely designed by computer. And when we talk about de novo proteins, or perhaps in Spanish its most faithful translation would be proteins from scratch, it is precisely this idea of being able to imagine, design, and in general create proteins whose functions or structures we have not found in nature. That is, actually designing it from scratch, and for which the authors who took on the task of designing this protein, crystallizing it, and expressing it, evidently had a whole background at the level of protein structure, at the level of function, but even the algorithm behind being able to characterize this type of biomolecular structures and see which ones would be most praiseworthy if they could be expressed. And this was only achieved in 2003. We are talking about the fact that from then until now approximately more than 20 years have passed to get to where we are today, where simply with a few clicks we can even generate proteins that can be expressed in the laboratory. And now perhaps, although we do not have the great knowledge regarding how these molecules work, since even an undergraduate student can access this type of algorithms, design their proteins, and express them in the laboratory, something that previously required even doctoral students or, perhaps more especially postdoctoral students, and even well-established researchers, who could discriminate which proteins could be the most permissive in order to reach to experimental success. And this is precisely what we are talking about when we say precisely the central dogma of protein design, how we design proteins. To begin with, what we have to define is what we want to solve with our protein. That is, what function we want it to fulfill, a more structural function. A function of a catalytic nature, that is, enzymatic or even binding to a specific ligand. And once this first aspect is resolved, we have to assess which structures would be capable of satisfying said function. Because, as we already know, there will be some proteins that can precisely contain said function due to their thermodynamic stability, their flexibility, among other aspects. And as you also now know, many possible sequences actually correspond to a structure. So it would also obviously be necessary to determine which specific set of amino acids would be the most optimal to be able to solve the function that we are trying to design. And this is previously what we intend to do. That is, we characterize, for example, in the case of an enzyme, the active site, that is, the catalytic residues, or those that stabilize the transition state, and thus identify which would be precisely the residues that would be capable of carrying out catalysis. Later, we find a folding that becomes capable of housing our specific function. In this case, we are seeing that it would be a protein similar to what a team barrel is, and that later, once resolved, that perhaps the team barrel is an optimal structure to host this type of function. Well, now what we have to do is precisely identify the identity of the side chains of our protein, so that perhaps we are talking about wanting to express our protein in terms of conditions at room temperature, that is, approximately 37 degrees. Maybe in this case, we don't need something very thermostable like 67 degrees or 80 degrees Celsius. And then, for this specific temperature requirement, perhaps a particular set of waste will be able to satisfy that condition. But really, when we look for a condition, we are referring to a whole set of conditions such as its thermostability, its level of expression, its solubility, the consideration in what type of host we are going to express our protein, among other aspects. And when we turn to see how precisely this dogma of protein design occurs, this is really what generative AI algorithms are doing today, where we can take algorithms as diffusion models and generate that type of conditioning on what would be precisely the residues of the active site, as well as a particular ligand, which is precisely the one that is sought to be catalyzed, which, for example, is precisely what algorithms like Bolt's design do, which we already covered in what the AlphaFold class is. And that by executing these steps of noise removal, or denoising, we can precisely arrive at what would be the generation of a structural candidate capable of housing said structural motif composed of 1, 2, 3, 4 residues and specific ones capable of carrying out catalysis. And although perhaps we do not reach the current resolution necessary to be able to solve this type of enzymatic designs, we do have the capabilities to be able to generate structures that can be folded, which are also quite expressible. But perhaps we have not yet reached this resolution at the level of your Armstrongs, so that the enzymatic design, which is in fact one of the tasks that is still being addressed today, can be satisfied in a fairly easy way. And although there are advances, such as the algorithms of Rosetta Fall Diffusion 2 or even 3, progress is being made precisely in what would be its application to enzymology in general. And so we continue. Because we want to generate proteins from scratch, because it is not enough for us with the proteins that we already have in nature. And this is represented by this small diagram, where what we represent is precisely the diversity of proteins, as well as their possible functions associated with each of them. So precisely what interests us is to explore this space of both structural and functional diversity, because, for example, within all the possible proteins for a particular 100 residue protein, we find a combination of 10 to 65, which is a gigantic number. And when we look at how many proteins are stored in the databases, that is, proteins that we might think are natural, whatever that means. We are talking about presenting a few panels within all the possible diversity, represented by the set of 10 to 65 combinations of residues. And specifically when we look at the diversity represented in what would be the database of protein structures par excellence, which is the protein data bank, it really is a microcosm regarding the possible reality of the diversity of this type of biomolecules. Therefore, finding new sequence spaces that perhaps yield new structures or even new functions is of our interest to be able to solve tasks that perhaps were not contemplated at the beginning, because perhaps now it is a task of perhaps removing compounds that have been generated by us and that throughout evolution, since these types of proteins were never faced with this type of problems with which to interact and even co-evolve over time. So then there would be no type of interaction between this type of compounds with respect to the proteins that we can find in nature. And this is precisely what has been done throughout all these years of research. Starting from enzymes that we have characterized quite well, and that it is quite evident that the vast majority of them are actually natural proteins. Again, you do whatever that means. And eventually then, characterizing them and through their characterization, both bioinformatic, evolutionary, and experimental, then extend and generalize what attributes would make said protein at a structural and functional level carry out said function. So, based on what would be similarities at the sequence level or at the structure level, well, we can write down new proteins from what would be the set of enzymes that would be possible. And based on all this information, we can precisely return to various aspects and design our proteins considering what would be, for example, the catalytic residues, what aspects they must satisfy, for example, in thermodynamic terms, among other aspects, to precisely extend our capabilities and design new proteins. And this is the way in which we have been working until today, and it is not until approximately 2020 to 2023 that we have begun to adopt in a fairly exhaustive way what would be the use of this type of generative artificial intelligence. Algorithms to be able to solve our tasks without the need to go through this entire path of quite refined understanding in this sense. In fact, this is precisely one of the desires of this programmable protein engineering capability. Being able to investigate both at the level of sequence space and even extending beyond the alphabet of proteinogenic amino acids to what would be the use of non-canonical amino acids, such as selenoscysteine, among others, which could perhaps provide us with new benefits, a completely new catalytic repertoire in conjunction with other molecules that are not canonical as new cofactors, that what they do is expand all the biochemistry that has been developed by evolution, but with which we can now intercede and precisely expand the diversity that we observe today, not necessarily the diversity that has been explored throughout the thousands of years of evolution in total. So that in general terms we are talking about that we can adopt a protein, for example, a Tim barrel, a Rossmann fold, among other specific folds, and give them a repurpose based on, for example, what would be the promiscuity of their catalytic activity. And then, through the specific, localized mutation of some specific residues, improving their genetic function. Whether it is binding to a ligand, enzymatic catalysis, structural function, so that it is precisely able to satisfy our requirements. We can also generate proteins from scratch, based on what would be the interactions that we want to design. For example, perhaps having a super-stable protein, which is a structural link, so we would have to have a fairly well-established hydrophobic core, or even to address what would be like an unnatural chemistry, to understand precisely the power to treat this type of compounds that have been synthesized by our human activity, and that proteins throughout evolution never had the opportunity to interact with this type of molecules, something that we see quite well represented by this type of research by Francis Arnault's laboratory, who precisely repurpose a protein well known to proteinologists in general, which is cytochrome P450, in order to degrade a set of molecules that are precisely produced by us at the level of megatons per year, and that is not biodegradable in environmental matters, which even accumulates due to our industrial activities, which are precisely the siloxanes, which, as you can see, are bonds of the silicon type with oxygen, when in reality, well, the biochemistry to which proteins are most associated, since they are bonds more usually of carbon with oxygen or carbon with other entities, and not so much with silicon, something that is really completely new in biochemical terms. And thanks to what would be the retouching of certain positions in this cytochrome structure, well, it can even degrade molecules such as siloxanes, which practically never had anything to do with proteins throughout evolution, and thus be able to precisely treat problems such as the bioaccumulation of this type of compounds, which can be quite toxic at an environmental level. We also found inspiration regarding what the structure of proteins would be, in a slightly more inspiring, artistic way, of what would represent, for example, motor structures such as the flagellum of a bacteria, with respect to what would be the engines that encourage the flight of the airplanes that we use in our daily lives, where, although they do not explicitly have the same function, they do share certain structural similarities that invite us to reflect in this sense. In fact, when we see precisely how the bacterial flagellum rotates, we find a certain beauty in this type of structures that allow cells to move. And not only the cells of bacteria, archaea, but also precisely the eukaryotic cells through processes even more complicated than just a bacterial flagellum. And even thanks to protein design, we have reached the capabilities that we are capable of developing this type of biomolecular motors based on all the protein science that we know today. So here you have precisely examples of this, of how by characterization at a structural level of the interactions between various macromolecular complexes, we can currently design molecular motors such as the flagellum that was assembled over billions of years of evolution. And so we then come to this reflection of what is the historical development of protein design algorithms that led to what would be the stage of protein design based on artificial intelligence. Something that you can consult quite well in what would be this review article that I highly recommend that you review after this specific class, where what they tell us is that these artificial intelligence tools they have already reached a degree of maturation where, really, their integration into what would be our work is not only possible but essential, and perhaps colleagues who refuse to adopt them are already doing more due to perhaps intellectual conflicts regarding their adoption, and not so much their performance than what these types of tools are capable of doing. Here what we observe is precisely the progress that has been made to reach this establishment of generative AIs that allow us to both analyze and synthesize protein structures or sequences. We have that what the bases are precisely are aspects such as precisely the first protein to be sequenced, something that we already saw in our protein structure class. Also aspects such as the first structure also to be characterized, which at that time we were still making models based on plasticine. The establishment of our main databases, such as the Protein Data Bank. Other databases, such as UniProt. Its most refined sections, such as SweetProt. Or even series from which we can deposit genomes that have been sequenced, such as the GenBank database. Or even tools that allowed us to compare protein sequences in the 90s, such as BLAST, which, as you remember, we already covered in the classes that precede this specific class. Later we are talking in the decade approximately of the 90s of what would be a first computational phase, where there were developments like the ones I mentioned before of the top 7 protein, which involved the computational and experimental characterization of proteins that did not exist in nature, at least as far as we had detected at this time. And also, obviously algorithms emerged here that precisely allowed us to better characterize our bioinformatics searches, such as hidden Markov chains, as well as visualization suites, such as PyMol or Chimera, which make it much easier for us sharing our ideas with other colleagues, and thus increasingly developing this type of protein science. And finally, we reached what would be this stage of protein design based on artificial intelligence, where now the databases that we have are not only several tens of thousands, but there are even millions of sequences deposited in the databases, as well as several 100,000 in the PDB, which together obviously go hand in hand with the appearance of these algorithms based on deep learning, such as convolutional neural networks with the AlexNet moment, or precisely the revolution caused by transformers, even by diffusion models, which led precisely to the achievement of AlphaFold 2 to be able to solve the task of predicting structures at the monomer level, as you will also already recognize. And after what AlphaFold 2 involved, there was a huge amount of research into this type of algorithms into what is the essence of proteins, establishing new algorithms such as Foldseek, ProteinMPNN, ESM, and EFE, among others that we will investigate in slides later. The establishment of the AlphaFold database with approximately 200 million protein structures. The creation of the first protein language models that allow us this versatility provided by protein languages, such as ChatGPT, but now in the sphere of proteins. And finally reaching what would be the appearance and application of diffusion models as means of generative artificial intelligence, and thus generate proteins or sequences using algorithms such as AlphaFold 3, Rosetta Full Diffusion, Beamcraft, among other algorithms that we will also investigate in more detail in later slides. So here we have that, at least we as humanity, until November 2025, have designed a set of approximately 1738 proteins, which is quite well compiled by this new database, which is the protein design file, which represents more than 40 years of research in this area, since approximately the 80s, as we saw previously, where we can say that there are several phases within this area of research. A first phase where it is precisely associated with the type of rational design, that is, knowing the physical, chemical, biological, evolutionary bases of proteins to be able to design proteins, either completely from scratch or giving them a new purpose based on quite skillful folding. Something that at least during this first phase, we really accumulated relatively few proteins, until we reach approximately 2010 where you can see here what the slope of this bar would be, where we generally reach less than 200 designed proteins even in 2010. And that later in what would be the decade from 2010 to 2020, thanks to what would be the arrival of these bioinformatics algorithms, we can no longer only base ourselves on what would be the rational design, but now we even incorporate first versions of machine learning algorithms that are even incorporated in suites such as Rosetta or Foldex, which precisely allowed us to do a bioinformatics characterization, and thus increase the chances that our designs would really be effective, which made us go from approximately 200 designed proteins to a total of 1,000 proteins heading towards 2020. And that is now thanks precisely to what deep neural network algorithms are, in general deep learning, or perhaps more specifically language models and diffusion models. It really seems that the trend, at least provided by this slope, would be an almost exponential development of our abilities to design proteins. Something that we can verify in more detail by reviewing this quite beautiful project, in my opinion, which compiles all these years of research in protein design. In fact, in order to synthesize what the forms would be, as well as the methods that were developed to generate this type of protein designs, you can review this review that, well, let's say that it is quite recognized within the world of proteinologists, which compiles quite well what have been the greatest efforts in being able to design proteins in this sense, and that in a very curious way. Well, here they find quite capricious shapes, although also, quite curiously again, it seems that there is a great dominance of alpha helix structures, an aspect that we will also review in later slides, that perhaps this type of alpha helix proteins were easier to express, characterize experimentally, and that perhaps now we are already making this leap to include much more diverse architectures. And in order to precisely review how artificial intelligence algorithms have impacted the new design of proteins, you can review this other review that is also very curiously inspired by what is an article that is based on what were the first results of the GPT-4 architecture, implemented through ChatGPT, where we even find Well, in a relatively analogous way, what would be the titles of these respective projects, which in this case would be like sparks of function in the design of proteins, or in this case, it would be sparks of general artificial intelligence. In the first experiments with GPT-4, where they also compile, in a fairly systematic and well-narrated way, what would be all the sets of designs that had been achieved at least until February 2024. Through algorithms based on deep learning, from its use with what would be models first through machine learning, such as Gaussian processes, random forest, subsequently passing through what would be deep learning models, such as convolutional neural networks, and finally arriving at what would be the language models or diffusion models of today. So, thanks precisely to this type of algorithms based on deep learning. Well, we can talk about the fact that protein design is being democratized to make it much easier and more accessible to people who perhaps do not have a background in protein science in general, or even at the level of bioinformatics and deep learning itself. In fact, here we can see a characterization of the type of proteins that have been developed over time. In this sense, on the yes-axis we have a metric that approximates in a fairly large or coarse way what the complexity of this type of designs would be. A metric known as RCO, or the relative contact, of what would be your contacts between the proteins. So we compare this metric of the complexity of a protein based on its contacts with respect to what its size would be in number of residues, where we find that what would be the distribution of natural proteins, again, whatever that means, since it is much more distributed compared to what would be the set of proteins designed completely from scratch by us as humanity, which are still quite limited in what would be a range of approximately 200 to even 300 residues in length, and whose complexity really is not as much as they could be, mainly proteins composed only of alpha helix regions, and that when we turn to see proteins that have a greater complexity measured by RSO, we see much more elaborate architectures where there are already both interactions of alpha helices and folded betas in various aspects of their specific architecture. And this is quite interesting because when we take what is precisely this database of protein designs of approximately 1700, we observe that the designs that we have achieved are in general terms less than 300 residues in length, and that only a few handfuls of protein designs exceed precisely this length, which is illustrating to us that it is quite complicated to actually design proteins that are quite large, which is quite sensible to expect because a larger protein also necessarily requires satisfying more contacts, more properties, such as what would be thermostability, among other properties that allow them to be expressed within the cell. And now we see precisely the comparison of what would be the rational methods, the methods now based on physics and the methods based on deep learning. Again, with what would be our complexity metric, which is the RCO, as well as on the x-axis, the time measured in the years of research there is, where we can see that precisely the first protein designs in what would be the 90s. Well, we are really talking about proteins with minimal complexity, which were practically small alpha helix motifs, which were subsequently developed more and more thanks to what would be the set of protein science algorithms, such as Markov models, the Rosetta suite, algorithms such as PyMol and ChimeraX, which allowed us to visualize in a friendlier way how our designs behave. And precisely thanks to what would be deep learning algorithms, we are reaching designs that are already relatively more complex, that can precisely recapitulate the distribution of natural proteins that we have observed to date. in such a way that even thanks to this type of algorithms, we can design large molecular complexes, such as these proteins that meet the condition of being. Well, in volumetric terms similar to the letters of the alphabet, in this case illustrated by the word DNA, which when we see it under the microscope are really expressing themselves and adopting these shapes similar to letters. Or even through what would be frameworks that assemble multiple artificial intelligences, we can even now design proteins of up to 1,500 residues, which, as you can see, it is really exceeding these capacities that are registered in this graph, which is telling us that these types of algorithms are being quite effective in being able to generate this type of macromolecular entities, even though they are as large as 1,500 residues could be, and that again when we see it in the microscope, it is actually being expressed. This has led to, for example, statements in preprints, such as this article, which is precisely where the MPNN ligand algorithm is presented, a variant of protein MPN, which are precisely sequence generation algorithms based on a structure. Where the authors throw this text, which caused me quite a bit of curiosity, where they say that precisely the precision of MPNN ligand has made it quickly become the de facto method adopted by this group to be able to design the interactions of proteins with nucleic acids, and that throughout the characterization presented precisely in this work, it made it have great results in experimental terms, and that, when we really turn to look at the statements even from colleagues, that many of us have a great intellectual admiration, and that we respect a lot like David Becker could be. In an interview, he actually even recognizes that the capabilities of these algorithms based on artificial intelligence is almost miraculous in such a way that they have even given it a particular name that, in this case, could perhaps be translated into Spanish as wild biochemistry, where really it is just simply launching into doing this type of analysis and finding that they are really working quite well, and that, well, really, if we come from this conventional educational framework of how the rational design of protein science works, well, we will really be quite surprised when we find the effectiveness of this type of tools, to the point that laboratories such as this one, which is at the frontier of protein science research, adopt it as the default tools to be able to carry out their particular analyses. So even if this laboratory adopts them, it would be highly recommended that we also look at how to apply it to what would be our specific research frameworks. In fact, when we talk about what the scales in biotechnology research would be, we are talking precisely about several strata in this sense. When we characterize a molecule, we are obviously talking about a molecular scale, where we characterize properties such as its sequence, its structure, its function, its thermodynamic stability. But when you get to climate, for example, to what its application as a product would be, it is necessary to consider several scales, such as how this new protein would be integrated at the level of metabolic networks, even at the level of how it would perform within both cellular and cell-free expression systems, and even what would be the first scalability trials, in what would be the bioreactors that we can have as microbial consortia or small trials. Tissues that can express the proteins that interest us, and eventually scale them up to what are these large protein synthesis plants, so that we can really produce proteins in quantities necessary to really satisfy the use that we want to give them, or even when we reach applications at a larger level, we have to consider both ethical and social environmental aspects of the deployment of this type of protein-based technologies. So when we talk about a successful protein design, we are talking about a characterization along all these scales, in addition to what would be precisely the success at the level of its specific functional activity, also of the specificity of its function, for example enzymatic, which concerns only a specific catalytic reaction. Also in terms of the thermostability of said protein, depending again on the context in which we want to develop it, and how applicable, how much this technology can be developed throughout all these spheres as we previously reviewed. What we have here are some red flags, some little red flags, that can help us precisely rule out proteins. That perhaps they are not made the best to be able to solve our experimental tests. This is based mainly on what would be the behavior of globular proteins and perhaps is not as generalizable to other types of proteins as intrinsically disordered proteins could be. We are talking about when we are dealing with globular proteins, then the packaging of the hydrophobic core is quite relevant. We need this hydrophobic core to be well established, so that in thermodynamic terms during folding, what would be its three-dimensional folding is adopted in a relatively easy and fast way. So if there are some cavities, if the interactions between hydrophobic amino acids that are buried in the structure are not maximized, then there will not be good packaging of our structure, and it may be that it becomes more flexible over time and does not fold to carry out the function that we want it to perform. And remember, the most important thing is that our protein folds, and then we ask it what things it is capable of doing. But before it performs its function, it must adopt its specific three-dimensional folding. We also found another flag network, such as the presence of cysteines, which in this case, although they can provide great stability, it turns out that if they are not really paired with what would be another cysteine residue, well, they can generate aggregates like these amyloid fibers that we previously reviewed, which can generate a defect in what would be the folding, and even affect the folding of other proteins forming these aggregates. We also found that what is the physicochemical properties related to electrical charges is also precisely affected in terms of experimental development, in such a way that what we want is for the charges of our protein to be relatively homogeneously distributed, as it would be in this sense. And in this case, we could perhaps see a set of residues that have a negative charge, marked here in red, forming a kind of groups or clusters with a high charge. And precisely because of these charges, it is that then, since it's electrostatic dynamics at the level of the interactions, it can carry out with other proteins, and even the dynamics itself within the cell can be much more unstable in this sense. So what is desirable is precisely to maintain a certain homogeneity, and that only in specific regions is it good to find that type of charges, because perhaps they are the ones that are carrying out the binding to a ligand among other types of interactions. And finally, we have this other property that we previously reviewed, which is precisely having residues that are hydrophobic, but in this case they are exposed in the cell. Or vice versa, residues that are hydrophilic that are buried in the protein, which is not plausible, because we already know that the behavior of proteins is similar to droplets of water and oil. Regarding the fact that the hydrophobic residues, which are those that precisely direct this hydrophobic effect for the adoption of the three-dimensional folding, must be buried in the protein, while residues with a behavior similar to water, that is, hydrophilic residues, should be located on what would be the surface of the cell. And in this case, we can see precisely which charged residues are positioned inside this protein. Something that is not plausible, and that should therefore considerably lower the success of our protein in experimental terms. This is only one factor. In this case, we reviewed that it was solubility, but to talk about success at an experimental level, there are multiple factors, both intrinsic to the protein and extrinsic to it, that we have to consider. For example, in addition to solubility, we obviously also have to reflect on its foldability and thermodynamic stability. an aspect that is more related to what would be the specific folding that we are addressing. For example, we could be talking about a cytochrome P450 with respect to perhaps a beta-lactamase type enzyme or a Rothman type or Timbrel type enzyme. Maybe some folding within those may be more suitable to satisfy the function that we want to design. But also when we consider the factors extrinsic to our protein, we find aspects such as the host where we are expressing it, the experimental conditions in which said protein is being expressed, or even more refined aspects such as the bias in the use of codons that exists between each of this type of hosts and their corresponding replication translation machineries. Even if we insert our gene in the form of a plasmid, we also have these expression cassettes that can also be regulated by multiple promoters that can favor or disfavor its expression within the cell. These are just a few features that we have to investigate, but really when you look at what we already reviewed in the protein biology classes in general, you can find several data here, along with this table that you can also review in more detail, where you will find more information about it and thus maximize the success of your experimental designs. For example, preventing it from having toxic behaviors, many repetitions, many cysteines that are not linked, among other aspects. With all this context, we then get to explore what the main paradigms are in carrying out protein design. To understand what rational design would be, evolutionary design, and in a relatively more modern way, what design based now on artificial intelligence would be. And really what has happened with the new theoretical developments is that the borders are being drawn between what we could classify as rational, evolutionary, or design based on it. So perhaps we are beginning to talk in a more comprehensive way about what it means to carry out this type of approach. When we talk about rational protein design, what we are really talking about is a fairly refined understanding of how all the involved parts of our biomolecular complexes work. To understand, if we want to carry out a reaction that goes from A to B, then we can consider the two main ingredients at the protein level that we have, such as the scaffolds, or what would practically be the protein folding, as well as what would be the active sites or residues that are capable of, for example, carrying out some catalytic mechanism or binding to a molecule that interests us along with other molecules such as cofactors or even metals, in such a way that we then know specifically which component is to modify, mutate in this sense, and integrate into what our design would be, so that we then move from reaction A to reaction B. This is the part of rational design, and I repeat, it is based on a fairly refined understanding of biology, physical chemistry, evolution, even what our elements would be. Designed in the sense. Within the world of rational design, there is also a sub-aspect known as subrational, which is also based on a relatively refined understanding of what the physicochemistry of proteins is, but at a coarse-grained level, where now what we do is recombine multiple elements, which is also known as recombination, in the sense that we are combining the words recombination with engineering, and engineering obviously referring to what the components of protein structures are, both at the level of structure and at the level of function. So we have approaches that are top-down or bottom-up in relation to how precisely we are integrating the parts of the proteins with respect to, for example, recombining some regions that are only alpha helices with other regions that are also alpha helices, or give more freedom to the system, so that even now no twist regions are introduced that would make the protein more flexible in some aspects among many other mechanisms that we can do, even starting from the fact that perhaps the first designs using this type of approach are not made so functional in terms precisely of the catalytic metabolic reaction that we want to carry out, We can make use of experiments such as the overexpression of chaperones, so that they can rescue the folding of these first sketches of the function that we want to achieve, and as we optimize our designs, actually achieve a protein that is completely stable. Within the rational design, we also find what would be the de novo design, which, as we already covered, really consists of thinking, imagination, and the creation of proteins that do not have homologs in nature, in such a way that we must even think about what would be the catalytic motifs that could be capable of performing a function. And based on this idea, build a structural scaffolding of a specific fold capable of housing said functions. Later, we move on to evolutionary design, where we mainly find approaches that exploit the diversity of protein sequences. And in this sense, one of the most used techniques is this so-called directed evolution, which practically consists of generating a library of proteins, or specifically of their respective genes, and apply some selection method that allows us to scan which of these proteins would fulfill a function to a greater or lesser extent, something that is also known as fitness, as we previously explored in the protein function class. And then once the best candidates have been identified, we use them to inoculate the next generation of proteins using other methods that they diversify their sequences, that is, mutagenesis experiments, in such a way that we interactively carry out this cycle of diversification of genes, that is, of proteins, the process of screening and selection, as well as amplification, to increasingly improve the desired function that we want to achieve. And within what evolutionary design is also found, we find other approaches such as the design based on consensus, or what is the reconstruction of ancestral sequences, which what they do is exploit the information from multiple sequence alignments and obviously give it a more frequentist approach in the first case or a more phylogenetic approach in the second case, to find key mutations that help us improve the activity of our protein in terms of thermostability, or even perhaps its specific function. And within the protein design paradigm based on artificial intelligence, although there really is not a consensus, and this is, let's say, an organization that I propose, we find what the world of object representation is, which is representation learning in general terms, which what it does is, well, what is the richest way that we can represent, whether in interactions between proteins, the protein itself, the sequence, the structure, the sequence, and the structure in parallel. That is, as you can see, at a computational level, we can represent proteins in multiple ways, and these shapes precisely condition what our algorithms are learning. So if we, for example, represent proteins only from a mathematical language of secondary structures, we would only be representing this diversity in terms of the 8 possible states, or even 3 possible states that we can find in secondary structure. But when we now diversify this alphabet to perhaps 1 letter for each of the residues that make up proteins, you realize that we then diversify our alphabet, and therefore the type of information that can be modeled by our algorithms. And in a relatively similar way it happens when we represent protein functions, protein interactions, protein structures. Practically any phenomenon or biological object can be represented in some specific way, and this representation has to be rich enough in information to be able to distill precisely the phenomenon that we want to model using some artificial intelligence algorithm, or more particularly perhaps specific deep learning. And also within what this paradigm of protein design based on artificial intelligence is, we find what is the application of generative artificial intelligence. That is, models that are capable of synthesizing sequences, structures, or in parallel both sequences and structures. Obviously, thus exploring a hypothetical space of possible solutions that could satisfy said three-dimensional fold, as we can see in this small image where, due to the structural context, An artificial intelligence has to be able to model the distributions that allow us to predict which amino acids would be the most likely to satisfy said region. So, in the following slides, what we are going to explore is not exhaustively what each of these main paradigms are based on, but rather the advances that have been made in each of these in terms of algorithms based on machine learning or deep learning specifically. So let's see what we are talking about when we talk about rational design. Remember, it is based on a quite refined design of practically all parts of our biomolecular complexes. And in this sense, before exploring what this whole range of approaches would be, I would like to talk about what would be some quite outstanding experimental protocols that you will very likely find in the literature, and that even these types of experiments are in fact what provide us with data to be able to train our algorithms in such a way that the development of this type of experiments that can be executed massively provides us with a lot of information, which is quite useful for training models. In this sense, one of the main techniques that is most used to explore the functionality of proteins is deep mutational scanning. In this sense, what we do is generate a new library of protein variants at the level of their respective genes, and we apply selection methods based precisely on the activity of our protein, in such a way that then we would only preserve what would be the variants capable of satisfying the limit that we set as minimally functional. And then here you can see that, of which are the blue variants, only one managed to have a specific combination of amino acids capable of satisfying the function required by the limit that we imposed in our experiment. And so it also happens with what are the green variants or the purple variants. And thanks to this type of approach, we now have relative information that we can compare with respect to a protein that we have used as a seed during our experiment. What is known as the wild type, or the wild type, has sometimes been translated like this. And based on what our seed protein would be, we can calculate metrics of how functional it is relatively. So then, if this blue protein presents a combination of mutations with respect to our seed protein, wherein residue 60, an alanine was mutated into a proline, we found that this functionality metric that we assigned through our experimental methods resulted in a deterioration of its specific activity of 0.33. But it turns out that what would be some purple proteins present a mutation at residue 36, where a serine is mutated to a threonine, causing in this case its specific activity to increase. And so then we come to the construction of this type of graphs, where what we find specifically is how much is beneficial, found here in red, or how much is harmful in blue, the effect of these mutations precisely. And then we find precisely a specific position, and if this position mutates any of these residues, since in this case there is an increase in what would be its protein activity. And so we can find combinations with which we can even adjust mathematical models that allow us to predict what the fitness of our protein variants would be. And thanks to these types of experiments, we have also been able to train quite skilled machine learning-based models in being able to detect, even without having to do these selection and variant scanning experiments. Among them, perhaps other quite notable experiments would be these new protocols that even allow us to measure the thermostability of our proteins in specific domains, using massive approaches that allow us to quantify up to almost 1,000,000 domains, which is quite important, without having to go around purifying each of our proteins and measuring their activity in the laboratory. And we do this thanks to recombinant DNA technology, particularly the complementary form, so that what we do is synthesize our proteins with their corresponding DNA sequences, in such a way that we have both the genotype and the phenotype of our sequence, which would be precisely our protein. That is, then we have, let's say, two ways of representing the protein at the amino acid level and at the gene level. And then, with various strategies, what we do is join them in such a way that we have the protein with its complementary DNA. And then we use a series of enzymes to help us estimate how thermostable our proteins would be. And these proteins will have to bind to a magnetic ball through what is the union with antibodies in such a way that only those proteins that are really well folded will be the ones that will really stick to these balls that we will later proceed to massively sequence. And thanks precisely to these massive sequencing methods, we can approximate a degree of thermostability based on the combination of amino acids that our protein variant has. And this is actually quite intelligent in the sense that we can save a lot of money, because instead of doing a one-by-one test, now thanks to complementary DNA technology and massive sequencing, we can quantify close to 1 million variants at a cost of approximately $2,000, obviously excluding the costs of exogenously synthesizing our proteins. So really this also offers protocols that really give us a lot of information to train our algorithms. And these are also a type of experiments that, although perhaps they have begun to be a little out of use, have been part of the backbone on which we have built our understanding of what protein function is. Because we find that through what is this phenomenon of neutral drift? Since it is the exploration of this sequence space through mutations that are neutral, it turns out that the accumulation of these proteins that are neutral can even be beneficial to be able to generate a starting point to use during subsequent mutagenesis tests, because precisely what neutral mutations do is diversify, let's say, the possible evolutionary routes with which it could be proceed our evolutionary test, and thus explore different functions in this sense. That is, neutral drift assays make it a much more robust system to mutations, and therefore we can explore a sequence space at a functional level that is much more diverse than starting from a protein that does not have so many mutations that are neutral. Again, then, we also begin to cover what this idea of rational design is, and in this case, one of the most recognized tools within the world of protein engineering or protein design, whatever you want to call it, are the Rosetta protocols. This suite of bioinformatic algorithms that do is structurally and energetically characterize proteins in such a way that, based on their interactions based on molecular forces, we can determine if, for example, our protein could form some type of unfolded state, and what energetic conditions based on its interactions would lead it to a correctly folded state. And although this is not a course on how Rosetta works and the hundreds of algorithms that Rosetta has, because everything you can do with Rosetta is really quite extensive, I would recommend that if you want to learn how to use this tool, well, you can take this small course that they developed at the Protein Design Institute, where they precisely give us an introduction to what its implementation in Python would be, because before it was a program compiled in the C++ language or C++, I don't remember, but it behaved in a more friendly way to what the Python ecosystem would be, so that we can then use it even in what are our JUP or notebooks. Even, for example, we can also use what is Pyrosetta, which remember is this Rosetta application, but ported to what is the Python ecosystem, within what is Google Colab. And so you can see that here, under a suggestion from a colleague called Haska, he actually suggests that we run, let's say, this type of code, using in particular these first lines. So what is done is that in your Google Drive session, which is connected together with Google Colab in its entire ecosystem as Google in the cloud, we can signal that what is Rosetta be installed using a memory cache, so that then what is all the weight that this suite of algorithms represents stays in our cache, in our Google Drive. And then every time you are installing or running Rosetta, well, Google will simply access precisely this content that is already stored in your Google Drive, and it will be much easier to execute what Rosetta's algorithms already are. And in this case, if you obviously already know how to work with Python, you will recognize that you have to import the libraries, which in this case is PyRosetta. And here, for example, specifically we are doing this quantification using the empirical Rosetta function. Remember, it has as many physical-chemical terms as statistical terms to assign an energy term of how probable our protein is. And then here we invoke what would be our empirical function called Rev 2015. Later, we import other necessary modules to be able to precisely represent our protein, which in this case is this PDB called 3NIR. And well, in a fairly easy way, we can precisely qualify in energetic terms of the empirical Rosetta function what the state of our protein would be. And I want to put special emphasis on this, because previously installing Rosetta on our computers was really hell. Because we had to download all the code and then compile it on our computers, and depending on how well you can perform in this type of activities. And just like on the hardware that you have available, which is just the compilation of the Rosetta suite, it could last up to even more than a day or 6 hours. And thanks to this caching mechanism, we can simply install it once, and every time we need to run our analyses is stored in the sense. So with this we save a lot of time, and also through the interaction with the Python ecosystem, we can make much easier analyses regarding, at least in my opinion, what its implementation as a C++ library was like. Also to be able to investigate the function of our proteins under the paradigm of rational design, since we have, which is both docking and the molecular dynamics that we previously explored in the class of protein function, where briefly, to summarize, we have precisely the simulation of the physical-chemical conditions over time of our protein, in such a way that here what we observe is, over time, which residues surrounding what is the ligand of the protein are establishing some interactions of some specific type. Something that we are seeing in this type of graph, where we see on the x-axis an idea of what temporality is, and on the y-axis what the residuals would be. And thus we can identify precisely which residues over time consistently establish interactions with our ligand, and subsequently investigate what type of interaction and why they would be established, which helps us to further characterize the function of our proteins in the sense that they are binding to a specific ligand. As you may also remember in our protein function class, we talked about what these force fields are. This way they have to parameterize protein structures through molecular dynamics tests. And force fields really are diverse in their representations, as you might understand. So we have some force fields that are at the level of all atoms, in such a way that we represent it in a relatively static way, and consider what its molecular force interactions would be as well as other statistical aspects in what would result in a function parameterized by a set of parameters. Precisely. We can also represent what would be practically the condition of all atoms, but also giving it a greater range of flexibility by considering in a more detailed way what their electrical charges would be. Through force fields that, in addition to being considered among all atoms, also consider this flexibility due to the polarizability of these electric fields. So now we include even more information that helps us precisely model our proteins more finely at the cost that obviously we have to increase the computational resources to be able to do so, and that perhaps under some implementations it could be restrictive to be able to apply this type of algorithms, for example, to proteins with 100 residues, which are relatively simple, and prohibitive for proteins with 800 residues, which evidently involve a much larger number of atoms. In order to deal with large macromolecular complexes, we have force fields such as those implemented by the Martini algorithm, which in this sense, because they are large complexes, we have to opt for coarse-grained strategies. In such a way that what we are really representing in a general way is practically the backbone of our protein, and in a more simplified way what the side chains would be, thus reducing the number of interactions and parameters that we need to be able to model the interactions over time, and we can even reduce it even further to what would practically be an alphabet of only two representations through force fields that have one more identity of the hydrophobicity of the residues, so that we then practically consider which residues have a behavior more similar to water, and which others have a behavior more similar to oil droplets, that is, that they are hydrophilic and hydrophobic, and thus calculate what the interactions would be in a much more general way. And within this world of force fields, a recent aspect that has begun to be investigated using artificial intelligence algorithms, more specifically deep learning algorithms, is precisely the force fields learned by this type of algorithms, in such a way that we can combine several molecular dynamics that meet certain requirements. For example, same type of modeling, same time interval, etc. Same type of proteins, or perhaps generalize it to practically every type of molecular dynamics that we can gather and normalize it in some way, in such a way that here the most expensive thing is precisely to train a neural network that allows us to parameterize all the information recovered by this type of molecular dynamics, such that then now the neural network is the one that learns to parameterize these interactions. And then once we have our statistical potential learned, well, we can precisely execute our predictions against a new protein. But then what is expensive here is precisely the training, since we are talking about practically a protein that will have, for example, 300 residues, and that these residues will have to be modeled over a long time, and we are even talking about well more than 1,000 proteins, 10,000 proteins, depending on the dataset that we are using to train our complexes, however. This evidently remains an area that is still under constant investigation, and which has demonstrated some quite interesting results in terms of its application to the investigation of protein structure and function. In fact, it has been shown that these types of potentials based on neural networks have great potential. When we look back at the history of what molecular dynamics is, we find that the first dynamics of this type was run in 1977 with a protein that is relatively small with approximately 58 residues, and obviously with a time span that was quite short as it could be, because 8.8 peak seconds later, because we increasingly improve our computer algorithms by applying heuristics, applying other statistical methods, characterizations that allow us to represent coarse-grained proteins without losing so much information of what would be the fine grain, which is their atomic representation. And then we even get to the capabilities of being able to model interactions at the level of quantum mechanics, which is quite refined to be able to know precisely how bonds are being broken, how they are being formed, among other aspects that we previously reviewed in the protein function class. And in this case precisely, what the calculations based on quantum mechanics were quite limited only to what are small molecules of a few atoms, and really somewhat prohibitive to be able to extend to what is a protein of 100 residues. That represents many more atoms than just a few dozen. Later we move on to the development of this type of quantum mechanical algorithms, but now applied to GPUs, in such a way that we can now extend, thanks to this highly parallelizable computation, this type of analysis based on quantum mechanics to be able to model small fragments such as regions of alpha helices, along with other terms that would allow us to know in great detail how they would be behaving over time. But then we only have to reduce ourselves to this small part, and subsequently the algorithm could be developed even further. So now we can apply these quantum mechanical algorithms, but now with a fragmentation approach to our protein. So we are modeling our protein in its entirety, but it is necessary to make certain cuts, because our algorithms simply do not allow us to model the entirety of a protein using quantum mechanics. We would then have to consider other more coarse-grained algorithms, and only perhaps focus on the sites that interest us if we have to make a characterization of this level. However, it turns out that thanks to these potentials learned by neural networks, we can now actually have relatively practical runs of quantum mechanical algorithms on what is the entirety of our protein, as you can see in this case. That even once they have been trained, and again considering both their benefits and limitations, according to the data with which we have already trained our algorithms, they can run precisely orders of magnitude faster compared to what the classical mechanisms of quantum mechanics parameterized by our handmade algorithms would be, compared to what the algorithms parameterized by neural networks run. And in this sense, as you will remember, molecular dynamics analysis really helps us a lot to be able to characterize the behavior of our protein within the cell over time, in such a way that we have precisely what would be both experiments and their bioinformatic equivalents to be able to characterize these behaviors. In some cases, we may be interested in knowing in a more general way what our protein would be, so we can carry out NMR experiments or X-ray crystallography, whose equivalents at the level of bioinformatic analysis would be Monte Carlo analyses or molecular dynamics. But as the dimensions of our biomolecular complex increase, it is evident that we need new strategies capable of dealing with even millions of atoms. So we then move on from these molecular dynamics analyses to other configurations such as the implementation of the SACS experiment, CRIOEM or even CRIOET, when we are talking about large biomolecular complexes, proteins that have up to more than 100 chains within what the complex represents. And in this sense, it is precisely that we are now left with this limitation of quantum mechanics algorithms even using this fragmentation approach, where we simply can no longer model even what the behavior of a complete cell would be. So this is precisely where this type of potentials learned by neural networks jump and shine, because precisely we need force fields that are much more sophisticated that allow us to distinguish these types of behaviors both on a macromolecular scale as well as a super-refined scale to be able to distinguish aspects such as the transition state of an enzyme, among other behaviors that our protein may have. And among them, one of the perhaps most promising models, in my opinion, is this series of models called ORF, which are currently in version number 3, which are precisely potentials learned from training on molecular dynamics data. And that in this case we can see the examples that can be explored here, for example in what would be the study of a carbonic anhydrase, or in this simulation that we are seeing that is actually the spike protein of what is SARS-CoV-2, which is this virus that causes COVID, where we can find that we are actually already explicitly considering the water molecules within the interaction, which allows us to know in much greater detail how precisely this type of protein would behave within what the cell would be in this sense. Like other quite interesting approaches within the world of molecular dynamics, we also have what is the extension of AlphaFold, not only to be able to model these small aesthetic photographs that represent the predictions made by AlphaFold, but to extend it even to what is the characterization of the conformational assemblies that really represent the proteins. So that, for example, here we have an algorithm called AlphaFlow, which what it does is use a series of algorithms quite similar to diffusion models, which in this case are called flow sampling or flow matching, which in summary what they do is the creation of a vector space. And once this vector space has been created, what is done is a linear interpolation as you can represent it here with this purple line, in such a way that during training it is also being provided with noisy structures that in this case would represent the multiple conformational states that proteins can adopt over time. And it is thanks to this sampling of flows and the respective vector field that is being modeled probabilistically that we can sample some states in this linear interpolation. And thanks to the sampling of this interpolation, we can precisely approximate what the results of molecular dynamics would be. That in this case, we observe a small protein that is being modeled for 300 nanoseconds, which turns out to be relatively consistent with the predictions of the AlphaFlow algorithm. And even more surprising in my opinion, it would be, for example, the implementation of this other algorithm called bioinformatics, which precisely takes advantage of everything that AlphaFold 2 learned about its training and specifically adopts. What is the architecture of the evoformer and somewhat decomposes what is the structure of the structural module. And in this case, we take the representations learned at the MSA level. Specifically, what would be our unique representation of the protein that we want to model, along with the pairwise representation, and we coupled it to a diffusion model that precisely allows us to model what are the conformational assemblies that could represent our protein. And they did this in a quite interesting way, training on what are practically the predictions that AlphaFold made, which are approximately 200 million structures, to learn. A first distribution that I would do is, let's say, learn these structural patterns of the proteins that present a higher quality in terms of PLDDT. And once we have a rough parameterization of what the behavior of these conformational assemblies could be like, we do 2 stages of fine-tuning. In this case, fine-tuning, using precisely data from molecular dynamics of up to 200 milliseconds, which is quite a long time when you start to review what this type of experiments is, along with what would be the parameterization provided. By what is the prediction of protein stabilities through an algorithm that they developed in this algorithm called fine-tuning, or fine-tuning. Prediction of properties through fine-tuning, which precisely means that what this first parameterization learned during pre-training would be, now reflects in a better way what the interactions would be over time using these 2 data sources, and which is also quite interesting in terms of the results that we can obtain with bioinformatics, because it runs orders of magnitude much faster than what classical molecular dynamics algorithms would be, such as AMBER could be in this sense. Regarding what these more semi-rational or recombination approaches are, we also practically find what is the recombination of multiple parts to find stability in the interactions, and thus improve their activity or specificity towards some specific activity. In this sense, what we have to do is specifically look for some scaffolds, some 3-dimensional folds capable of satisfying some requirements such as geometric limits. And then we can use algorithms such as PROTLEGO or PRODOMINO, which do is a transplant based on several parameters, based on what would be the interactions of molecular forces, also as other statistical parameters that allow us to identify which regions would be most likely to be transplanted to another protein, so that they could mediate what would be the interactions between proteins in the form of an interface, or even transplant, for example, nucleation centers that allow the protein to adopt its 3-dimensional folding more quickly over time. And so we really, when we look at these protein transplantation strategies, we can find that there are strategies that are very fine-grained, such as the transplantation of what the side chains are. Transplantation at the backbone level, as could be in the case of a spin region, which perhaps in this case we are not so interested in the stability that an alpha helix region provides us, and perhaps we want to transplant it through a completely flexible region or vice versa. Or a much more transplant, generalist, aggressive, so that we can even insert complete regions of folded betas and alpha helices, as well as configure their connectivity, which in this case allow us to obtain a more thermostable protein over time. And in this sense, Precisely what matters to us is knowing the complementarity between protein interactions, in such a way that some molecular interactions are satisfied, such as the electrostatic van der Waals forces, the hydrogen bonds, the hydrophobic interactions between these interfaces, so that their interactions are precisely stabilized over time, and it becomes capable of recapitulating the function that we desire. So that then we can even talk about what a stability design is, where we have our folding energy landscape in this way, and that thanks to what would be this modification of the interactions between pairs of proteins, or even at the level of the amino acid sequence of a specific protein. What we do is make it more likely that our protein adopts its native state within the cell in an easier way with respect to this other energy landscape that is more populated by intermediates that are not necessarily as stable with respect to what the native state would be. And then, by configuring these interactions, by configuring these residues, since we maximize the probability that our protein is applied more easily during what would be the expression in a specific cell. Well, now we move on to explore what evolution-based design algorithms are, where again the idea is to exploit sequence diversity information from multiple domains of life, or perhaps from a specific family of proteins. And let's turn to see the hypotheses that this type of evolutionary information suggests to us. In that sense, one of the first approaches most used during the first years of protein design was what is known as consensus-based design, where what we do is start from the hypothesis that precisely the amino acids that are really the consensus within an alignment could contribute more to thermodynamic stability with respect to others that are not precisely this protein consensus. Here we observe what is a relatively small alignment. The desirable thing would be to have quite large alignments, diverse both in sequences and in sequence diversity, in number as well as diversity, and then we can observe that there will be some residues that are not completely conserved, and that perhaps there will be others, for example, these three residues, that they are conserved even in the entire alignment, something that is even relatively rare to find when an amino acid is 100% conserved. We usually find conservation ranges greater than 95%, but perhaps then, when finding in this alignment that they are conserved up to 100%, we can think that it is because they are fulfilling some important role within our protein, be it at the folding level, at the structure level, or even at the function level. And then if I have a sequence that I want to improve its function, I can observe that in my sequence it turns out that here I have a lysine where perhaps an arginine should go. So perhaps I can take what this arginine is and make a point mutation to see what it would be causing in the phenotype of my protein, and see if, for example, it is having an effect that increases its thermostability among other aspects. In this sense, we must know very well what the biology of our proteins is, so that, for example, here we have our favorite protein for this course, which is the class A beta-lactamase TEM1, and then we can do a search for homologs in multiple databases to build our alignment, which in this case we are talking about 7,000 homologs that, as you can see, are quite diverse at the level of sequence similarity by this margin from red to blue. And then what we can do is map this conservation information onto what is our T-mono beta-lactamase structure in such a way that we can generalize the idea of conservation in what would be three large regions, as you can find them here in the form of concentric circles, where it turns out that precisely the residues of the first layer or shell, which we can really think of proteins as onions, perhaps will be related, and evidently this translates into them being highly conserved in this alignment because they are quite relevant for what would be the catalytic function of our specific enzyme. This is precisely in the case of this protein, which is an enzyme. But you will need to know practically the biology of the protein that you are modeling in order to be able to associate some type of function related to this type of residues that are highly conserved. we also now find a second layer within our molecular onion, where perhaps they are not directly involved in what the catalysis or binding mechanisms are. But what they do is contribute by reconfiguring the neighborhood of the active site to be able to precisely establish molecular, electrostatic interactions, among another set of interactions, such as allosteric regulation networks that allow carrying out the function that the protein performs within the cell. And finally, we also have what would be the third layer of our protein, which are really less likely to affect what the activity is. But since it is precisely the layer most exposed to the solvent, perhaps they would be facilitating some aspects such as its foldability or stability, thus avoiding some behaviors such as aggregation. Since remember, we really want a protein with electrical charges that are relatively uniformly distributed and that are not concentrated in some specific place that would cause our protein to be destabilized. This again will depend a lot on the biology of your protein. And so, when you want to apply this type of approaches based on consensus design, obviously you have to consider this idea of the layers of onions to know which amino acids would be involved. Or even if they don't know anything about the function of their protein, they can obviously look at the most conserved residues and make point mutations within each of them, and thus evaluate through experiments what functions they would be fulfilling within the cell based on the results they are obtaining from those experiments. Now when we talk about what precisely these ancestral reconstruction algorithms are, we are really talking about a set of quite beautiful algorithms in my specific opinion, where what we do is start from precisely a multiple alignment of sequences. And so we now run phylogenetic analyses to be able to precisely reconstruct a phylogenetic tree that allows us to have hypotheses about how evolution occurred within our specific family or superfamily. And then, from what would be the internal nodes of our phylogeny, we can apply algorithms that what they do is approximate what sequence should represent a specific node, in such a way that what we would be approximating is the sequence that would correspond to a specific ancestor. And if we have our phylogeny quite well characterized, perhaps even with some sister and rooted group, well, we can even have an idea of the temporality of our protein family, how many years of evolution there have been, and how its function has really been configured over time. So what we have here is a mapping, for example, of some specific activity, with respect to what would be precisely the location of these nodes in our phylogeny, which we could assume that, being gozal nodes, they could represent ancestors from several million years ago of evolution. And we can see how perhaps some functions could increase as evolution occurred, while other functions were left aside. And then find, for example, what set of mutations would have been relevant to look at the proteins that we currently observe, and that presented the phenotypes that we observe, and thus make better characterizations and hypotheses of how evolution would have occurred over time. We can see this quite well represented, exemplified with this article, where they once again explore the enzyme most dear to us, which is beta-lactamase T1. And in this sense, obviously a multiple alignment of sequences has to be reconstructed, of homologs belonging to what is the class A beta-lactamases family, or in general even to the beta-lactamases family in general terms. Well, serine beta-lactamases, which are precisely the beta-lactamases that correspond to the folding of class A beta-lactamases. And in this specific case, what we observe is an idea of the temporality of each of our protein variants that are called P, NSA, AG, NSA, among other acronyms, based on, for example, measurements of the denaturation temperature at which these types of variants stop folding, where it turns out that what would be the most ancestral sequences within our phylogeny, which would be this set of proteins here, are denatured up to approximately 70 to 80 degrees Celsius, compared to what would be the modern variants of beta-lactamase, such as TEM1, which are denatured in a range of approximately 60 degrees Celsius. And so we can, for example, look at what the conditions were like in that primitive Precambrian land, where we are really talking about high-temperature conditions, which perhaps would coincide with the fact that these specific variants have greater thermostability compared to what would be the more modern TEM1 beta-lactamase. And we can even map what its function would be based on what it is. Its affinity for certain beta-lactam substrates, such as the antibiotic seboxmin, with respect to a more penicillin-type antibiotic, such as benzylpenicillin, where we found that its activity of the set of ancestral proteins really had a more generalist aspect, where they really were not so good, but that later, in terms of what the catalysis of this type of penicillin compounds would be, it really went from what would be a more generalist aspect to a specialist one. And this is how we can characterize how evolution has occurred over time, and have hypotheses that allow us to assess how this phenomenon would have occurred. And even this is so interesting that there are databases like this one called Revenant, where we find precisely proteins that have been produced, resurrected with this ancestral reconstruction approach, so that approximately 211 proteins are compiled, of which even 55 have a structure deposited in the PDB. So we could even do quite interesting evolutionary analyses, considering that this is a fairly limited set of proteins, but turn to see what aspects they share with each other based on the time scale to do more evolutionary analyses. Even when we turn to see what type of developments or applications there have been within what is the ancestral reconstruction of sequences with respect to deep learning algorithms, we find that what these types of algorithms are, we can really use them as a kind of data augmentation that allows us to model a landscape of functionality or fitness landscape, make it much more navigable, and therefore improve our predictive capabilities of protein functions. Because when we do the type of ancestral reconstruction analysis, we really need to have an alignment that becomes large in terms of sequence diversity as well as the number of sequences. However, this is not always the case. As you know, there are some proteins that are more represented in the databases compared to others. And in this sense, for example, here we have a phylogeny that was repeated many times to see what the most probable configuration would be for a set of proteins called phosphotreaterases, which we have here marked by the acronym PTE, that even this phylogeny was reconstructed by a total of 300 sequences, where thanks to what are precisely these ancestral reconstruction algorithms, we can sample the internal nodes to generate this idea of increasing the data available to explore the fitness landscape of our proteins. So that when we represent the sequences of our proteins by what would be the embeddings of a protein language model, such as ESM1B, and subsequently reduce the dimensionality of these embeddings to only 2 dimensions using the Disney algorithm, we can find that actually, the diversity represented by our sets of proteins that we recover from the database is quite regionalized in this space. And when we show what precisely the internal nodes are within our phylogeny, we explore a sequence space that is relatively more diverse with respect to the set that we find in nature. And so the idea of this is that we want to model precisely a fitness landscape that becomes much easier to navigate in order to find precisely some optimal ones, depending on the function that, in this case, our proteins are performing. And then we can integrate the information coming from the plausibility of our phylogenetic trees into what is, for a change, a transformer architecture. As you know, the transformer has actually found applications in practically any domain of science. And then we practically train this transformer with what is this entire enriched set that, remember, we started with only 300 sequences, and thanks to this increase in data, we now have 10,000 sequences. And then once we train our transformer, which is more of the BERT type, as you can see here, which is masking our sequences, we can recapitulate its embeddings and then generate embeddings at the protein level and use them to be able to train some regressor that allows us to approximate what would be the fitness of our proteins. And this is precisely what we are observing in this bar graph through this metric called Dirichlet energy, which practically what it does is an approximation of how navigable or how rough or complicated this representation of the fitness landscape is, in such a way that higher values correspond to fitness landscapes that are much more complicated to navigate, and lower values represent a fitness landscape that is much easier to navigate and therefore from which we can make better predictions. And when comparing the type of fitness landscape that can be modeled with other protein language models such as those of the ProTrans family, others such as UniRep, directly ESM1B, which are even models with 650 million parameters or even 3 billion parameters. Well, it turns out interestingly that what is their trained transformer, which in this case is called LACE, has practically a few hundred parameters and not thousands of parameters, since it turns out that it builds a much more navigable fitness landscape, which translates into more accurate predictions of what the fitness landscape would be. And so you can see that what this combination of ancestral reconstruction algorithms is, together with what is the transformer architecture, they can help us model or represent in a more enriched way the diversity of proteins at the level of their sequences. Now we explore what experiments are based on directed evolution, which, as you already recognize, is precisely this idea of first generating a fairly diversified library of genes. And in this sense, we can do it through several protocols. Obviously, the process of choosing each one of them, because it is our job to be able to precisely design an intelligent protocol based on what we want to improve in such a way that we have, for example, mutagenesis that is completely random, where a PCR is used that is prone to making errors, and then various mutations are inserted into our genes because our polymerase is bad in this sense. And then we evaluate precisely which variants could be good in this sense. We can also find, in the event that we only want to improve a specific region of our protein, what would be directed mutagenesis protocols, which in this case only concentrate on specific regions that are complementary with complementary or recombinant DNA in this sense, and then only make relatively targeted mutations to a specific section of the gene for our protein. Or we can even do perhaps slightly more aggressive approaches, such as completely aggressive recombination between pairs of sequences, to see what results we could obtain at the gene level. And once we have generated a fairly diverse library, we have to explore the best candidates based on what the function of our protein would be. And in this sense, the screening and selection protocols will really depend a lot on the function that you want to improve and that your protein plays. So that, for example, we can find proteins whose phenotype is quite clear, such as the green fluorescent protein, which in this case its phenotype is very distinguishable. A protein whose activity is not as good is not going to bloom as much compared to a protein that happens to be blooming quite brightly. And then we can run it through some experiments, such as laminar flow cytometry, and then detect using a laser beam which one is flourishing to a greater degree in that sense. Or in the case of a protein that, for example, provides resistance against antibiotics, we can possibly grow bacteria that have these variants and apply different concentrations of our antibiotic. For example, 250 micrograms per milliliter, 1,000 micrograms per milliliter, or even more aggressively, 2,500 micrograms per milliliter. And again, see which variants were successful, and then mass sequence them and calculate these relative metrics of functionality or fitness in order to generate the best candidates that will serve to inoculate the next stages of this iterative cycle of directed evolution. Finally, now we have to amplify our sequences, and here in this sense. Perhaps the protocols used in PCR are precisely the ones that are used the most, where what we have is practically an idea of Bates, where we practically have different protocols such as the deployment using phages of what would be the deployment with ribosomes, even with RNA, or even with what would be yeasts. This will also depend again on the type of function that you want to quantify, as well as, let's say, the entire theoretical context of your protocol that may allow you to quantify a greater or lesser number of variants, depending also on the economic resources that you have at hand to be able to do this type of assay to show your protein variants. And thus, we then comply with what would be the cycle of directed evolution of diversification of gene libraries. What would be its selection and the amplification of variants in such a way that what we are really doing is, let's say it in a cartoonish way, having Darwin in a test tube, that is, applying these principles of natural selection but in the conditions that we can replicate in a greater or lesser way within the laboratory, in such a way that if we observe the diversity represented by the proteins that we can observe in nature, whatever that means, we can expand our horizons thanks to the application of these types of experiments based on directed evolution. However, when we look at the entire possible sequence space that a protein can represent, it really is enormous, and these types of protocols will only explore a neighborhood relative to the natural proteins that we observe. And in fact, this finding was so relevant for the improvement of proteins that the Nobel Prize in 2018 was awarded to Francis Arnott, who was the biologist in charge of developing this type of protocols based on evolution, as well as to George and Georgie, who designed these first variant protocols to be able to precisely do this type of phage display assays and thus replicate our proteins. And then what would be to make our cycles of directed evolution more and more robust and improve, depending on the selection criteria that we apply, the function of our specific proteins. However, as you may think, what these directed evolution protocols really are, they can be relatively limiting, in the sense that they are only showing a small fraction of the entire sequence space. Here again we have this protocol where diversity is generated. We quantify a fitness, and then we use the best variants to inoculate the following generations, and we repeat the cycle, which really translates into an exploration of sequence space that is being more in nature as a local exploration. And in this sense, there has been progress in the incorporation of machine learning algorithms, where what we are, well, we can really couple within this protocol the idea of training a model. In this case, it could be a logistic regression, even something more sophisticated such as it could be a regression based on Gaussian processes that allow us to have even a degree of uncertainty about how much fitness our variants would have, and then select in a more intelligent way based on what our algorithms suggest to us, which would be the best candidates to be inoculated in the next generation. So what we do is start from a much more diverse library, here represented by green dots, and then thanks to what would be the ability of our regressor or classifier to be able to detect variants of interest, we can more easily identify what would be the variants that improve fitness in a more consistent way. In this case, what we do is explore the sequence space in a much more diverse way than the classically directed evolution that we have done until now could be. And even when you look at the type of protocols based on machine learning applied to directed evolution, they have really diversified quite a bit. However, these types of AI-based protocols may not necessarily really be good for all proteins, because these types of algorithms improve our abilities to model the fitness landscape, when, for example, the fitness landscape is quite complicated to navigate or is quite rough. Also when, for example, our fitness landscape is really populated by many variants that have no activity or even their epistasis is much more complex, in such a way that even the change of a small mutation can be completely deleterious to even what would be the three-dimensional folding of our protein, and not only its function. So then, for example, we have this decision framework where we can ask a question: is it easy to navigate or is it difficult to navigate? In this case, if it is easy to navigate, then we can simply adopt these approaches that are relatively simpler, where we can make use of these algorithms such as MLDE or ALDE, where practically what is done is to train precisely a regressor or a classifier, or even use active learning approaches, which is a constant way of training based on the results that we have in our experiments. But when the fitness landscape is much more difficult to navigate, well then, now we have to ask ourselves the question of whether our predictors, for example those provided by protein language models, among others, are good at predicting the fitness of our variants, even under a regime known as zero-shot, where we do not have to do fine-tuning, but can simply adopt the model that the developers uploaded to code platforms such as GitHub or GIMPface. And then, if the benchmarks really suggest that it is good to be able to predict the behavior of our specific protein family, then we can choose to either build libraries that help us do fine-tuning or enrich in another way precisely to be able to improve our predictions and subsequently see what type of other variants of algorithms based on directed evolution coupled with machine learning to apply. Such as this could be the FTMLD or the FTALD, which are several variants that have been developed within, for example, Francis Arnold's laboratory, together with other colleagues who are trying to increasingly improve the capabilities of directed evolution protocols. And we also have other quite interesting approaches, such as this algorithm called EVOPROT-GRAD, which practically what they suggest to us is to mix the modeling capacity of these large protein language models, such as those of the ProteinMPNN family, those of the SM family, among other models that you can find, as well as finding models, other algorithms that we necessarily have to train, which in this case are algorithms from a supervised learning regime, that we can use architectures, become convolutional, multi-layer perceptron, or even transformers if the data allows it. And so now we have suggestions or hypotheses of what would be the best variants used with an unsupervised approach, represented by protein language models. With respect to a model that is supervised, represented by the model that we train with the purpose data that we have. So what EVOPROT-GRAD does then is parameterize the conjunction between the hypotheses provided by both the unsupervised and the supervised models, and thus find which variants, depending on which residues are being mutated, would be the ideal ones to be able to precisely improve the activity of our proteins. And well, once we have explored what these main paradigms that were previously used for protein design are, we will now proceed to the topic that interests us in particular for this class, which is the topic of protein design but based on artificial intelligence algorithms. And in this sense, within the two main topics that represent this class, we will address this first one, which is precisely the topic of learning representations, which precisely can be used for a posteriori tasks once this type of information-rich representations has been learned. And obviously, first we must recognize the type of task that we want to carry out in this sense, because there is great diversity, and each one can be adapted to a greater or lesser extent to our specific objectives. For example, we have what are classifications at the whole protein level. For example, if a protein would correspond to some type of folding similar to Rosman, similar to a Tim barrel or similar to a beta-lactamase based on, for example, its sequence, without having to look at its structure. We also have other tasks that can be residue classification where we can identify, for example, those residues that would be establishing interactions because they are the binding sites for a ligand, or perhaps those that carry out some catalytic reaction. We also have self-monitoring tasks, which in this sense learn information-rich representations that can be used later. And in this case, it is quite well exemplified by what AlphaFold is through the representations learned by the evoformer, which is precisely the pairwise representation and the MSA representation. This is achieved by hiding certain segments, and the algorithm, in this case a transformer, becomes capable of identifying which residues are most likely to satisfy that particular mask. We also have tasks that are at the level of interaction between residues, so that we can specifically identify which residues could be establishing some interactions at the protein and protein level. Or we can even elevate the task to the next level, which would imply the complete interaction of proteins with proteins, thus possibly establishing the interaction and configuration of multimedical complexes, for example of proteins with high symmetry. We also have other tasks related to what would be the search or retrieval of information where, for example, we can ask ourselves if a sequence or a structure can be similar to other structures deposited in a database. So then we have to have an algorithm capable of scaling based on the elements contained in said database. And finally, we also have other algorithms related to what the ranking would be. For example, being able to rate what type of molecule or ligand would be more likely to interact or perhaps be more easily degraded by a protein that interests us. So, you can recognize the different modeling tasks that you can adopt for what would be the research of a specific protein. And even about all these specific tasks, they can be diversified even more. For example, when we adopt what would be a modeling strategy at a structural level, we can precisely classify at the level of secondary structure, similarity at the family level, at the domain level, of folding among multiple aspects. The same thing happens when we use a sequence-level perspective, where we can classify small motifs or signatures at the sequence level, some regions that could be involved in allosteric regulation, or perhaps precisely in what catalytic activity is. We also have other predictions that are more of a genetic nature, where we can, for example, characterize what the effects of single nucleotide modifications would be. Or even detect, for example, which ones would be candidates to deal with diseases such as cancer. We also have other tasks related to what would be the prediction of physicochemical characteristics. For example, the behavior around what would be the enantioselectivity of different compounds in relation to proteins, the stability of proteins, among many other aspects. So that really in order for us to choose some representation, obviously, we have to think deeply about the nature of our data. If we are working with sequences, with structures, how many of these we have, as well as what quality our dataset is. The type of people who could run our algorithm to analyze your data are questions we also have to ask ourselves. Subsequently, once we have analyzed the nature of our data, we have to designate what would be a classification task. And that is done, for example, at the regression level, classification, and even at the level, for example, if it is at the residue level or at the whole protein level. And finally, and comprehensively above all, we have to consider the computational resources we have. So that, for example, it is not the same to develop an algorithm that has a purpose more for an academic environment, where usually, for example, what the algorithm's inference would be may not matter as much. Regarding what would be its deployment as a product at a business level, where obviously what the inference would be matters a lot, because it would have to scale even to queries of thousands per second, or perhaps even millions per second. Because it would perhaps be serving a market with a large sector that is interested in using our product, so then also thinking about the computational resources and how they will be executed by the community that will make use of the algorithm is one relevant variant to be considered in what would be the training of our algorithms, because there are some that can run faster than others. There are some that can also better model some property compared to others, but it is the set that matters to us to be able to train a good algorithm capable of dealing with the problem that we want to solve. And in this sense, when we turn to see what the diversity of representations that we have for the same object, which would be, for example, proteins really are, we actually find multiple scales. And within each of those three scales, we also find multiple resolutions. For example, When we talk about representing a structure, we can really represent it in multiple facets, such as at the level of the three-dimensional coordinates of each of the residues, or even not only of the residues through the characterization of the backbone, but now at the level of what their atomic positions would be. Or even perhaps generalize it and only characterize what the surface would be in a more volumetric way. In the same way, for example, when we analyze the sequences, we can consider what would be a single sequence, but obviously analyzed by thousands, or the set of sequences seen as alignments, which is evidently one of the bases of success of algorithms like AlphaFold. We also have another facet where we can explore what the characteristics of our biological objects would be. For example, if they are part of a biochemical pathway, what physicochemical properties do they have related, for example, to their charge, the distribution of said charge, among other aspects. And so you find that there really is great diversity, and within each of these facets of representation, there's also a greater or lesser degree of resolution. So this is precisely what we see in this new diagram, where we can have some representations that can be more appropriate to an atomic context, both at the sequence level and at the structure level. Later, we move on to the next level of protein representation, where it could be precisely the identity of the residues, or for example, what would be the beta factors of the protein structure, or even elements that would be representing the protein as a whole. So we talk, for example, about composition at the level of secondary structure, among other aspects. That finally we can even represent it down to an organism level, where we can precisely choose metrics such as growth under specific conditions such as temperature, or perhaps some limitation given by an antibiotic, among other aspects that perhaps we can also encode so that our algorithms are capable of learning this type of information, based precisely on the training data that we are providing. Something that is quite irrelevant to think about because really the way in which we are representing our data is the way in which the algorithm will be able to do its searches and learn representations that are useful to make its predictions. In this sense, here we address the main ways of representing proteins, both at the sequence level and at the structure level. For example, we have some descriptions that are global, where we can use libraries such as BioPython or ProAdLearn, which in summary, what they do is give us a characterization at a statistical level, for example, of some aspects of the protein sequences, such as some statistical metrics related to what is zoentropy, the presence of some signature at the sequence level, among other physico-chemical characteristics, such as this biochemical consideration index called Y index, which you can investigate in more detail what it means. But in summary, we can precisely represent our proteins as scalar values, or even as vectors provided by this type of physico-chemical descriptions. We also have another representation that is precisely at the amino acid sequence level, or at the nucleotide level, as it could be represented in this scheme, for any general sequence, where this is actually one of the most widely used inputs by most deep learning algorithms to represent proteins by default. So what we are really talking about here is a representation also known as a one-hot type vector, which really does not have a direct translation into Spanish, which would perhaps be like raw encoding of the sequence, where what we have is really a representation of our sequence in the form of a matrix, where you can see that on one axis we have, for example, the possible nucleotides that we are trying to model, and on the x-axis, what would practically be the identity of the amino acids that correspond to our sequence. So that if an adenine nucleotide is found in our sequence, then a 1 is drawn in our matrix and the rest of the values are left 0, because this is the identity that X is adopting, our first residue. And this same extends to the rest of the residuals, having a binary identity matrix that should be as large as the largest sequence that is represented within our dataset, such that we then have to, for example, override or extend what would be the identity of these zero-filled matrices, when the maximum length of the protein integrated within our dataset is not satisfied. We also have other representations, such as at the graph level, which in this case what it does is take a protein, for example, its structure, and identify some residues that in this case could be involved in some type of interaction, or perhaps what would be the contacts at the three-dimensional level, either through hydrogen bonds, through covalent interactions with the peptide bond, among multiple other types of contacts that can be established within a protein. And based on these contact determination criteria, precisely define a graph. Such that then, for example, a residue based on a distance, for example 8 Armstrong distance, we can consider that it is physically establishing contact within the protein in its three-dimensional version with another residue that is precisely within that radius of distance. And so we are encoding an idea of geometry within what would be the representation of our graph. We also have more three-dimensional or volumetric representations, where we can use libraries such as Boole, where precisely volumetric approaches are used, or more specifically such as these ideas known as voxel, which is practically the creation of a three-dimensional mesh where each of the cubes that correspond to our mesh correspond to a defined volume of our protein, and then each of these cubes, since they would actually be characterizing several attributes such as the volume contained in said segment, the charges, the type of interactions, among other aspects that only correspond to said specific voxels. We also have other representations such as these, which is only at the level of what is a mesh of the three-dimensional surface, where perhaps we are interested precisely because they could be, well, relevant to the interaction that could be established between pairs of proteins. And finally, we also have what would be a holistic representation of what would be the entire protein, where we can represent it in its entirety as a general graph. And finally, we have what are the representations learned by algorithms, where what we do is train some supervised or self-supervised, or even unsupervised, learning algorithm. And what would be the representations learned throughout the different layers of said model? They can serve to a greater or lesser extent to be given as input to another algorithm, something known as transfer learning, and thus predict some properties at the level of regression or classification. For example, here we have some representations that can be established in what would be a protein-ligand complex, where what we have is, well, in the neighborhood of what the ligand is, a geometric mesh defined here by several polygons, in particular a triangle, where we can, with this representation, adopt a more geometric approach. But we can also recover physical-chemical information, such as what the presence of charges would be in terms of electrostatics, as well as the presence of, well, that side chains could serve as electron donors or acceptors, or even for their hydropathic behavior within the cell. So then we can even represent physical-chemical properties, which we can obviously add, and together they would represent a kind of fingerprint specific to this region of the protein surface. And in this case, we also find that this fingerprint, well, obviously it can have a certain complementarity with other proteins, which obviously our algorithm should be able to learn this type of interactions, in such a way that then these two fingerprints are complementary, corresponding to the idea that these two proteins would be interacting to establish some type of protein complex. When we talk at the level of molecules, ligands, it is clear that we also have different representations, and each of them recapitulates different types of information. For example, SMILES, which is precisely the type of representations used in the AlphaFold3 algorithm, we could say that it is another representation of the textual type, where what we do is represent what the molecule would be. And in terms of their links through a text chain, in what would practically be atoms of carbon, nitrogen, oxygen, among others that actually make up the biomolecules. We also have other approaches to represent molecules, such as structural approaches, which mainly represent proteins as graphs. And then we have their corresponding vectors, which we can add depending on the degree of resolution that we want to give to our graph. We also have other characteristics, such as representations based precisely on different properties. For example, it is very common to find what Morgan fingerprints are, or molecular fingerprints, which what they do is characterize the atomic neighborhood of each of the positions of the atoms. So that, for example, at a first level, when we look at the first bonds established by some type of atom, well, we evaluate the identity of said elements, which in this case, for example, when we look at this nitrogen, it is establishing contacts with carbon atoms. So this connectivity information is also information that can help us make predictions. And then we can even extend the radius of our interactions in turn to see precisely the type of atoms and their corresponding elements that would be establishing said interaction. And thus add more and more characteristics, finally representing a binary vector, where we can encode the identity of a molecule based on what would be the identity and contacts of each of its atoms. And finally, we also have other types of representations, which are more physical-chemical, molecular, volumetric, which include both local and global properties, such as dipole-dipole interactions, or the momentum of these molecules over time. So actually choosing a representation at the molecular level also determines the type of information that can be learned by our models. And although this is not really a cheminformatics course, here they have different ways of representing small ligands precisely. We have precisely this idea of fingerprints, where here you can find a great diversity of mechanisms by which we can represent them. Where perhaps one of the most widely used libraries to analyze this type of cheminformatics analysis is RDKit. But we also have different descriptors. We also have representation at the graph level, or there are even applications of transformers to the world of cheminformatics. They would also be learning this idea of embeddings, and thus represent the identity of these compounds in a relatively information-rich way. And in general terms, there are also many libraries to represent what chemical compounds would be. And in this case, perhaps MALFIT would be an interesting library to explore when you really want to focus your projects a little more on what would be cheminformatics, and perhaps not so much on what would be protein design. Here we will now evaluate the importance of choosing a good representation, because really from these representations, we can solve problems that previously perhaps had not been able to be solved, and that perhaps we thought could not be solved. In that sense, what we are observing on the screen is a sequence alignment between two particular homologs that are separated by approximately 1 billion years of evolution, which is a protein that is ubiquitin, and another which is another protein called SUMO3. And in that sense, when we aligned our sequences, we found that they barely have a sequence similarity of 16%, where you can find here that these residues joined by a black line are really the few residues that have been conserved throughout the thousands of years of evolution. So when we do a search using algorithms like BLAST, you now know that finding a homology signal actually becomes very unlikely when sequence similarity drops below 25% similarity. Because then we're talking about this twilight zone, where we really have no certainty or probabilistic certainty, however you want to put it, that these two proteins are homologous. But then, what if instead of representing proteins as an alphabet of amino acids seen through their residues, we represent proteins with a new alphabet, but now it is not at the residue level, but rather a structural alphabet. And this is precisely what the Foldseek algorithm does. It will also represent the proteins with a 20-letter alphabet, as could be the alphabet we use for canonical proteins in general. But in this case, It is an alphabet learned by a deep neural network. And in this case, by now representing with this structural alphabet and not an alphabet at the sequence level, we find that the structural similarity is 54%. And it is quite clear when we look at what the structures would be that they are really homologous, since they are quite similar structures in the type of three-dimensional supplement. And obviously, this is reflected in the type of coincidences, where we observe here several black bars that are present in comparison with what would be the similarity at the sequence level, which really helps us to more easily detect the possible homologous candidates that any sequence could have. And then you might wonder how Foldseek can build precisely these types of structural alphabets. And this is quite interesting because here what they did is take a set of proteins that are practically the proteins deposited in the PDB, because they are the only ones that we have with their well-characterized three-dimensional structures. And they are not predictions made by AlphaFold. And then we can define certain limits of interaction between pairs of residues in such a way that we establish an idea of an atomic neighborhood, which really is an atomic neighborhood, established by different quantitative characteristics, and then our algorithm, which in this case is a type of autoencoder called variational autoencoder, but which also, instead of following a continuous distribution, uses certain adaptations so that they are quantized and can be used to predict more discrete values such as 20 specific letters. Well, then it is our autoencoder that is in charge of learning representations through what would be the autoencoder of being able to encode and decode what would be our atomic neighborhoods, such that then this is precisely what we are observing in this graph, which in this case is an analysis similar to what is the reduction of dimensionality from the embeddings of this type of autoencoder. So, since we are forcing the algorithm to represent the great diversity of possible atomic interactions in only a 20-letter alphabet, we precisely find these projections in only 2 dimensions. Where here what we find is this idea that a set of states that the autoencoder is capable of representing through its embeddings. We can assign it to a letter, which in this case, for example, would be K, which does not necessarily mean a residue, but rather means a structural state. We also have other atomic neighborhoods that we could assign to what the letter W is, and thus practically to any other letter, such as the letter A, the letter H, the letter T, where it is the definition of its structural neighborhood, based on the type of interactions and physical chemistry, contain that the identity is actually established to identify said letter. And this is something that the autoencoder actually learned through this process of building and rebuilding. And what precisely is it that we see on the right? We really can't observe great detail of this, because really here what we are observing are the coordinates of the backbone. But we then identified that the autoencoder is capable of representing the structural, or rather atomistic, diversity of protein interactions, seen only as an alphabet of 20 letters. And the importance of Foldseek is such that thanks to this type of algorithms, a new way of analyzing what structures would be on a massive scale was opened. Because we did have algorithms that allowed us to compare protein structures, but they were not so fast to be able to scale to these scales of millions or hundreds of thousands of structures. So when we see the performance, the level of how fast Foldseek is in relation to these algorithms such as TM-line or DALI, we found that Foldseek is between 10 to 4 to 10 to 5 times faster than these structural alignments, which precisely makes it possible for us to scale this type of analysis to levels of hundreds of thousands or even millions of proteins, or rather, protein structures. And this evidently served as inspiration for other algorithms that appeared after Foldseek, such as this one called reseek, which tells why we limit ourselves to an alphabet of only 20 letters, determined by an autoencoder. We can even extend the richness of this alphabet, in this case with an alphabet of many states, which here I really don't know how to read this number, but you can see that it is counted in millions. So now you have to find a relatively optimal way to actually represent this alphabet, and thus assign some determination of homology to actually compare the structures. And it is thanks to Foldseek that what would be the clustering analyses of the AlphaFold database, which is made up of approximately 200 million protein structures, could actually be carried out. So these would be the first two studies that, thanks precisely to this algorithm, allowed us to identify clusters at structural levels that could even represent new three-dimensional folds, as well as characterize which types of folds are most abundant, and that would even be expanding their diversity at a structural level. We also have other studies, such as this one, that we already explored in the AlphaFold class. We're using the protein structure classification scheme provided by CAD. We can characterize what would be all the possible domains contained in the 200 million structures predicted by AlphaFold, in such a way that, at least within the scheme of what the CAD is, we found that when comparing with all the possible counterparts within this scheme, there would perhaps be a probability that there were approximately 7,500 new foldings that would be pending to determine if they really are new foldings through experimental analyses, or much more refined analyses at the level of homology analysis. And finally, thanks to this type of algorithms, we also have structural databases that are massive, where what we really do is recapitulate what the AlphaFold database is, composed of approximately 215 million proteins. And we also adopted what is a database that is even 3 times larger than AlphaFold, composed of 606 million structures predicted by another algorithm called ESMFold, which we will explore later. And then once we have the entirety of this, we're talking about something like 800,000 protein structures. So in order to really identify clusters, well, we have to use algorithms that allow us to really deal with these quite massive amounts of data. So then, at least within this first preprint draft, what is done is to use their sequences, and using an algorithm similar to BLAST, which in this case is called MMSX, we clusterize all the sequences at an identity level of 30% sequence similarity. And then we get a total of 187 million clusters at the sequence level. But once we discard perhaps only those predictions that have greater or lesser quality, we retain a certain number, which we now have to cluster at a structural level using Foldseek, actually identifying something like 72 million structural clusters. Therefore, we can turn to this database when we are doing analyses at the level of structural biology, and simply with what would be a structural blasting carried out with Foldseek, perhaps more easily find homologs defined at the level of their structure and not at the level of their sequence, which would allow us to make much more diverse datasets because the structure is being considered in this sense. Well, now we cover what learning representations is, but created by these great language models. And in that sense here you can see multiple applications of transformers in general to the world of biology, where we really find transformers at the level of genomic scale, transcriptomics, proteomics at the level of chemistry, or even at the level of a type of analysis known as single cell, where there really is a great diversity of the type of transformer architectures, but we can really represent them by what would be the 3 main variants, which would be the original transformer version, that is, this architecture encoder, to a decoder, or only those intended for what would be the decoder or only the encoder, and then precisely adopt what would be a behavior more of the representation learning type, as it would be with BERT, or more generative, as it would be with GPT. And obviously, we have a great extension of the applications that there can be for this type of large language models. But since we are talking about proteins, it is clear that we are only going to focus on what is the application of large language models for what is the representation at the level of protein sequences. And this is a topic that we already addressed previously in these two classes regarding transformers as well as AlphaFold so well, if you don't really know what a language model is. Well, I really recommend that you review these two classes before continuing with this part of the class, and really hear their adaptation to how they are used to really be able to model proteins. As you will remember precisely from our class on what transformers and large language models are, we can say that we have two main variants of types of transformers that can be used. In this case, we have variants that are trained with what would be the prediction of the masks. That is, we hide residues, and then through training the language model must be able to identify which residues have a greater probability of being assigned to said residue based on what the entire sequence would be, or we also have what would be a more autoregressive or causal modeling approach, where in order to predict the state of an amino acid, we have to consider the state of all the amino acids that precede it, and thus practically guess what would be the next most likely amino acid to satisfy said sequence. And each one has precisely its pros and cons to be able to model some phenomenon that you want to study. We also have to train differently at the level of, for example, their loss function. They are also more or less sensitive, and some can be used to precisely generate proteins in an easier or more intuitive way, while others have the ability to generate information-rich representations, such as BERT-type models, to be able to use this transfer learning approach. And in each of these cases, we have some examples such as the protein language models of the SM1 or SM2 family. And in the case of autoregressive models, we have others such as the PROGEN or PROGPT algorithms that we will review later. So in this sense, in order to cover the diversity of language models, we are first going to focus on this type of protein language models with a BERT-type architecture or with a masking-type modeling task. These types of language models, as you now recognize, what they really do is learn to represent protein sequences as information-rich vectors, which we usually refer to not as vectors, but with a new word which we call embeddings. And what these types of BERT-type models do is start from a database of millions of sequences, and in short, we have to do the typical preprocessing for any machine learning task. And once we have our original proteins, we will proceed to mask them like any other BERT-like task. Once our proteins are masked, then it is the task of the language model and its architecture through what would be its last function, among other aspects, to predict the identity of said residues based on how likely it is that they correspond to said mask. So that, for example, when we compare these residuals, which is GAMA, were correctly predicted with respect to what the original sequence would be. But we also find, for example, in this case, what would be the residues M, A, L, A, since they were wrongly predicted because what would be the original sequence is really the residues B, I, P, A. And what would be the correction of these errors is that the signal is backpropagated throughout the entire architecture of the transformer to be able to precisely mask the sequences again, and now detect which residues would again be the most probable. And simply with this task, the BERT-type transformer really learns representations rich in information throughout the multiple layers that can constitute our transformer, in such a way that it is usually a practice quite adopted by many in the community. Taking what would be the embedding of the last layer, where we usually find more information related to the structure of the proteins. And then we are talking about having embeddings that, in real terms, what they do is represent a receipt of our protein. But in order to represent the entirety of our protein, it is necessary to make a kind of average, using a technique known as mempooling, in such a way that we average all the values of each of the dimensions of our embedding. To have precisely this representation now at the protein level, where in general terms at the level of what information we can find in each of these channels, we really do not know because perhaps some statistical patterns that the model learned could be related to what would be the secondary structure others. We simply do not have the remotest idea that they could correspond others perhaps would correspond to what would be an idea of folding. Or it is usually the case that these types of algorithms actually learn concepts that would be overlapping, so they would not really be easily dissociated as a single dimension. So this information is actually contained at the vector level, and then it can be easily used by other algorithms such as random forest to make a prediction or regression theory through what is known as transfer learning. And as you remember, what language models really do is learn a probability distribution based on the tokens with which the model has been trained. So that, for example, in the case that a language model of the GPT type receives as a conditional text what would be the phi child, through this probability distribution, we find that the tokens that direct us to the words game or school, they would correspond to the tokens with the highest probability under what was the training regime of this type of models trained with text. So, how would this type of behavior be reflected by language models when we train them no longer with text, but now with proteins? And there are quite interesting behaviors that we see represented by this structure of the protein, where we find residues that are on the surface marked in red, and others that are buried in the core of the protein in blue. And then, when we look at the probability or likelihood assigned to each of the residues found on the surface, we find that these types of protein language models assign a greater likelihood to residues whose physical-chemical behavior is more hydrophilic in nature, which coincides with the type of residues that are precisely distributed on the surface of the proteins. And otherwise, it happens when we inspect what type of residues receive a higher probability when we turn to see what the core of the protein is, in such a way that it is the residues with a hydrophobic behavior that receive a higher probability, which would reflect that some statistical patterns learned by this type of protein language models coincide with these physical-chemical behaviors of the residues. Something that we can see well generalized or summarized in what would be this distribution graph with boxes and whiskers, where it is quite clear that what are the residues assigned to the core regions of the proteins really receive a greater probability when they are found with a more hydrophobic behavior, and vice versa, when they are really found in what would be on the surface, which is relatively consistent with what we know about proteins. And that curiously, they learned these language models by masking the tokens, among other training regimes. Also, as you may remember, we actually already reviewed what would be some of the main protein language models most widely adopted by the research community, such as the models developed by this research group in Meta, which in this case present two large families that are known as the ESM families. In this case, we have different variants for each of these families. In this first project, the ESM1 family is presented, where the ESM1B model is the most widely used, and subsequently all aspects of this type of models applied to proteins were further developed, which would be the ESM2 family of models, which is in fact one of the most widely used by the community. Similarly, we also found this other project, which is a collaboration between the NVIDIA laboratories, Google, and the Booker Trust Laboratory, who also trained several variants of what the transformer architecture is to identify which would be the most suitable to model protein residues. And so we find different architectures such as ProtElectra, ProtXLNet, ProtBird, ProtAlbert. And where really through the comparison in different benchmarks they found that the most successful variant among all those they trained would be the one related to what the PROTT5 architecture is with what would be the XL variant with the data from the UNIREF sequence database that, in this case, you could adopt it to really be able to carry out the analyses based on the computational resources that you have available. In that sense, one of the most interesting characteristics that these types of protein language models trained only with sequences give us is that they learn to predict statistical patterns that could be correlated with the concept of the functionality of said proteins, or what we also know as fitness. Something that we see represented by this graph, where on the x-axis we find a great diversity of datasets ranging from antibiotic-resistant proteins, fluorescent proteins, proteins that practically perform a great diversity of functions, and on the y-axis we find the one that also correlates these fitness. Measurements carried out experimentally with what would be the probabilistic assignments made by different models. In this case, in blue, we find precisely the ESM1B protein language model. In orange, we find another language model trained with an architecture that is no longer transformer, but is an LSTM-type architecture. In red, we find another language model that in this case is a specific architecture for each family, and that is trained with an autoencoder-type architecture. And finally, we found a supervised model that is really trained with machine learning algorithms, and not with architectures related to language models. And what we found in general terms is that the performance of the ESM1B protein language model, represented by the blue dots, seems to outperform all the other models in this fitness prediction task under a zero-shots regime. That is, without having to do training with a supervised learning scheme to be able to predict which variants would be most successful. So then, we could use it as a kind of oracle to identify which variants would have more functionality compared to others. This has been developed in a little more detail, so that a benchmark was even built, which is known as Protein G, and which constitutes approximately 250 datasets that have been standardized and come from deep mutational scanning type experiments. So, for example, if you are looking to find what type of language model could best serve your protein family, you could consult the results present in that type of benchmark. Or similarly, if you train a model that aims to predict fitness in a generalized way, you should also evaluate your models using that type of benchmark that is widely adopted by the community. And this has also led to extensions, for example, in projects developed by Google DeepMind, which were also interested in being able to predict the fitness of proteins, or in this case, the pathology of the mutations that can be found in each of the residues, through a model that they called alphamescence, which, in short, helps us identify which residues have a greater or lesser probability of being pathological for the protein. That is, they can be quite harmful at a structural or function level. And so, for example, identify that residues that are found relatively in turn regions or very much on the surface, as they can support a greater number of mutations, and that is why they are colored blue compared to those that are found in the core of the protein or in the main body, which are colored red, indicating that it may be more dangerous to make mutations in this type of residue. Another extension that these protein language models have had through this concept of the probability that they assign to each of the residues and that can be added and represented to each of the proteins is the idea of being able to trace some evolutionary trajectories based on said learned probabilities, something that is explored precisely in this project, where, as you will remember, any type of BERT-type language model obviously has to mask the sequence. And then, through a series of attention blocks, it learns precisely to identify which residues should satisfy that context and thus predict precisely the most probable set of residues. And during this training, BERT-type language models learn representations rich in information that we know as embedding, but they also learn to precisely assign a verisimilitude, a probability to each of these sequences, as well as to each of the residues that compose them. So that thanks to this concept of this probability, we can generate a kind of fitness landscape, not now directed with what is experiments, but directed under what is the probability that this type of protein language models learned, in such a way that we generate precisely this landscape, where some variants will have greater fitness, represented by increasingly yellowish colors, compared to other variants that will not have as much fitness, or that is, this probability learned by the model, which are presented in more black or purple colors in this case. Once we have assigned the probabilities to each of our proteins for a specific protein family, we can generate a certain directionality based precisely on these probability scores. And once it has been drawn, this directionality between mutations, between proteins, from least probable to most probable, we can generate a graph that allows us to connect the entire family of proteins that we are modeling. Once this graph has been made, we can infer an idea of what the putative ancestors or roots of said graph would be, so that we then have a structure where it is precisely part of proteins with low probability learned by the language model, and that would extend to what would be those with the highest probability, which in turn allows us to draw a kind of gradient or temporality, or specifically, pseudotemporality used within that project, in such a way that when we turn to look precisely at this pseudotemporality, it allows us to identify which sets of mutations would have caused the probability to increase along that evolutionary trajectory. And this is precisely what they were in charge of investigating in this project. So now it remains to assess how it is really reflected in evolutionary terms. And that is why they explored what this phylogeny is for the nucleoprotein of the influenza virus, among other examples. And it's quite interesting that for this specific protein, we know that we are modeling an entire family here. However, there are some particularly specific variants, which are known as H5 and H7, which turns out that their phenotype is really presented in this way by an invention of convergent evolution, and that they are not really derived from what would practically be a phenotype from H1N1, which is precisely what this type of phylogenies is presenting to us. That due to its dichotomous behavior, which necessarily has to split the data in two each time a branch is extended, an event of evolutionary divergence is recapitulated in this case, something that is not really compatible with what we know regarding the evolution of this type of variants within this influenza, a nucleoprotein family. And then when we look at the representations created through this probabilistic approach, we find quite interesting things. What we see here is a two-dimensional map, where each of these points actually represents one of our proteins. Only in this case, obviously we have to take what are the embeddings that represent each of our proteins, and other embeddings are usually of high dimensionality. In this case for SM1B, we are talking about a vector of approximately 1280 dimensions. It is clear that we as humans cannot see that dimensionality, so we have to project it to two dimensions that allow us to generate this type of maps. So what is done here is to implement a dimensionality reduction algorithm known as UMAP, which allows us to take this dimensionality of 1280 dimensions to only two representative dimensions that would recapitulate the greatest amount of information possible from that high dimensionality. And then we observed, well, let's say a good context recapitulated based on the fact that the variants that are assigned to what is the H1N1 group seem to be relatively well organized, as well as other variants such as what would be H3N2, among others. And it turns out that when we zoom in to see what the dynamics of the H5 and H7 variants are like, it turns out that they seem to have an independent origin that converges to what the H1N1 genotype is, which really coincides with what we know about the convergent evolution event that led these variants to present a phenotype quite similar to the H1N1 variant. So what this project really represents is a new perspective of being able to explore different evolutionary trajectories, and thus generate hypotheses about how evolution would have occurred for a specific family of proteins, which at least, in my opinion, is quite interesting, as well as other behaviors that are evidently explored in this type of projects, where it turns out that when we turn to see which residue is paid more attention to during training, It is quite interesting that these tension patterns actually recapitulate some structural properties of the proteins quite well, in such a way that when we map precisely these tension patterns, represented by orange lines in this case, it turns out that residues that, although they are distal in the sequence, have a greater degree of attention to each other because they happen to be in contact in the three-dimensional structure, which suggests that although this type of transformers was only trained using a masking regime, which is really only seeing sequences and no three-dimensional information, they learned to distill information that seems to be related to the contact patterns in the structures of the proteins. And we even see other cases where many residues seem to be paying attention to only one in particular. And it turns out that when you actually look at why this would be happening, it turns out that this residue is quite important for the function of this specific protein, which is a protease. So then there would also be some cases that would be detecting statistical patterns that would correlate with precisely residues that have some importance at a functional level. Some patterns can even be identified, as in this case, that can be completely assigned to a folding in such a way that, for example, what would be a folding pattern similar to an alpha helix, a beta folded, or even to what would be complete proteins such as timbals, beta-lactamases, among other structural properties, is identified, which was actually quite interesting. And really throughout the architecture of the transformer, there are also different capacities to be able to identify this type of patterns of secondary structures, something that we explore precisely with what is the ESM1B protein language model, where what we observe on the y-axis is the capacity to be able to use the information derived from this model to be able to train a logistic regression, and thus see that these patterns of secondary structures are also precisely identified regarding the depth of what its layers would be. And we observed that precisely during the first layers, there seems to be not enough information to be able to identify regions of secondary structure. Subsequently, this identification capacity improves in what are the intermediate layers, and finally it improves even more to be able to reach what would be the last layers of our transformer, where there is greater performance in terms of identifying regions of secondary structure, which would be reflecting to us that it is in these last layers where perhaps there is greater information at a structural level that is encoded, and from which we could take advantage to be able to make predictions that have a nature related to the structure of the proteins. This type of behavior obviously caught our attention, and we then took on the task of investigating whether we could extend the predictive capabilities at the level of protein structures using only language models. And this is precisely what was explored in this project, where what the authors found is that we can precisely use attention maps to predict contacts. What you observe again is the architecture of a BERT-type transformer, where we start from a sequence, we mask it, and we build what are the arrays of queries, keys, and values. Subsequently, the tension is calculated, and we proceed to the entire concatenation part that ends in what is the multi-layer perceiver. And this is repeated for several layers. So what you do is take these stress maps and make them symmetrical. Well, as you remember, the tension is really asymmetrical, and so we need, let's say, to normalize them in a way. And once we have symmetrized them, we simply train a logistic regression, concatenating all this vector information to be able to predict the contacts, using as a reference precisely contacts derived from three-dimensional structures, which precisely allows us to even predict the structure of proteins using only the information that was learned during training from these attention maps. And this is something that is explored in more detail in this type of graphs where we identify, which is also being able to detect this type of contacts at a local scale of between 3 to 6 Armstrongs distance, a smaller scale of between 6 to 12, a medium scale of between 12 to 24, and a very long distance scale of 24 to more distances. In general, and it is quite curious that in general in almost all the patterns we find along the layers, as well as for each of the heads of the transformer. It is precisely in the intermediate layers of the model, where there seems to be more structural information that allows these types of protein language models, trained only on consequences, to predict properties such as contacts. That is, it would be reflecting structural patterns, even though only consequences were trained. And this is also recapitulated when we eventually predict contacts that are already medium to large scale, where it is quite interesting how even these, which are even more complicated to predict because they are completely distal at the sequence level, can be recapitulated in order to recover this type of contacts. So it is then suggested that this type of language models could be learning statistical properties correlated with the structure of proteins. And this was evidently something that caught attention and even contrasted with this idea of what escalation laws are. Could we then improve the capabilities of protein language models to predict structures by making them larger and larger? As you will remember, these types of scaling laws, which are not really laws but are empirical observations, really what they represent is that as we increase the size of our model, and consequently the data with which it is trained, the performance of certain tasks will improve. At the same time, even new tasks appear that perhaps we had not previously considered. And we see this represented with this diagram of a small tree, which really represents a language model trained with text, which has approximately 62 billion parameters, where we find some skills such as understanding the language, translating between languages, or even making text summaries. And it turns out that when we increase the scale of our model to approximately 540 billion parameters, we find that new capabilities appear such as the ability to identify proverbs, an ability of logical inference, even increasingly complex pattern recognition. So it made quite a lot of sense to think, can we now extend these scaling capabilities to protein language models? And it turns out that yes, in fact these types of scaling laws would really serve both GPT-like architectures, that is, autoregressive models or more causal modeling, as well as BERT-type architectures, which follow a masking training regime, where what we observe is again on the yes-axis, how the loss is reduced depending on how much computing we invest, as well as different varieties of model parameters. Where what we observe, as you already know how these types of scaling laws work, is that we train several variants of models based on the number of parameters, and by adjusting these types of lines, it allows us to know or have certain predictive abilities of what computing capacity we need, as well as what parameter capacity we need to implement in our models to reach a certain loss. And this is quite interesting that it also happened with the protein sequences, and not only at the text level, which is what transformers were really designed with. So we would be thinking or suggesting that it is really a behavior more assigned to what the transformer architecture is, and not to the data that we are trying to model. So we could really scale many properties depending on the data that we are trying to model. And this is precisely what this group of researchers who developed the protein language models of the ESM families were in charge of investigating, where they were one of the first groups to scale protein language models. And this is precisely what we observed when they launched the ESM2 family, where we find models ranging from 8 million to 15 billion parameters. On the y-axis, what we observe is the ability of these models to predict protein contacts. And on the x-axis is how many homologs are found for these contacts in what were practically the databases with which these types of models were trained. And it is quite interesting that, for example, the more homology information that is found in the databases and therefore has been used during training, the predictive capabilities at the contact level are improved. But these predictive capabilities also improve as we precisely scale the number of parameters of our protein language models. So it is suggested that they really do reflect this scaling capacity at the contact level. And it is quite interesting that what is the comparison of the ESM1B family model with respect to the ESM2 variant of just 150 million parameters, they result in behavior that is quite similar to each other. So what is suggested here is that the variants of the SM2 family will be able to recapitulate more information, because even the variant with 650 million parameters has better predictive capabilities, depending on what the contacts that are being modeled would be. We could then adopt some of these protein language models and extend their predictive capabilities now by coupling them to increasingly sophisticated architectures. And this is precisely what they did. What was really done was adopting the Evolutionary architecture along with the AlphaFold structural module, but taking advantage of what would be the embeddings learned by the ESM2 family. In particular, what they specifically did is take what is the ESM2 model in its variant of 3 billion parameters and precisely couple these embeddings to what would be the structural module together with the Evolutionary to formally predict what the three-dimensional structure of proteins is along with the idea of recycles. And specifically, what they did is train a model which they called ESMFold, where what is actually done is not to use the MSA representation that AlphaFold has to learn through Evolutionary, but rather this component is replaced by what would be the representations at the level of the protein embeddings. And what would be the pairwise representation is not really part of what the structural templates are, but is simply initialized from what would be the relative positionings of the sequence. That is, we do not really need to build a representation at the MSA level, as the AlphaFold 2 algorithm does, but we can simply use the information from the embeddings of these language models to predict the structure of the proteins. And later, there are mechanisms very similar to the architecture of AlphaFold 3. Such as they could be precisely the mechanisms of triangular tension or tension at the row level, as it could be in this case of self-tension, which would really be providing tension at the level of the embedding. And thanks to this mechanism, coupled to the ESM2 family, quite interesting predictions were actually achieved. Here what we find is a comparison of PLDDT trust metrics of the predictions made with AlphaFold compared to the predictions made with ESMFold for approximately 4,000 proteins, where we found a Pearson correlation of approximately 0.8, which suggests that they are quite consistent with each other. And when we look back at what the folds are also being modeled using TM score as a reference, we find that AlphaFold 2 clearly has lower performance compared to AlphaFold 2 or RosettaFold. But when we decouple the information provided by the multiple sequence alignments to these two algorithms, it turns out that, in this case, ESDNFold surpasses it considerably, because they were actually trained not to consider this type of information, and since they look quite deteriorated when we remove said alignments. So we're talking about now having a protein structure predictor that works much faster because it's not having to rebuild. This type of representations derived from alignments, but simply part of what was already the training of protein language models, and we take advantage of said information contained in the embeddings. In fact, when we look at how fast ESG-unfold actually is compared to AlphaFold, it turns out that it is up to 60 times faster. And even though it's too fast, you also have to take certain precautions, because it is not as good as AlphaFold 2 would be to explore in more detail. In this graph, what we have on the y-axis is precisely how long it takes to predict a structure in terms of seconds. And on the x-axis, what we have is how many residues we're trying to model for said protein. And in general, what we observe is a behavior that is really what the use of yes and fold is for. Some proteins that are relatively short, such as 100 or 200 residues, will be predicted in just a few seconds. For proteins between approximately 600 residues, we will need approximately 1 minute. And for fairly large proteins like 1500 to 1800 residues, it can take up to approximately 800 seconds. But when we see this blue line, which is precisely the one that represents ESDMFold compared to the red line that would represent AlphaFold, we really see that SIM has an advantage in terms of the amount of residuals that we can model as a function of time. So if you have a protein that is quite well represented in the databases, it could really be more interesting to use ESMFold to predict these structures, since it runs much faster, and they would spend less computation to actually be able to predict a dataset of approximately hundreds of structures, or even massive datasets of 10,000 or 20,000 protein sequences. Here we also have another comparison between what the TM scores would be for a total of approximately 2,400 protein structures, where although they do not offer us a correlation metric between what the predictions of AlphaFold 2 would be with respect to ESMFold, we find that under these lines that represent a margin, 81% of these proteins are found. And when we look at the difference in terms of TM score, it turns out that only values less than 0.1 are presented. So we are talking about fairly consistent structural predictions that would reflect a fairly good capacity of this type of algorithms such as YES and INFO, which were trained only with information from protein language models and not with structures. Well, yes, necessarily with structures, but taking advantage of what embeddings are to be able to do this task of structural prediction faster. And it is thanks to this ability to accelerate structural prediction that the Meta team really extended these capabilities to be able to predict the structures of a total of 772 million structures, which is represented in this database called ESG Metagenomic Atlas that you can explore in a fairly dynamic way, and that you can even use your web server to be able to predict protein structures in a matter of a few seconds. However, despite the fact that there are 772 million structures, it turns out that only a subset of about 225 million would be the ones that have really good predictions in terms of PLDT. So we're talking about even almost a little less than a third that would represent good predictions. So really perhaps not all the structural information in this database would be useful, but precisely some of it would be quite poor quality in this sense. But obviously, it is interesting how even with this dataset of 225 million structures, it has a scale quite similar to the scale provided by the AlphaFold 2 algorithm that also works on the scale of approximately 200 million structural predictions. Also, ESMFold as well as AlphaFold 2 can be used to characterize which proteins would be most likely to be successful in experimental campaigns. However, we have to be quite careful when using ESGNFOLD as a kind of oracle for design or screening purposes in our experiments. And this is something that is explored in this specific research, where what is done is to evaluate two types of proteins in general terms, those that are soluble in water, or others that are much more difficult to model, which would be transmembrane proteins, because this type of proteins do not find many genetic representatives deposited in the protein data bank databases. So it is more sensible to wait for these types of proteins, because you cannot learn as many statistical patterns derived from them, as they become more difficult to model. And what is done here is to start precisely from a reference structure and generate several putative sequences for said structure, and see which of them are successful at an experimental level. So what we do is measure two metrics of how well our variants would be modeling for a structure. On the y-axis, what we have is the structural similarity using the RMCD metric, and on the x-axis, what we have is the PLDDT value assigned by AlphaFold. And what we found is that, in the case of proteins that are soluble in water, which would be the variants that were experimentally successful because they folded correctly, well, they are relatively well located in what would be the regions with the highest PLDD and lowest RMSD. And the variants that did not have this experimental success, that did not manage to express themselves, are distributed throughout this other large margin of consideration between both metrics. Something that, when we see it at the level of their distributions represented by these boxes and whiskers, we find that there really are significant statistical differences between their distributions. And the same thing happens at the PLDDT level, as well as at the RMSD level. When we turn to see what the behavior of transmembrane proteins is like, we really have something much more dispersed, where there are actually some variants that did manage to be expressed in the laboratory, but in this case, they are more dispersed in what would be this entire region. With respect to this region, that is the one with the highest PLDDT and lowest RMSD. So it is then suggested that they are more difficult to model. Despite this, if there is a certain difference in the distributions, both at the PLDDT level and at the RMCD level, so in general terms, we could think that AlphaFold 2 would allow us to identify what type of proteins can be more successful at an experimental level. And obviously so, well, let's say it. Better direct our economic resources and the time that we are going to invest in experiments to be able to characterize variants that we only know through computers. And now let's see how the ESGFold algorithm behaves. And it is quite interesting that when we see both the proteins that are soluble in water as well as the transmembrane ones, ESGFold really does not have this capacity to be able to discriminate which ones were experimentally successful and which ones were not, as you can see. They are therefore relatively compacted, grouped in regions that do not allow us to distinguish between these two groups. And when we look at their distributions in terms of PLDDT as well as RMSD, there seems to be no significant statistical differences between both groups. So it really seems that ESMFold could not be so useful to be able to identify which protein variants would have greater success in what would be experimental campaigns. However, the authors do not really suggest that ESGFold should be completely discarded, but rather that due to its great speed, and when other types of analyses are carried out that they present in this work, they found that ESGFold could actually serve as a great first filter that allows us to evaluate the designability of the backbone. That is, if we have too much garbage at the level of protein sequences and structures, then we can precisely predict their structures and discard those with the worst EP and LDT ratings. And this first filter, we would later have to go to a greater extension, using AlphaFold in this case, to now be able to identify which proteins could really have this ability to fold correctly and thus have experimental success. So, depending on the type of experiments, as well as computational and temporal resources that you have, they can then use these two types of oracles to increase the chances that their computer designs will actually be experimentally successful. Similar behaviors have also been observed in other works, as is the case here. As you may remember, in this domain encyclopedia project, the 200 million structures predicted by AlphaFold are adopted, and what type of folding is found among these structures is investigated using the CAD reference scheme. And so, they found a total of about 371 million domains in all of these proteins. And within these 370 million domains, it turns out that about 7,500 perhaps could be new folds that appear to have no structural homologs in the databases. So now the idea is to investigate whether AlphaFold predicted these new foldings. How would ESGFold be predicting them? Which is precisely what we see on the... And what we observe in a quite interesting way is that when we see these density maps where darker regions represent a smaller number of proteins and brighter regions represent a greater density, and therefore a greater number of proteins, of these 7,000 in total, it turns out that when the PLDDT predictions for a protein from ISDINFold take values of approximately 30 to 40 of PLDDT, it turns out that the predictions made with ALPHAFold2 have PLDDT predictions greater than 80, as you can see. And this results in a fairly consistent way with practically all this margin, where ISDINFold consistently assigns low PLDDT. Based on the predictions of AlphaFold 2, where good PLDT values do occur. So in general, what is suggested in this research is that ESMFold can be a good choice when trying to explore foldings that we already know previously. That is, that they are fairly well represented in the databases. That is, also that there are quite a few homologs, any of those three perspectives that you want to understand. And really, when we want to explore new sequence spaces, which perhaps are not so well represented by many homologs, then ESMFold is no longer as good at characterizing these proteins. Because then it would seem that it is evidently going to be reflecting the biases of the data with which it was trained, and that perhaps it would not be as capable of exploring these new sequence spaces in terms of folds. And just as a little gossip, really what has happened with the ESM family of protein language models is quite curious. This type of models has had an enormous impact at the level of the protein science and artificial intelligence community, such that when we see the number of citations of the articles, we find that for the SM1, SM2, and SM3 family, we're talking about approximately 3,300 appointments, 5,000 appointments up to 800 appointments respectively. So this really reflects a great impact, and when you look at the type of work, what these families of models would be has really been widely adopted. And despite the success of this team, known as the ESM team, it turns out that Meta at that time had other interests. In particular, Zuckerberg had a greater interest in the development of the metaverse, as well as in his language models of the Llama family, and so this biological research team was completely dismantled. And then this research team, represented in this case by Alexander Rives, was really motivated to found his own startup, which was called Evolutionary Scale, and thanks precisely to, in addition to their success at the bioinformatics level, they managed to raise an investment round of approximately $142 million to be able to start their research projects. And despite this, we also found some announcements, such as this one from John LeCun, who was then the main artificial intelligence researcher at Meta, who announced that Rob Fergus was hired precisely as one of the main leaders in what would be Meta's machine learning sector. And even more curiously, in more recent announcements, in about 2025, Alexander Rives has now announced that he is actually joining what is the Chang-Zuckerberg Initiative, a biological research initiative that Zuckerberg has together with his wife, who is called Chang, where they actually do research on biology with various centers, such as those that are located in Chicago, New York, San Francisco, or even institutes dedicated to microscopy, where the aim is to build a computing supercluster with approximately 10,000 GPUs to be able to train protein language models for the next generations of SM. For example, SM4 or SM5, trying to take advantage precisely of the new databases, as well as the talent on the part of this team. So it is quite curious how this technology, which was only developed in approximately 2020-2021, well, she was completely fired and rehired again in 2025. Well, now we will explore precisely how these language models that have been trained only with protein sequences are able to predict protein structures. And for this precisely, we have several hypotheses that are raised in this work that I strongly recommend that you review because it contributes a lot to the interpretability of this type of models. We have three hypotheses. One would be that precisely these models have learned folding patterns that could reflect the biophysics of folding, that they would understand these concepts of energy and conformations and identify those most likely configurations that reflect a native structure. A second hypothesis is that they would have the ability to represent foldings, but they evidently represent it in the form of co-evolutionary statistical patterns of protein families. And another third hypothesis is that this type of protein language models may not be representing statistical patterns that correspond to complete faults, but rather they would be corresponding to small structural motifs that are reflected as small segments represented by these little arrows that would be recapitulated through contacts. So this is precisely what they took on the task of investigating which of these hypotheses is the one that could give us a greater understanding of how this type of protein language models trained only with sequences operate to predict structures. It's pretty clear here that protein language models trained with sequences don't really learn explicit biophysics. Because of this type of behavior, as we reviewed previously, ESM2 really depends too much on the number of counterparts it has been trained with. As we saw, those proteins that didn't have that many homologs really couldn't also be lost in terms of their contacts. And so, if you learn biophysics, regardless of the sequences you were trained with, you should generalize this ability to identify which protein is most likely to fold. However, this is what does not happen. And we really see a great correlation between the number of homologs, as well as with the performance of their predictive capabilities at a structure level. So we still have to explore what is happening in these two cases. Would I be learning statistical patterns associated with complete foldings or patterns associated with small motifs at the sequence level? Well, in order to illustrate precisely that biophysics is not really being learned, is that we turn to look at some quite particular proteins, which are the isoforms. In this case, we see a myoglobin, which is represented here in the structure with the alpha helices and, oh no, they are only alpha helices, wow. Well, at the level of what would be the cartoon structure, as well as at the level of the surface of the protein. And then we see that the charges are also distributed using a metric known as SAP, which really gives us an approximation of the degree of hydrophobicity based on the color, while more red would mean that we are talking about residues that are more hydrophobic in behavior, and in white residues that are more hydrophilic. And then when we take what would be the isoform of our protein, that would correspond only to these regions that are colored in different colors, and that it is not including what these alpha helices indicated with the letter A and the letter B would be, we find how these isoforms are being predicted with what are the AlphaFold 2, OmegaFold, and ESMFold algorithms. And it's quite interesting that in all three cases, we found that the predicted structure contains these patches of residues that are hydrophobic and that are exposed. And as you already know, this type of behavior should not really occur. It is unlikely because this does not really distribute our loads homogeneously, and because a folding in this sense is not really being predicted because it's native. Structure would not be adopted because these residues must be buried in the structure and not exposed on what would be the surface, and despite that, what these three structure prediction algorithms are, they are predicting it as if it were something real, even though it does not really happen. And in fact, this not only occurs at the level of myoglobin, but a total of approximately 18 isoforms were evaluated, where we found that consistently throughout these three algorithms and the 18 isoforms, they always tend to be predicted, with a large number of hydrophobic patches, such that then they would also be predicted incorrectly. Although PLDT values are found, for example in the case of AlphaFold, between approximately 50 to 90%, and in the case of 10-10-fold, between perhaps approximately 55 to 80%. And the same in terms of RMCD, which would be the reference, except for these regions that are not part of the isoform. They are also found to have quite low RMSD values of approximately 3 to 0 Armstrong's resolution. So there is really an error here on the part of these types of structure prediction models, which reflects the fact that they are not learning biophysics, because if they were learning biophysics, these hydrophobicity patches should not be represented this way. So once we have ruled out what this hypothesis is regarding biophysical knowledge on the part of protein language models, we have to see what happens with the other two hypotheses of understanding at the folding level or at the motif level. And for this, the authors developed an unsupervised approach to be able to derive what the protein contacts are only from the embeddings of the protein language model, which they called categorical Jacobian. And here we have precisely a comparison with two other methods that are based on what supervised learning is. In this case, we can know the structure of the proteins through the approach offered by ESMFold, which takes the embeddings of the ESM language model and couples it to the AlphaFold 2 architecture. We also have another supervised method, where it is less powerful than coupling the AlphaFold 2 architecture. In this case, you simply train head-to-head. able to predict the contacts that can be something more subtle like a logistical review, and we can precisely obtain these contacts. But thanks to this approach, which is completely unsupervised, it allows us to learn more about what the protein language model learned during its training. And so now what we have to do is that the information of the three-dimensional contacts is also being recovered as we mask what the embedding information is, since this is really the task with which this type of protein language models are trained. And to do this, the authors developed this quite creative experiment, where what they do is, we start from what is an observation of some structures that we previously know establish contact with each other, and that they can be separated by approximately 15 amino acids, between 15 to 100 amino acids, or up to more than 100 amino acids, as we could perhaps see in orange in this particular figure. So, if we know precisely that there is contact between these regions, even though they are separated in the sequence, what we now proceed to do is proceed to unmask regions adjacent to said contact regions. So that, for example, here we are unmasking regions adjacent to these contact regions that are represented by approximately 15 residues in total. By being able to unmask this type of regions adjacent to the contacts, in principle what the model would be learning is an idea of structural reasons, due to the contacts being recovered gradually in relation precisely to the information adjacent to the contacts. To corroborate, for example, if something similar to a global projection of protein folding is being learned, then instead of recovering information adjacent to what the contacts are, we could unmask regions that are completely random from what the rest of the sequence is, which in this case, as you can see, some completely random residues are unmasked here in blue, or residues that arbitrarily, a margin of 30 residues of distance, is left with respect to the contacts that we previously know. And so, if the protein language model learned a global concept of what foldings are, this simple unmasking in a randomized manner should be able to recapitulate the contacts in a better way compared to what would be the contiguous unmasking of the contacts. And here we precisely have an example of what this type of protein looks like. We have precisely this example where a protein is in blue, and these two alpha helical regions are in contact in the three-dimensional structure, even though they are separated by a margin in the sequence. And when we look at what a particular contact map is, we observe precisely this type of patterns, which would be equivalent on this other axis or triangle of what this graph would be. So if we observe that, for example, what is residue approximately 150 in relation to approximately 430, it would be establishing several interactions that are represented in what would be the three-dimensional structure of the protein. And here's something interesting that they found is this behavior, where it turns out that, in some cases, when the neighborhoods close to the contact regions are unmasked, contact information suddenly appears. So for example, we have here some contact regions in orange, and only 13 residues neighboring these regions are unmasked. And in this particular case, no information really appears regarding the contacts of said protein. But when we unmask a total of 14 residues, information about this type of contact suddenly appears, and not only in this region, but also even in a more established way along the diagonal of the image. So it is quite interesting then how suddenly, by unmasking just one amino acid, even information of this type is represented. And this is a behavior that was not only observed in this protein called SUSD, but for the total number of proteins that they studied in this case, which are approximately 1,400, this behavior was observed 65% of the time. So when we evaluate this in general terms, well, how well contact information is being unmasked and recovered. And in general terms, what we observe is that unmasking the regions flanking the contacts can be up to 3 times more effective in recovering said information compared to other unmasking strategies. Here again, we observe on the y-axis what is how well the information is being recovered, compared to how much of the sequence is being unmasked. And we have it both for proteins that are separated by a margin of 15 amino acids between their contacts, 50 to 100 amino acids between their contacts, and more than 100 amino acids, so they would be quite distal contacts in the sequence. And what is observed is that this strategy of unmasking the regions that are neighboring the contacts recovers in an easier way with even less context, what is the information of the contacts with respect to these other two completely random or random unmasking strategies, but considering a margin with respect to the contact regions, and this extends both for proteins that are separated by 15 residues. Well, better said the contacts that are separated by 15 residues by 50 to 100 residues, or even more than 100 residues, so that in general what is suggested is that really the protein language models trained only with sequences would be learning statistics related to the motifs, and that this is really being done for all the protein families with which said models were trained. And here in this project, they also make a quite interesting observation where they tell us that really these coevolutionary statistics that are being learned for all the protein families present in the uniprot would need a total of approximately protein language models with 261 billion parameters, which is really outside of what we can currently understand. Because, for example, the family of ESM2 language models barely reached 15 billion parameters, so it would really require too much computing to be able to train protein language models of this level. Well, we have to make certain assumptions that are relatively sensible to see if we can reduce this number of parameters. So here we assume the fact that most proteins establish up to 4 contacts at their locality. And through this premise, we arrive at a combinatorics where, when we already solve what the mathematics related to said combinatorics is, we find that protein language models with approximately 4 billion parameters would be needed to be able to learn the convolutional statistics of all families deposited in the Juniprot database, which is approximately 20,000 protein families. And this is quite inconsistent with precisely the orders of magnitude of the ESM2 protein language models, which they manage to outperform in terms of their performance, which, as you remember, there is a model that, as we saw, which is the 3 billion parameter model, when it extends even up to 15 billion parameters. It seems that there is no longer a considerable increase in terms of its ability to predict protein contacts, regardless of the number of homologs with which it has been trained. And this premise of 4 contacts in general really makes quite sense, because when we look at how many contacts some residues can establish, such as leucine, valine, isoleucine, methionine, approximately 5 contacts are established by this type of amino acids. Here in green we have other types of amino acids, such as aspartic acid, lysine, serine, which would establish a smaller number of contacts, but in general terms, for contacts that are short-chain, that is, locally they would be establishing about 3.6 to 3.7 contacts in general for each of these residues. Therefore, this premise of 4 contacts in general also seems quite accurate on the part of the authors to be able to approximate what would be an optimal training scale, to be able to adjust the coevolutionary statistics for 20,000 families. So when we look back at what this project is, we also don't. They completely dismiss this hypothesis that they are also learning a folding idea altogether. But at least the data suggests that they are learning coevolutionary statistics at the level of sequence motifs. So in general, the summary that we can say from these observations made from said project is that protein language models trained only with sequences will really be only as good as the number of homologs that have been used in training. Since there's a level of parameters, as you can see in this case, that would be the versions from 15 billion to 3 billion, where there really seems to be no considerable improvement. Even when we take the model that is 650 million from the SM2 family, which is represented by this blue line, it seems quite redundant when we see the performance compared to its other variants that have a considerable number of parameters greater than that. Other work regarding the interpretability of this type of models can be carried out based on the idea of the autoencoders that we previously explored, which, as you will remember, actually, the concepts learned in what the neurons of these deep learning models are really have a property called polysemanticity, where multiple concepts would actually be mapped to a single neuron in such a way that it is not clearly distinguishable what ideas each of them would be learning, because they are precisely overlapping in this sense. This is precisely the architecture that protein language models practically use, and so an adaptation of the architecture of the autoencoders has been developed, which are these sparse autoencoders that instead of making this bottleneck so that they learn representations rich in information, we start from the embeddings of some language model and expand its dimensionality to see if this polysemanticity can be separated from each of the neurons, and thus discover if we can associate a specific concept to each of the neurons. And thanks to this type of sparse autoencoder mechanisms, several investigations have emerged. As you can see here are 4 publications in this sense, which investigate what aspects protein language models are learning, in such a way that some characteristics have been found that are general to all proteins, as well as characteristics that seem to be specific to each of the protein families. In this case, we see some characteristics that may be relatively general, such as the detection, in this case by red regions by these protein language models, of regions that are disordered, or even more systematic patterns, such as a separation pattern of 7 residues only in alpha helices. We also have others that are related to what are conserved motifs or domains, such as a helicase or signal peptide domain, and other characteristics that perhaps are more closely related to the biochemical context of the proteins, such as, for example, the disulfide bonds belonging to what is the interaction between cysteines. And something interesting here is precisely the ability of these protein language models to be able to identify some characteristics that seem to be specific to protein families. In this case, we're looking at precisely what features the ESM2 protein language model learned in its 650 million parameter variant. And what we observe is that of the total of what these concepts would be learned by the neurons, by the sparse autoencoder. There are some that seem to be very general in this sense, analyzing what the class A beta-lactamases family is, which really do not seem to be so activated when we analyze this protein family in general. But there are a few activations within these characteristics that seem to be quite specific precisely for class A beta-lactamases, such as the ability to precisely distinguish the presence of a central alpha helix in what would be this type of beta-lactamase structures. And even if we only use this set of characteristics that are strongly activated by what would be the pregament of a beta-lactamase, we can train classifiers that are quite good using only said information from those neurons. So let's say that they would be learning specific aspects of these families that precisely help to distinguish them from the rest of the families with which the model was completed. Something more curious is when we see the type of distribution of these characteristics that are family-specific throughout the layers of what is the protein language model. Here on the x-axis, we observe precisely what the layers of the SM2 language model would be, 650 million parameters, which has up to 32 layers in general, regarding how many characteristics specific to protein families we find throughout each of these layers. And what we observed is that in the first layers there really are not so many family characteristics of the proteins, but in what would be the intermediate layers of the protein language model is where there would be a greater number of these types of family-specific characteristics. And finally, in the last layers, the information associated with understanding protein families seems to considerably decrease. This is a quite interesting behavior that really comes to debate a little the idea that the last layers of what these types of models are the richest at the level of information, as we saw previously, that they really house a lot of information in structural terms. But here what we are seeing is that at the level of ability to distinguish protein families, It turns out that the intermediate layers seem more useful, and not the last ones related more to the ability to distinguish contacts. So here in these projects, what they do is an attempt to classify the type of characteristics in such a way that there are some that are quite punctual, there are some that are periodic, like this fact that they appear every 7 residues in alpha helix regions, and there are others that are directed at small motifs at the sequence level. They are made from about 1 to 20 residues, from 20 to 50 residues, or from 50 to 300 residues. So what we observe is again this type of graph, where we see the number of characteristics found throughout each of the layers of the ESM2 language model. And for example, we observe that what would be the characteristics of short, medium, and large motifs are. They really do have a peak, and what are the intermediate layers of the language model, which also coincides with the distribution of characteristics at the family level. And what is suggested is that the last layers that seem to also recapitulate the structural information, since they would be specializing at the level of the probabilities that have to be computed in order to assign the probability to the mass tokens. And that is why perhaps the information obtained is reduced at the motif level or at the family level. This observation is not only unique to what would be the family of SM protein language models, but it has even been seen in other families of language models, such as pro-trans models or other variants of SM, where through some calculations that are relatively complex that allow us to know the geometry of this type of protein language models, a metric known as the intrinsic dimension is used, which allows us to approximate what would be the minimum dimensions necessary to be able to represent our data without losing information. And what we observe is again here, what would be the relative depth of these types of protein language models in relation to their intrinsic dimensionality. And what we observe is that there's a fairly large first peak in all the protein language models, and then there is a plateau. Then this plateau again begins to have another peak. And it turns out that precisely this plateau is where most of the information would be captured. That is, in relation to what these layers would be compared to these others, these are richer in information. And as you can notice, they really seem to be in the range of the intermediate layers of what each of the protein language models is, and not in the last layers of each of them, which was the convention to take. So what is suggested here is that precisely the information would be expressed better in the layers of this plateau, which would be the intermediate layers, and that it could really be a good strategy to use these layers and not the last layer of each of these language models to be able to do transfer learning tasks. These are obviously aspects that have to be investigated when training a specific model based on what your project is, so it does require more thought. It seems quite interesting how this default procedure, which was adapted from only taking the last layer, actually seems to not be as effective in being able to distinguish some other properties beyond capturing the structural information. We even found other quite interesting details from this other research that also suggests that perhaps the intermediate layers are also learning relevant information. However, in addition to that information, other quite interesting observations are also made. In this case, what is done is to study what would be the latent space of what the ESMFold model is, which, as you remember, ESMFold loses the structures of the proteins, taking advantage of what are the embeddings of the ESM2 model in its variant of 3 billion parameters. So in this case, we take the embeddings, we make a linear projection, and then we go through a multi-layer perceptron to generate an initial embedding for ESMFold, along with what would be the initialization of the pairwise representation, which will precisely be used to predict the structures of the proteins. And here in this research, what was found is that in what would be the dimensionality of this embedding, which is 1024 dimensions, really this organization of the latent space learned by ESMFold seems to be pathologically disorganized because the activations of each of the dimensions of this embedding, they are actually quite atypical because there are some that are activated up to 3,000 times more than others. Something that we are really seeing in this distribution of activations, whereas you can see there's a distribution that we see here where there are quite external values, such as these that are activated very little, but here there are also a few other activations within that dimensionality of the embedding that are activated too much. That when we focus on what this distribution would be, it would be relatively ideal or similar to what a normal distribution centered at 0 would be. But clearly in the entire distribution, it does not really reflect a desired distribution. That was approximately normal, but there seems to be some pathologies here that the authors point out because they are extreme values that do not demonstrate a learned normal distribution. And then, at least what is suggested here is that these massive activations could even be contributing to what would be the information present in the representations in the intermediate layers, because these massive activations are activated independently of the depth of the transformer, and that they would precisely be storing a large amount of information. Let's say that in this sense, if we wanted to think about it in terms of polysemanticity, perhaps these activations would be, well, keeping many concepts at a statistical level, and because they are keeping many concepts, they are maintained throughout what are precisely all the layers of the language model, and that would precisely cause them to be predicted even. Though we are not taking the last layer of what these transformers and these activations or this pathological behavior are not only on the part of ESGFold, but is precisely inherited from the protein language model with which it was trained, which is the language of an SGM2 in its version with 3 billion parameters. So what happens if we eliminate these 3 activations, which cannot be seen so well here in what this histogram is? But in total, there are 3 activations that are massive in relation to the others. Well, if we set the values of these activations to 0, it turns out that what would be the proteins that were usually well predicted, here represented in purple, they are now predicted in a completely collapsed form, represented here in green. And this is repeated for several proteins, as you can see, where when removing these 3 activations, the protein always completely collapses, which is suggesting to us that there is really too much structural information contained in just 3 channels of the dimensionality of the ESD-enfold embedding. And then here they take on the task of making several developments at the algorithm level, so that we can even reduce the dimensionality of the ESD-enfold, embedding up to 128 times, without losing the capabilities of being able to predict structures. What we observe here at the top is the dimensionality of the embedding with respect to what the DM score of the structural prediction would be. So when we use the normal embedding, well, we're talking about 1024 dimensions. And a TM score of 1 is precisely obtained, which tells us that it is a replica in this sense. But we can reduce the dimensionality of the embedding to 512, to 256, to 128 dimensions, and as you can see, the TM score is still preserved even with 0.99. That is, it has not deteriorated, despite the fact that it has already been reduced by 1, 2, 3 orders of magnitude. We can even reduce it to things as absurd as even only 8 dimensions of the embedding, instead of 1024 dimensions, and we would be having relatively good structural predictions of approximately 0.92 in the GM score, and only perhaps until when we go down to 4 dimensions, it drops to 0.78, which suggests to us that really both the protein language models with which ESGFold is trained and ESGFold itself, perhaps they do not really have a rich representation of fairly well-organized information, since their dimensionality can precisely be reduced, and perhaps they would be overtrained in this sense. Something that we will explore in more detail in the following slides. Another aspect that we also have to consider about protein language models is how we can build representations at the protein level, since there is really no consensus on this matter either. And precisely designing a representation that is capable of recapitulating a protein in its entirety requires reflection on the type of properties that we want to encapsulate in said representation. So it is not really that easy to think about which representation would be the most appropriate for the modeling purposes that we really want to approximate. So how is it that we can aggregate these learned representations at the amino acid level, which would be at the token level, into a fixed dimensional representation that represents the protein as a whole? As you may remember, we have what is the BERT architecture, where we practically have what is the ability to mask and unmask, and during this encoder-decoder procedure precisely, this type of embeddings is learned. And these embeddings can be used in a posteriori tasks through transfer learning, using other algorithms such as random forest, logistic religion, to predict aspects such as contacts, solubility, among other aspects. But then, how do we generate embedding representations precisely that the language model learns at the residue level, but that now represent a protein as a whole? And for this, there are several strategies. For example, there is being able to take an average of all the embeddings of each of the residues, and then simply average them and generate a representation that is of the protein as a whole. But this is precisely taking an average of all the dimensions of said embeddings, right? Another is to be able to concatenate each of the embeddings, and now not only have a classic vector of approximately 1024 dimensions, but now we will have a vector that has too many values that could perhaps be computationally expensive. Because we are talking about too many values in this sense. Or we can even train other neural networks such as an autoencoder that serves as a kind of bottleneck, where we start from all the embeddings that were learned at the residual level. And then you have to learn to represent at a fixed dimension what would be the representation at the protein level. When we go back to look at the literature, there really is a great diversity of the type of methods that we can use to generate representations at the protein level. But the one that is most used, by far, in the literature is simply choosing to take an average of what the representations are at the amino acid level to obtain the representation at the protein level. However, here they observe different methods. Some are parameter-free, that is, like this operation that simply means making an average. We also have to do the sum, or do other methods, for example, based on the Big Ranch algorithm, which is the one that Google uses or used to do its searches, as well as we have others that are precisely parameterized by other architectures, as this could be the case of an autoencoder. Again, although it would be quite advisable to actually be able to use this option of simply making an average, because if you have the time, the calculation, or if it is your goal to be able to compare this type of strategies, you could explore these other strategies beyond what is the simple average of the representations at the residue level. And this is also quite interesting, and is related to what predictive performance is across the layers of a language model. So there is really no consensus on what the optimal layer would really be to be able to have better performance at the level of prediction of various protein properties, beyond what would be the prediction in terms of structural contacts or three-dimensional structures. Here I show you an example. From a task that I used, for example, the language model called SM1B, to be able to predict the antibiotic resistance of beta-lactamases against a specific antibiotic called cefepime. And what we observe on the y-axis is precisely the Pearson correlation that is obtained from the predicted values with respect to the real resistance values of these beta-lactamases. And then we observe precisely the behavior of each of the layers of the SM1B model, where you can see that this idea that the last layers are the best is not really being reflected, but rather it seems that, for this specific case, what would be layers 14 or 15 would be the ones that have a greater ability to predict in terms of the Pearson correlation. And that his mistake also seems relatively sensible or a good idea to choose, although it may not be the minimum. For example, we can find that what would be layer number 25 is the one with the smallest margin of error represented by these lines, but its predictive capacity drops to 6.25 in Pearson correlation compared to these others that had approximately 65. But in general, what they can observe is that there is no clear performance that tells us which layer is the best to be able to predict, in this case, the functionality of a protein. Even for other projects, such as in this specific architecture, what is intended is to be able to predict the functionality of proteins using what is known as the genetubology term, which we previously explored in what is the class of protein function. In this case, they take advantage of the SM2 language model in its version of 3 billion parameters, and which, as you remember, has a total of 36 layers. In this case, what they do is take the representations learned from layer 36 35, 34. This averaging operation is done precisely. Then it is passed through a multi-layer perceptron and again it is concatenated. It is passed again through multi-layer perceptron in order to simply have a final representation that helps us to finally predict what would be the function that the protein would be doing. Here, as you can see, these are really arbitrary decisions, which are sometimes explained in the articles, sometimes not, and well, if they also have the computing capacity to be able to explore this, as well as the time, then they could give themselves to the task, but at the cost that it is clear that they need time to be able to determine which layer would be ideal to be able to make their predictions based on what we previously explored. Even, for example, when we look at the ESMFold architecture, the SM2 variant is used in its 3 billion parameter variant, which has 36 layers in total, and the dimensionality of its embedding is 2,560 dimensions. And then, in order to generate a representation that aggregates all the information learned by ESGM2, what is done is first to go through a weighted sum that has to be learned by the model. And subsequently, it is passed through a multi-layer perceptron that reduces the dimensionality to only 1024 dimensions. That is, it is taking the embeddings coming from all the layers of the SM model, which is clearly computationally expensive, and then projecting it to a lower dimensionality using the multi-layer perceptron. Here you can see with these three examples that there are some that use all the layers. Some will only use the final layers. Or in this case, perhaps what the intermediate layers would be like to be able to make your predictions. And again, this depends quite a bit on the type of things they are trying to predict, because predicting properties related to the structure of the proteins, such as contacts or their three-dimensional coordinates, is not the same as predicting in these other two cases what the function of the proteins would be. Something that we could expect is more complex to learn from this type of language models. And this is quite interesting because what the community's behavior has done is simply scale these types of protein language models left and right, without really reflecting as much on how much the results are being improved in general terms. Here what we see is a graph where the computing capacity is shown on the y-axis. Regarding the date of publication of various models in green that are general purpose or much more specialized models in blue to make some distinction. And what we observe is that, for example, what would be first models such as AlphaFold 1, since they had a relatively lower computing capacity, and subsequently this type of inverted computing has been escalating, reaching orders from 10 to 24 in terms of flops of floating point operations per second. To be able to train what are architectures such as the ESM3 language model or even AlphaFold 3, which in general what it tells us is that as we advance in time, we are investing greater amounts of computing to be able to train our models. Something that we see quite well represented in these three projects, where for example the largest protein language model that I am aware of is trained, which would be this variant called Explainability of approximately 50%. 100 billion parameters, also in this other language models are trained that we already reviewed, which are those of the SM family, SM2 in particular, which would correspond to approximately 15 billion parameters, or even others that have approximately 46 billion parameters, but which are of a more autoregressive nature, that is, generative compared to these two that are more of the BERT type in this sense. It's pretty crazy to think that we've scaled up to even 100 billion parameters, and we haven't really seen pretty amazing improvements in terms of the predictive capabilities of these protein language models. Here we observe again what the data of this model is, which is the largest that I am currently aware of to be able to predict properties at the level of structures, properties at the level of function, of the interaction between pairs of proteins or even what would be there. Applicability or thermodynamic stability of proteins. And what we find is precisely a comparison of what the large model of this project would be, which is represented by a T100B of the number of parameters, with respect to the models of the SM family in its different versions of parameters, where we see the model with 150 million, 650 million, 3 billion and 15 billion parameters. And now we use two strategies to be able to do fine-tuning and predict the properties. For example, structural prediction of contacts, using fine-tuning using LoRa-based methods, or doing fine-tuning by coupling a two-layer multi-layer perceptron to be able to predict said property. What we observe is therefore several slopes, where first in general terms it is suggested that the methods based on LoRa fine-tuning are relatively better for the four tasks to be able to predict these properties. However, when we do magical and complicated operations such as drawing lines and seeing that their slopes are also being reflected, we find that really the only task that seems to improve considerably is the one related to precisely predicting the structural properties that derive from this specific graph. So really, when you review the literature well, interestingly enough, it is only this structural prediction task that has been observed to scale as we increase the size of our models. And for the rest of the properties, such as function prediction, prediction of interactions between pairs of proteins or thermostability, a return rate is observed that actually decreases as we increase the computing capacity invested in these models, something that is known as diminishing returns in this sense. What we observe here is precisely the behavior of the predictive capabilities for various aspects, such as being able to predict secondary structure, being able to predict remote homology, being able to predict the degree of fluorescence of a protein, as well as its thermostability, using what would be a protein language model called ProteinBird, composed of approximately 16 million parameters. And what we observe in general terms is that, for most of the tasks evaluated in this publication, they don't really seem to scale as we actually train our language model. Something that, for example, we can see well represented in what would be on the y-axis, the precision of being able to predict the secondary structure, and on the x-axis, how much the model has been trained. Here we observe that as the training takes place, the model increasingly learns to distinguish in a better way to be able to predict precisely what the secondary structure is. Something similar happens when we look at the ability to distinguish remote homology, where in this case there also seems to be progress throughout the training phases. However, when we turn to look at properties related to the functionality of the proteins, such as fluorescence, measured in this case not through accuracy, but through the correlation of personnel on the y-axis, we observe that throughout the training, there really seems to be no improvement, and there really is a monotonous behavior that could even decline at the end of the training, or even for other properties related to functions such as thermostability. In this case, we even found slightly more explosive or random dynamics in such a way that it seems that there is not really a clear pattern regarding whether this type of dynamics are also about to be predicted at a functional level. Which is actually quite interesting, because as we reviewed, it seems that it is only the ability to be able to distinguish the structure of proteins, the aspects that are being learned by these protein language models trained only with sequences, and that what it is to model the function, well, it could really be much more complicated in this sense. And these are observations that have not only been made by these other models, articles that we previously explored, but that have been done by at least these other 5 examples, where perhaps I would strongly recommend reviewing this other one, where in general terms what they are telling us is that the current models of protein language models, among the consequences, it seems that they are not really well trained, and that they would even be overtrained. They have not reached this optimal training in terms of the architecture number of parameters as well as its projection in terms of the amount of data with which it is trained. So what has been found in this set of publications is that there are precisely these discrimination returns in terms of model performance that are not really growing as we scale our models, and that even performance plateaus have been found, where it really seems that in some conditions there is simply no more opportunity to find improvements in predictive capabilities, which also suggests to us that even what the current models are, in particular precisely the number of parameters that are used, is quite far from that optimal balance that could really impact being able to improve in terms of what the loss validation would be during training. And what this is really reflecting is that under the current training paradigm, where we have only adopted the sequences deposited in the databases, it seems that we have run into a kind of wall where, as we scale them, it really does not represent a significant improvement despite all the money and time invested in such a way that what we really need are better pre-training tasks. That is, loss, function, or other facets of incorporating multimodality to be able to train more and more better models that precisely improve their predictive capabilities both at the structure level as well as at the level of protein function. And for example, here we can see it relatively illustrated with the idea that the largest protein language models are not always the best option to be able to model some aspect. In this case, we see it illustrated with the ability to predict the secondary structure using two variants of the ProtRans family of protein language models, which are precisely the ProtT5 variants in their version with 3 billion parameters in blue, and in its version with 11 billion parameters in orange. And so, now what we observe is that they are also predicting the secondary structure when we train them only with a database called Big Fantastic Database, composed of approximately 2.1 billion sequences, in such a way that we find performances of approximately 82 and 81.6% to be able to accurately predict these secondary structure patterns. But when we train with a protein sequence database that is considerably smaller in terms of number of sequences, but is much more diverse, which is UNIREP50, represented here by a U50. This training approach was not only trained on this total of 2.1 billion sequences, but was also fine-tuned on this subset of approximately 50 million clusters of protein sequences that are much more diverse with respect to the type of masking dynamics that the transformer is carrying out, so that when this fine-tuning is done on a dataset that is more diverse at the sequence level, it turns out that the predictive capabilities of the protein language model are improved for both cases in its 3 billion or 11 billion versions, which would be reflecting the fact that the dataset with which we are training, or in this case, fine-tuning, also has a quite relevant effect on the predictive capabilities for a specific material. Here we have a second example where we now use the protein GYM benchmark to predict the fitness of various proteins. And in this case, we use what is the SM2 family of protein language models in its parameter variance from 8 million to 15 billion parameters. And we measure it through what is the Spearing correlation. And we observe that the smallest model of 8 million does not achieve good predictive capabilities until we reach the model of approximately 650 million, which would seem to represent the peak in fitness predictive capabilities, which subsequently decline even though we increase the scale of the model to 15 billion parameters. So again, really choosing the larger protein language model doesn't really necessarily imply an improvement in modeling capabilities for our specific tasks. And we're going to explore this specific behavior of predicting fitness level functionality in more detail when we investigate autoregressive models in the next few slides. So in general, let's say that to return to the general opinion of the community, here we have several quotes from various colleagues, for example from Eric Lindahl, who precisely suggests that progress in being able to predict structures and their functions, it really comes from being able to introduce more biology to our algorithms, and not necessarily making them much more sophisticated in this sense. We also have the opinion of Mohammed Al-Qureshi, who also suggests that adding what would be the biochemistry of proteins also tends to increase what would be the performance of this type of models based on deep learning, and even also allows us a window of being able to make them more interpretable. And in the case of Pascal Notting, what he makes to us is the suggestion that we really need a much more intelligent integration that is capable of combining evolutionary information as well as the structural information of proteins in order to improve the predictive capabilities of our models. Because until then, we had simply used the sequences left and right, without really looking at its representative capabilities at a functional or structural level. Something that in general terms I like to summarize quite well with what would be the opinion of another colleague named Kevin Yang, who really tells us that we need new ways to train our models, which we can compare quite well with what the AlphaFold architecture would be, which, as you can remember, is really not only about transformers coupled in a quite intelligent way, in a differentiable way from term to term, but also includes the development of quite well thought-out functions, such as the FAPE loss, the loss of the autoencoder, well, of the evoformer itself, or even statements that what they are doing is injecting or promoting to the model the fact that it learns to detect which sequences are at a high resolution, at a low resolution, or that they even follow the properties of the proteins that we find desirable, such as the fact that the bond distances are not precisely being violated or that there are no steric clashes. Until now, a paradigm had simply been followed where the canonical architecture of BERT was taken and trained under these archetypal masking laws. So if we want to inject more biology, obviously we have to develop both representations that can involve this, as well as laws function that allow us to precisely model both structural and functional aspects of proteins. And it is precisely what has been developed in recent years, such as the idea of making this type of protein language models that no longer only attend to sequences, but also now attend to structure information. That is, they integrate both information channels in order to improve representations. And in this case, we see some examples, such as the Zaprot architecture, or another that also seems quite similar, which would be Prot5, where in summary what this type of protein language model architectures do is no longer only tokenize at the residue level, as is handled for sequences, where each of the residues actually corresponds to token, but now the information of the tokens provided by the FALSIC algorithm or any other structural representation algorithm, such as RISIC, which we also previously explored, is integrated. And in this case, as you can see, we have an alphabet in uppercase letters corresponding to amino acids and an alphabet in lowercase letters corresponding to the alphabet of the structural state. So now we have a new joint alphabet that integrates both structural information and sequence-level information, which we can now proceed with the typical framework of these models, which would be masking. So in this case precisely, we represent the proteins simply from their tokens at the sequence level. And in this case, we have more enriched representations that integrate both sequence and structure through an alphabet that now includes two states for each of the residues of our protein. Finally, what we do is use the classic architecture where we proceed to impute precisely the values, mask them, and proceed to what is the projection to the embedding layer, and finally to the prediction precisely of which tokens would be most likely to be able to resolve said masking regions. And so now what remains to be seen is whether the structural integration through what is the FULSIC alphabet or any other structural representation really improves the prediction capabilities at a structural level or at a functional level. Something that we now see in these tests where what is done is to compare, for example, this type of models that integrate both sequence and structure, which in this case we are evaluating the SAPROT protein language model with respect to other protein language models such as one based on graph neural networks, or one inspired by the evoformer architecture, where we take the attention maps of the SM language models, but we also include a certain structural bias due to a pairwise representation. And we do this to be able to predict the structures using several datasets. In this case, we have datasets that are trained and validated, which would come from AlphaFold 2, which in this case are represented in green and pink, and we can see that in this case, from the architecture inspired by the evoformer, there does seem to be a good reduction in what is the training loss and the validation loss. However, when we now prove that the structures are also actually being predicted in another independent set, which would be this one in green, which would correspond to the structures of the PDB, we can perhaps think of it as a test set in this sense, Really here, the loss function is not really being modeled well. In this other case, well, not the loss function, sorry, it was an error. It was the validation loss. In this other case, for the graph neural network, there seems to be quite a consistency between what the validation loss is in terms of the training and the validation set of the AlphaFOT2 data, but when we externalize it to what the PDB sets are, your loss here actually gets worse and remains monotonous throughout the training. And in the case of Zaproth, it seems that it is reflecting good integrative capabilities of these two alphabets in such a way that it is really converging quite consistently for the three behaviors of the two validations present in the three data sets. So then I would be suggesting that it really can be a good strategy to be able to include structural alphabets along with sequence-level alphabets to improve training capabilities, and that it obviously becomes a little more stable over time. When we turn to look at what their predictive capabilities are, for example, in various tasks related to thermostability, the capabilities of predicting interactions between pairs of proteins, the capabilities of predicting metal binding, even detect their enzymatic SN numbers, detect their function through genetology, or detect their subcellular location. And we compare precisely what SAPROT is, which is this hybrid model of structural and sequence-level alphabet, with respect to other models such as those we previously explored. It turns out that it is the one that achieves the best performance in different tasks. In general terms, you can even see that what SAPROT really is. Well, it even surpasses protein language models like SM2 in its variant of 15 billion parameters, despite the fact that it is obviously much larger in this sense. So the largest models are not necessarily the best at predicting some properties, and perhaps this deterioration in their predictive capabilities could be related to what is an overfitting on the part of these language models, something that we will explore in more detail later. When we look at autoregressive models, since this actually does happen with the SM family type of protein language models, even the ProTrans family. Because of this, attempts have even been made to explore new ways of training this type of protein language models, such as the new architecture of the SM3 family, where it is really said that we have already reached a multimodal protein training paradigm. In this case, in order to train SM3, information was included, yes, at the sequence level, but also now at the structure level, using an autoencoder. It is similar to what FALTIC would be with a structural alphabet, but now information from the secondary structure regions is also being integrated through this approach called SS8, also information on what would be the extent to which the structures are being exposed to absorbency. Even functional information from the annotations provided by databases such as UniProt, which note, for example, if a residue is responsible for carrying out some catalytic activity, if another set of residues are responsible for carrying out binding to a ligand, as well as general descriptions of the protein. That is, it is incorporating text-level information from the descriptions that we as humans have made, as well as information derived from the structure of the protein. And this goes through several layers of a transformer, obviously much more sophisticated, in order to learn to represent them in a comprehensive way in this sense. And precisely what the SM3 approach is quite interesting because, although it is a masking type transformer similar to BERT, several methods are also applied that make it capable of having generative behavior in such a way that it can make protein generations in a non-conditional way. And this is projected precisely in these maps, that we really see a reduction in the dimensionality of the SM3 embeddings, projected to only 2 dimensions on the. Where we see is that really relatively plausible structures are generated in terms precisely of their folding patterns, but it also has the capacity to generate quite diverse sequences. In some cases, we can generate sequences that are quite similar to the sequences with which it was trained, which would be represented by these yellow points. But in other cases, quite diverse sequences are also generated that reach up to 10 to 20% sequence similarity, represented by those other blue points. And we can even condition the generative capabilities of SM3 to various contexts, as is also the case with diffusion models, only in this case talking about language models. In this case, we can condition the generative capabilities of SM3 to what would be a prompt related to a structural reason, for example, some catalytic site. And then we have to give it precisely this three-dimensional arrangement of the motif. And then SM3 is able to predict which scaffold could correspond precisely to said catalytic motif. We can also condition it in terms of we want a protein that is completely beta, but becomes capable of adapting to this other catalytic motif. So that what is the result is precisely a protein composed of pure beta sheets, where this catalytic motif is precisely found. Or it can even be conditioned to specific foldings, such as Tim-Barrel in conjunction with, for example, magnesium on one sites, in such a way that it is then understood during the training that all the information must be combined to be able to really couple what would be a magnesium site to what would be the architecture of Tim-Barrel, which really reflects exceptional modeling capabilities in that sense. And that did demonstrate to a lesser extent what the publication of this language model is. And taking advantage of the fact that we explore the architecture of the SM3 protein language model, we then move on to the second part of this class, where we explore the generative AI algorithms applied to proteins, where among so many architectures, perhaps the protein language models stand out the most, as well as diffusion models. But we are not necessarily limited to those two types of architectures to be able to implement generative algorithms. As you may remember, in the previous slides, we explored BERT-type protein language models, which are quite different from what GPT-type models would be. In that sense, there are several properties that precisely make them distinguish one from the other. But perhaps what I would like to highlight above all is the type of information that is provided to them to be able to make their predictions. Again, remembering how BERT-type models work, it is simply the random masking of various residues of the sequence. And during training, these types of models have to learn representations rich in information, such that they then allow us to predict which would be the possible residues that would go in these positions. And in this sense, it is relevant to recognize that, for example, if we stop at this point in the sequence to predict which would be the most probable amino acid, we have access to information both on the left and on the right, or in general, words to the entire sequence in this sense. Something that really does not happen with GPT-type models, because in this case, the predictions are based to predict the state of a residue on what would be all the residues preceding said position. And this happens for the next residue and the next residue. And this implementation also makes it easier to develop its application to what would be generative models, or even other aspects such as probabilistic ones, where we can designate a likelihood to a sequence which we can see reflected as an approximation of the fitness of said protein, which is easier to implement under GPT-type architectures and not so much in BERT-type models, although there really are, let's say, some methods to approximate these behaviors. And in this sense, we also find different models that, let's say, have been quite recognized in the generative AI research community, such as the protein language models of the PROGEN family, or the ProdGPT model, which we'll explore in more detail in the next few slides, in fact. But to be able to talk precisely about generative AI, we have to be quite clear about what a generative model is. And in summary, a generative model is one that learns a distribution, which tries to approximate the distribution represented by the data with which we are training our algorithm. In this sense, as you can see, we have several black points, which would be point x1, point x2, In this case, our original model is represented by a function pdx, and what would be the distribution learned by our generative model is simply called pgx to distinguish that it is a different function that could also be ax, gdx, or hdx, any other name you want to call it. And precisely what this model does is describe a probabilistic distribution that is defined by two key factors, which are precisely a latent variable and a neural network, which what it is going to do is take the information coming from this latent variable and transform it into something structured that could be images, text, or proteins in our case. In this case, we use what are latent variables, so that they represent a kind of distribution of the data. And as you will remember, one of the distributions that is most used in statistical terms is the normal distribution or Gaussian distribution, simply because it is also the most optimal in computational terms, but also because it is the one that perhaps allows us to represent a greater diversity of the data in general terms, since if we used another distribution that may be biased to one of the tails, then we would not be representing the majority of what our data would be. And in this case, this latent variable, which is really representing a normal or Gaussian distribution, we could really interpret it as a source of ordered noise from which we are going to sample coordinates, and which will be transformed precisely by our generative model in order to now have something structured. And precisely our generative model in question, since it is a neural network that is parameterized by a set of parameters that would be precisely the weights of our neural networks, these weights and biases of what we previously explored in the deep learning class, but also depending on its architecture. As we saw, some architectures have inductive biases that allow us to reduce the space of hypotheses that this type of models would explore in order to be able to say more accurately what would be a distribution that we want to reflect. And both the architecture and the parameters of our neural network, if we modify them, will precisely represent in a greater or lesser way a learned distribution that resembles the distribution of our real data. And this is precisely our task, to find the set of parameters that allow us to approximate both distributions as closely as possible. And here we must make a small nuance, that although we really do this by computing a loss function, it is clear that it will depend precisely on the behavior of said loss function, but also on the way in which we are displaying our data. And as you remember, really when we train a neural network, we don't give it all the data at once, because that is computationally impossible. But what we do is train using these small batches of information, or batch highs of what the architecture is, in such a way that we sample more and more different regions of our real distribution, and then through these samples of our real distribution, the model must learn to generate a learned distribution that becomes as similar to the distribution of real data. And here we must recognize something quite relevant, which is the intrinsic biases that are associated with what each of our data points would be, since it is clear that the way in which, for example, we carry out our measurements to collect said data, or the way in which we curate our data to be able to include them in our dataset in general, they really are the universe of hypotheses that our generative algorithm will aim for. Learn this distribution. For example, in the event that for some reason our instruments have not been able to sample this data that represents this part of the distribution of the entirety of our phenomenon, then simply the distribution learned by our generative model would be biased to only this small region of the entirety of the general distribution represented by the real data. Therefore, we must recognize that the methods that we have for measuring our real data, as well as the inclusion of them in our dataset, will obviously be a source of bias that can be seen both in their benefits and in the biases that these types of generative models have. And this is precisely what is intended to be made clear in this other slide, where different distributions of the data generate since they can generate or induce different parameterizations of this latent space, even though we are representing the same object. In this sense, let's say that we intend to model proteins in general, however. As you remember, we can represent proteins with multiple facets. For example, we can represent them by an alphabet of 20 amino acids at the sequence level. We can also represent proteins by their three-dimensional coordinates of their protein structures, or we can represent proteins through textual descriptions of what their activity would be in terms of, for example, highlighting only those residues that are capable of carrying out what would be binding to a ligand, catalysis, or that are more relevant in thermodynamic terms. And this is precisely what we observe in these comparisons of what would be different training regimes with respect to the learned distributions, where in all cases, whether we have trained a model at the structure level, a model at the sequence level, or a model that includes both data modalities, both sequences and structure, a fairly characteristic learned distribution will correspond to it. Here you can see that really. Although they are distributions of this latent space, let's say a little similar, each one is different from each other, and each one of them will explore a characteristic set of hypotheses that perhaps can serve us to a greater or lesser extent depending on the projects that we are developing. No. And this is quite relevant to recognize because of how we curate our data, how we represent it, how we show them, and even how we evaluate our models during training, is that we really see an effect on what the behavior of our model would be in its predictive capabilities, especially on regimes that are known as out of distribution, which we see in this new diagram, where let's say that we train a model for predicting the functionality of a protein or fitness. In this sense, we see that the function of our proteins is simply to be able to bloom in an even greater intensity. For example, in this protein that does not bloom as much, and this one that is quite well illuminated. So we train precisely on a data distribution seen as this map of different densities, so that once we train on this data distribution, what we want to happen with any algorithm is that it becomes capable of generalizing to new sequence spaces where there are new proteins that were not used during training, and yet the model is capable of detecting which trajectories would be the most likely to identify a region within this latent space where precisely the proteins with the greatest fitness in this sense lie. We are going to explore this generalization property in a little more detail in what this class is, but also in depth in what would be the type of data and biases. Here we see a brief example of the benefits we have by being able to unify several modalities and thus be able to have access to a greater set of information. In this sense, in this project, an architecture called ProtRec is presented, which what it does is learn a space where they are unified. Three modalities to represent proteins. One that precisely represents them at the level of descriptions of structure and function, through text, using a language model that is specialized in scientific literature, and is called PubMed-BERT. We also now include a protein language model that will precisely represent proteins as embeddings using the ESM2 language model in its 650 million parameter version. And we also include what these representations are learned by Foldseek's structural alphabet, so that we now have information on the text descriptions, structure and function of the proteins, as well as the sequences themselves and their corresponding structures. Therefore, it is clear that we have to have these descriptions in databases at the text level, at the sequence level, and their respective structures, which in this case we can even have access to through predictions made with AlphaFoov. And so now what we see is, in this latent space, the combo is that these three modalities of information that describe the same object are unified, but that now we can precisely have access to their other two counterparts in the event that we start from some input. For example, in this case, we are providing you with a protein in which, for example, we do not know its function. And then in this latent space during its training, the model is able to distinguish which descriptions of its structure or its possible function are most related to what our input protein would be. Something that, for example, we see reflected in this case, where we provide this structure of which perhaps we do not know its information a priori, and thanks to this mechanism of being able to map in this latent space what it's possible. Functions would be. Well, now we can recognize that it is an enzyme that catalyzes a conversion of this rather complicated compound that I cannot mention, among other possible functions that in fact ProteinMPNN ranks us as what would be the most likely function that our protein would be performing. In case we also have this other example, where on this occasion what we do is give it a description, but at the level of the function that we want to find for some protein, which, for example, could be a protein that is green and fluorescent, and then thanks to this understanding of the three modalities, then the model would show us which sequences would be the most likely to satisfy the description of the function that we provide as input, or even also find which structures would be closest to some structure as input that we have given, and that precisely in this latent space, since they become more similar to what our query protein would be. And this is quite interesting because, in general terms, ProteinMPNN actually supports up to 9 ways of searching for information, which, for example, could be from structure to text, from text to sequence, and from sequence to structure, as well as all their possible combinations, which is really something magnificent in terms for us bioinformaticians, since, for example, we previously did not have access to this type of algorithms that, given even just the sequence, could hypothesize what type of functions or structures could be related to said sequence. And now, thanks to these capabilities of this model that unifies 3 modalities, we can access even more information and subsequently curate it to stay with the best candidates that we could perhaps explore in the laboratory and thus validate if it is, for example, a function that the algorithm is suffering from. Even we can take advantage of these capabilities of being able to integrate multiple modalities of proteins and build something similar to a chatbot, such as ChatGPT. But now a chatbot specialized in protein biology, which in this case is called Ebola, or I don't know how to pronounce it very well, where again what we do is use the data from Uniprot, particularly 2 of its databases, which would be SwissProt, which is the one that has the greatest descriptions at the level of its sequences and structures, along with another that does not have as many annotations, such as Tremble, which also has several sequences in this sense. And then we can precisely take advantage of ProteinMPNN to precisely allow us to recover information in those 3 modalities. So that for each protein we now have descriptions of its possible family, whether for example it binds or not to nucleic acids, whether it carries out any catalytic activity, whether some disulfide bridges are relevant, whether it is perhaps related to a specific pharmacological medical treatment, or if even in order to carry out its activity, it requires the presence of some cofactor. All of this is information that enriches the description of the protein beyond its sequence or beyond its structure, something that we can precisely take advantage of to build a chatbot that now, by constructing a set of questions as well as the answers that would be associated with said questions, we represent these questions through embeddings. We also represent our proteins through embeddings, and then this transformer is responsible for unifying several modalities based on the fact that they are also capable of answering a series of answers that we give as an example. And thanks to this understanding, we now have an interface where we can provide a structure at the sequence level or at the 3-dimensional coordinate level. And chat, for example, regarding aspects of its function, regarding whether it can be implemented as a medical treatment, among other aspects. Something that I encourage you to try on what would be your web server, which is also quite friendly and does not need a deployment. Let's say, quite complicated in bioinformatics terms, but we can simply access a page and simply chat about our proteins. We also have other quite interesting generative guidance applications, such as being able to translate text descriptions into what would be structures, so that we can even design sequences using what would be a structure as input or prompts. As input, as is precisely what is presented in this specific model. In this case, for example, which is this model called PINAL, since it is capable of generating sequences from text description. And in this case, for example, I give you what would be the favorite enzyme of this course, which is this class of beta-lactamase, capable of degrading beta-lactam antibiotics. And then in my prompt, well, precisely I give you this description in a little more detail, and then the model, through its integration training of both facets at the level of structure, function, and structure. Well, it is able to suggest several hypothetical sequences, which in this case I folded using AlphaFold 2, and we can see that of the total of 10 structures, only about 6 would correspond to a fold of an archetypal class A beta-lactamase. And these are completely failed examples, because they did not really represent this type of folding, which I already recognize quite well. But it is quite interesting that even through what would be a description of the type of function that I want to have in my protein, the model is able to suggest some sequence that is capable of satisfying said prompt or said textual input. And this is something that is obviously not only seen in this example, but also in what would be its preprint, where in this case the prompt given is, please, design a protein that is an alcohol dehydrogenase. And then obviously they did not generate only 10 sequences, but they did it on a larger scale, where filters were applied such as that they were greater than a PLDT of 80, that their prediction at the food level of their error became greater than 7, and that even the score that ProTrack gave you regarding the plausibility of their function became greater than 15, for a total of approximately 1,600 sequences, and that even the authors took on the task of being able corroborate. Yes, they are proteins that are expressed in a cell, but they are also capable of carrying out the function that we requested of them, which was precisely a protein, which is in fact more of an enzyme, which is an alcohol dehydrogenase. So what is shown in the text is that 5 out of 8 of these enzymes really manage to be expressed, purified, and therefore really have their function. So it wasn't just about, say, that remain at a bioinformatic level and that comply with certain quality filters, but are already truly crystallized into a reality seen from what would be the experiment itself. Well, now once we review precisely these examples of how we can unify multiple modalities to be able to model the same object, which is proteins, we move on to illustrate the differences. 4 major sub-branches that we find within generative AI applied to protein science. As you will remember, we really have a whole class to explain this type of models, which are folding, where we go from a sequence to a 3-dimensional structure. So on this occasion, we are not going to explore them in detail here, and obviously in order to have more information, I invite you to verify what the AlphaFold class would be. In this case, we're going to focus on these other 3 major types of generative models. In this case, for example, we have sequence generation models, where we can clearly start from a completely general way, or even from a conditional way to start from some set of information that is reflected in what would be hypothetical sequences, where, for example, we have several models, such as ProGen protein language models. But GPT-2 or 1 that is specific at the enzyme level is called enzyme control. We also have another facet of generative models that, in this case, use the same information that can be conditional or non-conditional to generate structures. Precisely how some models such as Rosetta Full Diffusion or Chroma would work in this sense. And finally, we have another facet which is known as inverse folding, where what we do is start from a structure and then investigate what would be the putative sequences capable of folding in said form. something that is precisely known as inverse folding or also as fixed backbone design, because we are setting the possible hypothesis space capable of satisfying this specific folding, where we find several models, but perhaps the most representative are ProteinMPNN, as well as SM inverse folding. So now let's proceed to what the sequence generation models would be to make them clearer. Perhaps, well, I was even forgetting about these, where they are no longer only being modeled at the sequence level and at the structure level, but both facets are integrated to really reach a structure. That is, they are integrated both into the modality of their three-dimensional coordinates, as well as into the information provided by the alphabet of each of the residues, so that they are modeled precisely in parallel, and then we even have perhaps more plausible hypotheses in this sense. We also find some algorithms such as protein, plate, or protein generator. So now we proceed to investigate what the sequence generation models would be. And as such, really these models, as you can see in this small image, simply must be able to learn what the most probable residuals would be for a specific region, which can even be in an autoregressive manner, or it can even be in a way that is called filling or infilling, where we can provide different context windows at the level of the residuals, and then the model must be able to distinguish which would be the most probable residuals for said region. In this sense, one of the first autoregressive or generative models of protein sequences was the ProtGPT2 model, developed by Nolia Ferraz, in collaboration with Stephen and Birgit Hocker, which, as you can see, is a model that was already published in approximately 2022, although perhaps we can subtract it a little more from earlier in its preprint, approximately in 2020. And here what we observe is precisely the generative capabilities that this model has of being able to generate sequences that become quite similar to the natural proteins that we have. In this case, what we observe in this protein graph are a total of approximately 60,000 sequences that make up the proteins deposited in the SCOPE database, along with generations from ProtGPT2, where we will connect two proteins as long as during their alignment they have more than 20 residues that are aligned, and that they also have a probability of homology of more than 70% provided by this alignment tool called hammer search, where you can really see that we have the various architectures of the protein structures. For example, all alpha, all beta, alpha beta, or various combinations. And the white dots are the generations of ProtGPT2, where in general terms when we predict them with ESMFold 2, they have, let's say, PLDT metrics of greater than 70, so we could say that they would be relatively plausible, but they fall in line with what the proteins that we see in nature would be. You do whatever that means. And in that sense, well, it is obvious that we have to explore through various experiments that our proteins not only remain in terms of PLDT, but could even reflect other aspects that would invite us to think that they are proteins that are successful at the laboratory level. And for this, several experiments are presented, but I would only like to return to two here, where, for example, the first experiment is to take sequences generated by ProtGPT2, predict their structures using ESMFold 2, and use the Rosetta suite, this suite of algorithms that allow us to characterize the structures of proteins in energetic, statistical, or physical-chemical terms in general, using precisely an energy quantification protocol called relaxed backbone. So this is precisely what we are seeing in these two graphs, where we have a set of sequences that are natural, that is, they come from deposited databases such as UniProt. We also have a dataset corresponding to proteins generated by ProtGPT2, and a set of sequences that were generated completely at random and therefore would not have a plausible structure, where we can see here on the yes axis what your score would be through PLDT, through prediction with ESMFold 2, where we observe that natural proteins have approximately 80 PLDT, Proteins generated by ProtGPT2 have approximately 62 PLDT, and those generated completely at random have a PLDT of approximately 40. And when we quantify how much the energy of each of the residues of these structures would be, well, we have precisely the compared distribution between these three facets through what would be this energetic function of Rosetta that is presented as really Rosetta units. And we observe that what would be the distribution of the energies of the residues of natural proteins is relatively similar to the distribution of the energies of the residues using ProtGPT2, and that both are considerably different from what would be a distribution of residuals that are completely random, which would be representing the idea that the proteins that ProtGPT2 is generating actually have a relatively similar distribution to the natural proteins that we find in the databases. As a second experiment, we have the idea of doing a homology verification. Assuming that if we can recover homology, it is because more plausible sequences result from those generated by ProGPT compared to sequences generated completely at random. Again, then we start from our sequences generated by ProGPT2, and then we blast them or search them with this algorithm called HH-Blitz against the database called UniClass 30%. And then here we see precisely these curves that, as you remember, what we explore in what the class of protein structure is are the twilight zone graphs, where, for example, we can find for natural proteins that we are really finding homologs, since they fall within this margin where we can detect homology with greater certainty. In the case of generations created by ProtGPT2, we also found that most of the proteins synthesized using this generative model, we are also able to find homology in this database. Therefore, it is also suggested that they are not really completely random sequences, but rather that they are sequences that do result in a distribution relatively similar to the natural ones. Something that does not happen when we check whether the sequences generated completely at random. Well, we can find homology in the databases, where it turns out that 93% of these sequences that were generated completely at random fall within this margin of the twilight zone, where we can no longer really be certain that there is homology detection. And in this case, it would only be 7% of the sequences that fall within this twilight zone, and in the natural sequences only 4%, which would still be suggesting to us that the distribution learned by ProtGPT2 is relatively similar to the distribution of natural sequences. Obviously, there are more experiments, but obviously this can be explored further in what would be the publication and its supplementary material. But obviously, ProtGPT2 was one of the first generative protein language models that could serve in a non-conditional to conditional way. And this obviously captured the attention of several researchers to be able to extend generative capabilities to even more specific purposes. For example, it could be the ability to generate enzymes given what would be an enzymatic commission number, or that number. So in addition to tokenizing the protein sequence, now in what would be its header, we also assign the enzymatic commission number as a source of information. And this is precisely what this new model of the protein language called enzyme control is responsible for learning, where it learns to unify these two modalities at the sequence level and at the level of enzymatic commission number. It is then able to distinguish which enzymatic functions would correspond to which sequences in this sense. And this is precisely what we are observing in this new diagram, where what we do is take the generations carried out by enzyme control, and through their embeddings, then project them to two dimensions to see that this information is also being organized. And it is quite interesting to see that what this two-dimensional distribution would be seems to be reflected in terms of the number of commissions in sematics, where we find 2. This whole set of sequences that would correspond to this catalytic function, this whole group would correspond to this other specific catalytic function, and so the other groups where even when we do homology searches with Foldseek, We also find in some cases that we do not even find, let's say such similar folds, although they are from a fold that we already knew previously, so it suggests that new sequence spaces are being explored that could be relatively plausible to validate in the laboratory. And we can even extend this paradigm even further now with this new protein language model called RexSIME, which in this case, given what would be a chemical reaction of what would be the substrates and products, then the model must be well enough skilled through its encoder-decoder architecture to find what sequence would be capable of carrying out said catalytic reaction, which is quite interesting because now we are giving it, let's say it like this, a description of its catalytic function, but now at the level of what its products and its treatments would be, and that the model must therefore be skilled enough to learn to decrypt said information, to suggest hypotheses about the sequences that would be carrying out said catalytic reaction. Here we also now explore what fine-tuning is, which you may also remember from the protein language models class. What these types of generative models really do is learn a latent space where proteins are represented, where this latent space should provide information in such a way that, let's say, a protein that is not at all plausible because, let's say, it does not have any structure or is completely collapsed, since it would be found in regions of this high-dimensional latent space, completely different from another region of this space, where the proteins that could be relatively plausible do lie, which we can even sample in different ways that also help us generate sequences with greater diversity or less diversity, depending on how we configure these two main sampling parameters, such as the temperature, which in summary is a measure of the randomness during this sampling process, as well as other procedures known as top K or top P, which is precisely the way in which we choose our tokens to be able to precisely generate these sequences. At the level of, for example, frequentist or probabilistic sampling of said tokens, and thanks to these two parameters, even we can generate increasingly diverse proteins, although perhaps, for example, our protein language model is not conditioned on some type of specific information, such as information from enzymes or information from structures. But even if we say that we have relatively complicated cases because perhaps our scales are overpresented in the databases, we can enrich these regions of this latent space through fine-tuning procedures. We're then now, during the exploration of possible, most probable tokens to follow during this autoregressive approach, what we do is maximize this region of the latent space where sequences would correspond in this case, for example, of the team bar type. And then now, through what would be fine-tuning. At the level of Tim barrels, that is, we have a protein language model in which now we ask him to readjust its parameters in terms of this type of three-dimensional folding, because then the generations that come from said protein language model are going to be more biased to this folding, because precisely we told him to start from this specific set of hypotheses. And this fine-tuning procedure, as you will remember, we can do it, let's put it this way, in two main ways. One is to actually update all the parameters of our model, which is quite complicated because it requires high computing power. That is, you need GPUs such as a 3090, a 5090 with approximately 24 GB of VRAM for small models. For larger models, you would even need approximately a 100 with 80 GB of VRAM, which not all of us really have access to that type of hardware in this sense. Therefore, different optimization techniques have emerged that, as you may remember, could be precisely the low-raw fine-tuning methods, where we can decompose our model weight matrix into two submatrices, which would be matrices A and matrices B. And through various linear algebra tricks, well now, we simply have to train a set of parameters that is considerably smaller than training all of our model parameters to carry out this fine-tuning based on some protein or some family of proteins that we want to model with greater specificity. And so now we explore precisely the defect that this type of fine-tuning has, and what would be the generations of this type of models. Here we see three models regarding, for example, how many sequences it is making, as well as how much sequence similarity they present with respect to the natural proteins present in databases such as UniProt, where in this case these three models were given the task of generating sequences that are of the triose, phosphate, isomerase type. And then we find three models. What would be enzyme control, which has this ability to be conditioned at an enzymatic level by giving it its enzymatic commission numbers during the toucanization and training process, among other aspects. And what we observe is that this type of generation is more similar to what would be the proteins that we already have in the databases, which would be natural proteins. So perhaps we could assume that they could be, well, enzymes that would perhaps have a greater probability of being expressed in the laboratory, because they are more similar to the proteins that we find in nature. Then when we observe the generations carried out by ProtGPT2 in a non-conditional way, we have to apply several filters such as those that correspond to said fold corresponding to a triose phosphataseomerase, and then only have that type of folds and evaluate them right. Where now what we see is that the proteins generated by ProtGPT2 are considerably more diverse at the level of sequence similarity, because the majority present approximately 30% similarity with respect to the proteins deposited in the data vessel. Then you would be, for example, maximizing diversity at the sequence level. And an intermediate point would be, for example, the one provided by ProtGPT2, but fine-tuned to be able to precisely maximize the possible hypotheses that would correspond precisely to triose, phosphate, and sumerase sequences. Where what we see is that, in this case, most of the sequences fall within a margin of 50% sequence similarity. You can keep these types of aspects in mind when, let's say, generating your own sequences based on, for example, if you want to maximize sequences quite similar to what the proteins deposited in the databases would be or maximize their diversity, since in this case, for example, with generations not conditioned by ProtGPT2, they can precisely explore a sequence space that is not also represented in the databases that we currently have. These other aspects have also been explored by other researchers with their respective protein language models. And in this case, we explore precisely what the family of models called ProGen is, which are presented, for example, in these two publications, where in this case what was given in the author's task is to even enrich these conditional descriptions so that they also serve as facets to generate precisely sequences that, for example, are made. Well, let's say about a specific organism, for example Homo sapiens, that they fulfill a specific function, for example that it is a noctin, that they are located in some compartment of the cell, for example in the cytoplasm, also that they are, let's say, associated with some process, such as some cardiac pathology, and then through this training where these modalities of information are provided, then the model learns to relate them, so that we can have greater control during the sequence generation process. Which precisely, once the integration of these three modalities has been learned, well, more than three modalities, then we can now exploit the model in the sense of generating sequences, predicting their structures to characterize them, and choosing the best candidates to be expressed in the laboratory, and thus corroborate their function. And this is quite interesting because now we can say, for example, give me an amylase from Homo sapiens that meets several characteristics at the text level, and then the model must be capable of generating sequence hypotheses that satisfy these descriptions, which is precisely what occurs in the task of demonstrating experimentally, or using the ProteinMPNN protein language model. Fine-tuning is now done with a set of specific sequences corresponding to the lysozyme family, for a total of approximately 56,000 sequences that were used during this facet of fine-tuning. So, what is being indicated through fine-tuning is that it can model sequences with a more plausible probability distribution for this specific protein family. So now what they do in laboratory terms is we generate 100 sequences, and we see that they are also being expressed in the cell, right? Not only as a bioinformatics artifact. And this is precisely what we see in this graph, where on the y-axis which we also say is being expressed with respect to the x-axis, which would be a metric that groups proteins according to their margins of sequence similarity with respect to databases such as UNIPROTO, where, for example, the generations that are within a range of 80 to 90% sequence similarity of the total were able to express high levels of expression at 84%. And even more interesting would be the margin of between 40 to 50% sequence similarity, which is telling us that they are much more diverse at the sequence level. Also expressed, say, in high quality or at a high level of expressibility of their amylase function, approximately 5 of the 10 proteins generated using this model. And this is quite interesting because it is telling us that although they are quite divergent in terms of sequence similarity with respect to the proteins that we already know in the databases, the model is capable of generating sequences that are actually carrying out the lysozyme function. And that even when we express precisely all of our 100 generations of sequences, along with what would be other proteins that we show randomly from these databases that are natural proteins, it is quite interesting that in both cases it was highly expressed in 72% of the cases, which is telling us that the model really learned to identify, let's say, which proteins could have a greater probability of being expressed in the laboratory using this fine-tuning approach based on isozymes, something that, for example, you could apply in your projects if you intend to maximize the experimental success for a specific protein family that they want to explore. And this was even taken to the next level by presenting their next family of protein language models in what is the ProGen family, where this preprint presents precisely the architecture of ProGen 3 that they trained with 3 different versions of parameter models. In this case, the version with 340 million parameters, 3 billion parameters. And what would be the largest model within this type that is generative of 46 billion parameters? And then what we observe here in this graph is a total of approximately 9 million sequences generated by these models, where what would be their embeddings are projected to 2 dimensions. And what we observe in general terms is that the sequences generated by the largest model of 46 billion parameters, they contain within them the sequences generated by the model in its version with 3 billion parameters, and the smallest model with 340 million parameters, which is suggesting to us that as we are scaling our generative or autoregressive models, whatever you want to call them, we can access a greater diversity of sequence space. Something that they even took on the task of exploring in terms of experimental validation, with approximately 300 sequences that, as you can see, are represented by these little gray dots, where there are some sequences that were generated by the 3 models, such as these regions or these other regions that, as you can see, in this case the densities are higher, so they could be sequences with a higher probability, but the larger model, it is capable of generating sequences represented by these other dots that are on the periphery, to which the smaller versions do not have access, since then they would not be able to explore said sequence spaces. Then there will be some variants that simply cannot be generated by models that are relatively small, and especially those based on only about 340 million parameters. And this is quite interesting, because at least in what the prepress would be The authors suggest that scaling the models allows us to have sequences that are more valid in terms of various filtering criteria, also that are much more diverse, which even allows us this generalization outside the distribution where we can observe that the largest model is capable of generating sequences that the smaller models are not. And they even improve some other capabilities, such as the fact of being able to do this activity known as infilling, or the filling of a small region of a total sequence, as well as the experimental success that really seems to be consistent across the three models in this specific architecture. For example, what we see in this graph is a percentage of how many of these sequences pass different filters. For example, in this case, a first filter is applied to discard those sequences that have more than 25% of contiguously repeated amino acids. Where we can see that in the case of the largest model, with approximately 46 billion parameters, 88% of the sequences did pass this filter, compared to what would be the smallest version, where only 54% passed said filter. Which first suggests to us that, let's say, the type of sequences generated by the largest model are more plausible compared to the smaller model, something that is also reproduced when we apply two other filters, such as the fact that it was a sequence generated, and that it has finished, and that it has been finished with its corresponding termination tokens, or that even through what would be its alignment to the databases, be covered with approximately 80% of the sequence. Where, as you can see, what would actually be the generative capabilities of the largest model of 46 billion parameters is better in terms of the possible sequences that we could proceed to validate experimentally. Even when we cluster our universe of several million, generated by what would be our generative models. And then we see those clusters that so many peers recover when we blast them into the databases. Well, we see that even the number of sequences recovered by each of our language models is considerably higher when we use the larger version of 46 billion parameters. In this case, we see that when we take approximately 750,000 clusters of these sequences and blast them against the databases, we recovered approximately 4 million clusters. Something that does not happen when we use the smallest version, which would be the one with 340 million parameters, where of the 4 million clusters, we now recover approximately 2.5 million clusters, which is telling us that they are more plausible, or suggesting that the type of sequences generated by the largest model are more plausible. And well, this is even more interesting when we see it at the level of the experimental successes that the proteins synthesized by these models actually have. In this case, we see precisely the set of proteins that is shared by the three models, where, let's say, in general terms, all three pass this limit of 50% of them being expressed. Then we move on to the facet where there are only the proteins shared by the model in its version of 3 billion parameters and 46 billion parameters, where again, more than 50% of them are actually being expressed in the laboratory, despite the fact that they still present a sequence diversity between 40 to 60% with respect to the databases. Even for the set of sequences that only the largest model is capable of generating. In this case, even within this margin of 40 to 60% sequence similarity, we see that more than half of what would be the set expressed in reality does have experimental success. So again, these are not only hypotheses that remain at the level of bioinformatics algorithm, but they do actually translate into experimental success of the sequences. Now we move on to explore another facet within these generative models, which is being able to implement them through oracles or predictors of protein functionality or fitness, as you may remember. However, there is a quite interesting, quite characteristic behavior here, where it seems that this ability to scale the models, as we saw previously with ProteinMPNN, turns out to not go hand in hand with an improvement in fitness prediction capabilities. And here we must first recognize that when we actually predict fitness, What we usually do is take this type of generative models or with an autoregressive approach, because really when we take models that are masked, being able to compute, this type of probabilities is much more complicated due to the way in which they are trained with random masking. And in this sense, what the community has really done is adopt more autoregressive approaches to be able to precisely compute these types of fitness values. Although we are not really limited and we have to do, let's say, several tricks to be able to make a BERT-type model, we can derive from it, well, fitness predictions in zero-shot regimes. So, precisely the autoregressive models, through what would be this paradigm of being able to predict a state based on all the previous ones, are mathematically easier to be applied to be able to precisely calculate a probability that we can approximate as a measure of the fitness of said protein. However, it turns out that scaling does not seem to improve as the size of the model increases. It turns out that there even seems to be a peak, and this is precisely what we observe in a comparison of the different models trained in this ProGen family, where in addition to those 3 versions that we previously compared, an even wider range of models is offered when you review the preprint where 1, 2, 3, 4, 5, 6, 7, 8 versions of this model are trained, where you can see that on the yes axis there is a Spearman correlation regarding the probability assigned by this type of models with respect to what would be their actually measured values of the functionality of the proteins through these deep mutational scanning assays. And on the y-axis, we find precisely the ability to compute these models, and here we observe a quite characteristic behavior where, for example, even models with approximately 3 billion parameters represented by this blue or 100-colored line, they seem to be like the peak regarding computational capacity in relation to the Spearman correlation presented by their predictions. And when we move to the largest model, which is the one with 46 billion parameters, it turns out that the Spearman correlation decreases from 40 to 35. So this is quite strange then, because we might have been hoping that scaling the model would improve its predictive abilities for protein functionality. And this is something interesting, since it seems precisely that the predictive capacity of the model in the sense of fitness copes with a number of parameters of approximately 3 billion. And going from there, its predictive capacity even deteriorates. And this is something that is not only seen in the ProGen 3 publication, but is something quite recurring in any type of model that is presented in a new article each week. That there is always a ceiling in this type of models, where a Spearman correlation greater than 0.5 is not usually exceeded. And this, for example, we see in this other preprint in this other project, where another family of language models is presented, in this case called SMC. And again, we see this type of graphs on the y-axis, the Spearman correlation on the x-axis, the size of the model, the number of parameters. And again, this curve is drawn where there is a capping of approximately between 1 to 3 billion parameters, and subsequently decays, even when models with approximately 100 billion parameters are presented, as could be precisely the architecture that we already reviewed, which is called TRIMO-PGLM. So it really seems that there is no room for improvement where something is really returning to us. An improvement in terms of fitness prediction with respect to the scaling of the models, which is achieved precisely in this range of between 1 and 3 billion parameters. And so, now we have to turn to see, for example, what is the behavior of other predictive fitness models, where we really find that the performance varies too much depending on the type of function that we want to approximate. Here we find different benchmarks, for example, at the level of function of what would be binding, at the level of expression, at the level of catalytic activity, at the level of the fitness of the organism, for example, the ability to grow in a medium with antibiotic, or at the level of what would be the thermodynamic stability of the protein. And just for reference, I put here in red margins what would be even the largest protein language models that we know so far, which would be SM3 or TRIMO-PGLM, which have approximately 100 billion parameters. And although we might have expected that larger models would be more adept at predicting their function, it turns out that they are not. As you can see, really only in this case, in what would be predicting protein thermostability. Yes, SM3 would be found in the top 5, but in the other cases they are, let's say, relatively dispersed, and it really seems that there is not, let's say, a great model that is consistently always among the best performers. And that seems to be even independent of the type of modality that has been used during that training. Language models are made. Trained only with sequences, language models trained with multiple alignments of sequences, trained with sequences and structures, with structures and alignments, or with the 3 facets, sequence, structure, and function. Even regarding this facet that seems to be more unifying in that it integrates 3 modalities, we see that it is not really consistently found in the top 5 of best models. And even, as you can see, the Spearman correlations, they always tend to be within a range of about 0.5. You can see 0.502, 0.454, 0.533, 0.477, and 0.653. In general terms, it is quite rare for a model to actually exceed approximately a Spearman correlation of 0.5. And so it is quite sensible to ask ourselves why. What is happening that even though we are investing millions of dollars in scaling models, we are using a lot more data to train them? Why aren't they being good at predicting protein function or fitness? Well, first we have to make this recognition that the function of proteins really depends on the physiology of the host. That is something that is only known when the experiment itself is being carried out, and that is not really something that we can see a priori with a bioinformatics algorithm, which we see with this experiment. What we see here in this part of the diagram is a small phylogenetic tree made with what would be metalloproteinlactamases that give resistance to antibiotics, such as cefotaxime, meropenem, or piscilin, which we are going to express in different species. Even these species would correspond to the same family, which is the Enterobacteriaceae families. For example, we have that it was expressed in Escherichia coli, in Pseudomonas aeruginosa, or in Klebsiella. And the behavior of this type of metalloproteinlactamases is quite interesting depending on the resistance they are providing to each of their hosts. Or we perhaps find specific cases quite impressive, such as this beta-lactamase called CCRA, where it is quite interesting that when we express it in Escherichia coli, it is completely lethal. The cells die. However, when we express it in Klebsiella, it turns out that they do not die, and that even if it is given certain margins of resistance against cefotaxime, against meropenem, and against ampicillin. And when it is expressed in Pseudomonas aeruginosa, only in 2 of these cases of antibiotics does it survive, and in what would be its resistance against ampicillin, the cells do die. So here you can see quite clearly how the function of a protein really depends on the host. In this case, for example, with Escherichia coli they died. But with Klebsiella, it seems that they did survive, and that even if it gives them certain margins of resistance, and that even when you see and analyze the figure in more detail, you will observe that each enzyme provides a greater or lesser margin of resistance to each antibiotic depending on each species. So again, resulting in the main idea of this slide is that the function of the proteins depends on the physiology of the host, and that is always something that we have to keep in mind. Despite the fact that we have quite articulate algorithms or very good predictive capabilities. Here we also have to recognize another great aspect of what would be the investigation of why language models are not being good at this scale, to be able to predict the fitness of proteins. And this is precisely one of the premises that we use today. almost universally in machine learning, to model our data, which is the premise that our data becomes independently and identically distributed, something usually found in the literature as abbreviated IIB. Here, the independence properties are precisely that one piece of data in our dataset is not really providing us with information regarding another piece of data within our dataset. That is, obviously they are independent, and that during sampling they are not influenced by each other. And what the identical distribution is the fact that they preserve some statistical properties in what would be their representation in the dataset. Something that we can see better represented with this other animation, where we can observe what would be a completely biased sampling. Because in this case, our data collection method only focused on these small orange entity spheres. And what we intend is precisely to have this type of sampling that really becomes diverse, as you can see in this other section, where all the multiple modalities of the possible data hypotheses that we have are being shown. So this premise, although it is really assumed in any machine learning algorithm, sometimes it is very difficult to really validate that it is really happening in our data. And in fact, this is not the case with proteins. Proteins are not independently and identically distributed. And then you will wonder, why are they not? And then you'll remember what we saw in protein evolution class. For example, when we are talking about them being identically distributed, it is quite clear that this is not the case. The universe of proteins is tremendously biased. And we can see it with multiple examples. But in this case, perhaps the most marked, the most striking that I would like to share with you is the fact that there are some superfolds, which is precisely this idea that only a handful of small folds dominate approximately 30% of all the sequences deposited in the databases. And why? Well, it turns out that throughout evolution perhaps these foldings were more successful, perhaps in thermodynamic terms or perhaps also in terms of the possible solutions that existed during those conditions of the early Earth, as we already explored in the protein evolution class. And it is quite interesting how these supervolts are also related to proteins that are more diverse with respect to the type of functions they can perform. As you will remember, these aspects of the polarity of the protein in terms of its functional diversity are quite related to how the catalytic residues are coupling with respect to the scaffold residues. And when we talk about independence of distribution, we also find that this is not the case for proteins either. Again, in this case, we are addressing this idea of ancestral motifs that are shared by multiple folds, that are not really homologous, but it turns out that they are in multiple folds, as we could see in this motif called helix hyperin helix, which turns out to be in approximately 8 different folds that would correspond to up to even 15 superfamilies. And although they do not have, let's say, any degree of homology, any condition of homology that we can detect between these foldings, we do find this specific structural motif. And this again is due to the conditions in which the evolution of proteins occurred, because perhaps these small fragments, during what were these conditions of the primitive Earth, perhaps they were some nucleation centers that allowed them to fold more easily, or among multiple hypotheses as to why they could have been included in multiple foldings, even though they are not homologous. So here you can clearly see how proteins actually, they are not independently and identically distributed, and yet this premise is assumed to be central to being able to model and predict any behavior using machine learning algorithms. So what it is telling us is that we are starting from biased data and our predictions are going to be biased, and even this is quite well represented with what would be this idea that we also address in the protein evolution class, which is that the proteins that we observe right now really represent a minuscule diversity in even functional and structural terms of the entire reality of proteins that has occurred over 4.5 billion years of evolution, where we can even see it quite well expressed in Darwin's phrase on the origin of species where say, we can generalize it to practically any biological object that could be represented as a story imperfectly written in ever-changing dialects. Something that we also have to recognize is what our data is like in terms of the biological sphere in which we are working, and more particularly, obviously, in proteins, which is precisely the reason for this course. And then we come to the description of why our models, even though we are scaling them, are bad at predicting them. And then we recognize precisely the biases that are embedded in our data for evolutionary reasons of the object we are modeling, in this case proteins. But it is obvious that we need a somewhat more mathematical, more algorithmic explanation of why this is happening. And this is one of the things that articles like this one try to explore precisely. So to do this, we have to be clear about how these fitness estimates are made using generative models, which are precisely based on this premise of the relationship between a learned probability distribution with respect to a real probability distribution represented by the data that we connect, in such a way that any generative model that is being used to predict fitness precisely starts from this premise that they are identically independently distributed, something that we already know that proteins do not comply with. So then, there we are first recognizing a bias in the predictions that these models will have. And then, once we start from that learned distribution, what these fitness prediction models do is fit a probabilistic model, and then that probabilistic model is used to be able to infer a density that will be practically an approximation of fitness. What does this mean? Well, we have this learned distribution that was modeled based on this real distribution, and then here there will be some regions with greater density, and then those regions with greater density, the model will assign a greater probability. In terms of their plausibility, that is, how credible they are, how possibly real, come to express in the laboratory, because this is practically the possible set of responses that the model learned by observing what the real data would be. So then we are recognizing that it is, let's say, in a much more simplistic way, it is practically recognizing the data with which we use to train. And so here in this article, precisely the idea of exploring. Well, these generative models would also work in terms of fitness prediction in such a way that even the samples that we have are not really completely independent, and without having this property that they are independent, it is quite clear that our estimate of the original distribution, or in this case, what would be the stationary distribution, would really be biased, and these biases are being learned by our distribution learned by the generative model. And then they will take on the task of proving it mathematically. This is a preprint, let's say, quite complicated on a mathematical level if you want to review it. But in summary, they show that fitness is not really identifiable from sequence data alone. And so here we can explore an example of this. For example, let's say this is a phylogeny, and we see precisely how the evolution of our sequences occurred, where we start from the GLAKP sequence, where, let's say, throughout evolution, a situation suddenly arose from alanine to glycine, in this case GLGKP, and later another mutation appeared where GLGP occurs. So as you can see here, we now have three variants of what the proteins that we see today represent, represented by these terminal nodes. So if we have a very, very sensitive model, which is a model that can take into account these epistatic interactions, then the probability that it will assign to a protein, for example, GLAEP, which has not really been observed for these proteins that we currently see, will really be similar to 0 or will tend to 0. That is, during the training of these epistatic models that are quite sophisticated, well really. With the data you're looking at, you're not seeing a GLAEP type variant here, so then the probability or fitness you assign to it will be 0. Not so when we use a model where epistatic relations are not assumed, but rather they are assumed to be independent frequencies in themselves, where then, in this case, through this ability to not do much more complicated epistatic analyses, but simply an independence appears in the columns, since practically any variant that is given, again in this case the GLAEP variant, will have a probability or a fitness greater than 0, with respect to the fact that in this case it is not, let's say, limiting itself to the model. And in this case, obviously, this phylogenetic structure is not being considered given what would be epistasis throughout the evolution of proteins. If perhaps these concepts do not sound so familiar to you, I would also recommend that you review again what the protein evolution class is, where we really talk about what epistasis is and the effect it has on the level of protein diversification throughout evolution. And so, this is precisely what we are now observing in these new graphs, where what we observe is, in blue, a model that preaches fitness, but in a more simplistic way, using these independent frequency approaches, and a model that is epistatic, which is much more sophisticated and, let's say, better addresses this phylogenetic structure that occurred throughout evolution. And now we have it in two contexts that have been simulated by computer, where we are not going to go into much detail. But let's say that in this first context it occurs under what would be a regime where there is an evolutionary structure, and in this other one is where there is no, in this one there is no evolutionary structure, and in this one there is an evolutionary structure. And what we observe is that our most simplistic model in this case, let's say, is surpassed in the end compared to what our epistatic model would be. And this is precisely because there is no phylogenetic structure given by evolution here. Now, what would happen when there is a phylogenetic structure given by evolution? It is quite interesting that our most simplistic model, which in this case does not have these levels of power to address what the phylogenetic structure would be, is better in its predictive capabilities in terms of the Spearman correlation compared to a model that is epistatic and that is much more sophisticated. Which is quite interesting because one would expect that an epistatic model it would be able to better predict the functionality of proteins because it precisely captures the phylogenetic structure of evolution, which, well, incredibly does not happen. And even when we compare it with what happens when we use fitness predictor models trained with deep neural networks, for example, the IVE model, which is an architecture based on convolutional neural networks, or the WAVNET model, trained with a variational autoencoder, we found that the behavior of these fitness predictor models is quite similar, or at least above the margin of our epistatic models represented by these green and black dots, where we found that the independent model, the IF model, and the WaveNet model would be above our epistatic model, so they would then be reflecting precisely a behavior similar to this case, where there is evolutionary structure. And then, when you review more what this publication would be, which I suggest you go carefully and little by little because it is quite complicated, we found a quite interesting comparison regarding what the complexity of a fitness predictor model would be, measured by this parameter called H, which we are also not going to give much detail. But it is quite interesting that what we found is that as our model becomes more complex, going from left to right because it includes more parameters and therefore would pay more attention to a phylogenetic structure provided by evolution. Well, yes, its likelihood or probabilities are being better modeled by looking at this gray line, but at the same time, the correlation of its fitness predictions measured by Spearman's correlation turns out to decline. What it is telling us is that there is a point, let's say, a balance where very good predictions are given, but the phylogenetic structure is not being modeled well and vice versa. So in general, at least what is suggested in this publication is that larger models are not really good predictors of fitness, and that larger models and more data sets or more data are not necessarily. They may be reflected in a better ability to predict fitness due to these behaviors that we find even with simpler models that are not trained on architectures as massive as these based on transformers. And we obviously see this even better represented by this other publication, where what they really tell us is that the preference or biases on the part of these types of fitness predictor models, they are really a result of how they were trained. That is, the data that was used during training. Here what we observe is precisely the fitness prediction capabilities of two models. In this case, we see precisely an autoregressive model, such as the medium version of Bromgen 2, and the authors also took on the task of developing a protocol to predict fitness from BERT-type models, as could be the case of the ESM2 model in its version, with 650 million parameters, which now also allows us to compare both models, although obviously several mathematics was required to be able to implement this type of fitness prediction under this training regime. And what we observe are different fitness experiments at the level of organismal fitness, at the level of binding to a ligand, expression, catalytic activity, or stability. That everyone in general terms is represented by an average. In this black line, and what we observe both for what would be autoregressive models and masked models, is the fact that at the beginning it is observed that the likelihoods for some predictions correlate quite poorly. Then there are some correlations that are quite good, and then the model declines in its predictive capabilities again, measured as a Spearman correlation on the x-axis. And on the x-axis, we see the likelihoods or probabilities that precisely this type of models are assigning. And it is quite interesting that regardless of the type of functional task or type of fitness that we are trying to represent, this dynamic always occurs where it seems that there are precisely regions that are in underfitting or subadjusted regimes, and there is only a small margin of proteins that are well predicted. And here again, we find other cases where now it is not overfitted, but rather it would be overfitted. Here it is underrepresented, and here it is overrepresented. So what they are really telling us in this publication is the fact that what large models do is tend to overfit certain regions of the sequence space, where it is pathological in terms of fitness predictive capabilities, those proteins that have a low probability, because obviously there is a struggle in being able to predict which mutations could be good to be able to assign a higher fitness, but it is also a struggle in being able to determine which set of mutations would be really good to be able to predict fitness in regions where it is now overfit. Because then during training, these types of language models, well, it would have learned more statistical patterns at the sequence level, which would be being overfitted in favor of sequences that are overrepresented in the databases, such as these superfolds could be, such as the Tim-Bargel fold, the Rossmann folds, perhaps without which the beta-lactamases, and not like that, for example, our molecular meat that we reviewed in the alpha fold 12 class. And this idea is what we see precisely represented in these new diagrams, which really generalize the observations seen for both autoregressive models and masked models, where we find three general behaviors. In this case, when we look at this window of why they are being poorly predicted, what we see are different probabilistic spaces. In this case, only in the sequence space. They already make proteins that do have a large, considerable, quite good fitness, which we would be interested in finding and determining through experiments. Now, this space is being explored through our experiments, for example, those based on deep mutational scanning, generating these libraries of mutations, where we are exploring a considerable part of this space of sequences with high fitness. However, when we ask our model to assign us the probabilities of each of these sequences that lie in this region, we observe that the margin or sequence space learned by this type of models that are oriented in this case, which are in underfitting regimes, let's say that it is not so easy to be able to identify which sequences do have good fitness, as you can see. In all the possible space of hypotheses that these protein language models explore, it is not so focused on what would be the regions with high fitness. Now let's explore what happens in this other part of the diagram, where let's say it is this squeeze spot, or this moment where there is a great balance in terms of performance, where again we find the region of proteins with high fitness, the region that is possibly to be accessed through our demutational scanning experiments. And now what we see is that the likelihoods assigned by our language models, regardless of whether they are autoregressive or masked, are now much more focused on what would be the regions that would correspond to sequences with high fitness. So it is no longer exploring all the possible mutations that lie in this hypothetical space, but now it is limited to the set of mutations that it can explore and therefore can assign better Spearman correlations. And now we finally explore what this last behavior would be, where now the parts of the proteins are overfitted. Again, we have our high fitness space, our explored space between our experiments. And now as we see the language model, it is obviously overadjusted. And then simply, by reducing that margin of possible mutations that are beneficial, we are no longer showing greater diversity, as if we found it in this margin, where there is a good balance. So by overfitting, you are actually memorizing the sequences that were used during training. This is quite dangerous because perhaps then they are suggesting to us that these models are precisely memorizing some subset quite represented in the training, and that perhaps they would not be generalizing as we would like. And so the authors actually suggest that these types of protein language models that have been trained only with sequences tend to focus only on some specific families, perhaps those related to these superfolds, which means that the type of statistical patterns learned from these families are obviously underrepresented, and that, in this case, statistical attributes at a global level of the proteins are being lost. And then in this case, since statistics related to superfolds or widely represented families are preferred, we lose, let's say, the coarse grain that all proteins as a whole would represent. This is even another aspect that we explored a little more in detail in what is the type of data and biases. But also now you recognize that it can be both good and bad to be able to use these types of protein language models to be able to predict fitness. And as a solution to this problem, or, well, perhaps we could say an immense problem, what the authors suggest is a remedy that is even relatively simple, which is simply to do fine-tuning with the sequences that we want to increase their probability. However, this works when they are sequences that are in these underfitting regimes. But for sequences that are in overfitting, perhaps, we have to use other methods that perhaps eliminate some of the bias towards these favored proteins during training. So, for example, in the event that you are working with a protein that is underrepresented in the databases, such as our molecular horn, you could first corroborate whether it is really that poorly represented in the databases with two simple experiments. For example, they could take your sequence and simply blast it using the NCBI web server against, for example, the PDB database or another fairly general database called NR. And depending on how many homologs they recover, they could approximate to a greater or lesser extent depending on the number. Well, yes, the protein would be well represented in the databases. Or directly, if you can still access the UniProt web server, or even have clustered versions of UniProt or the complete UniProt on your computers, well, they can still blast their sequence using optimal algorithms to be able to scale to millions of sequences, such as Diamond II or MMSX. And then the same depending on the number of modules that you find in your search. You can determine if it would be well represented and therefore, let's say, statistically learned by this type of protein language models. And perhaps even more, if they want to go into detail, they can use what would be the CAD web server, which is this structural classification scheme for proteins, where they can even have access to the structural, functional, and sequence diversity of proteins. In this case, what they simply do is provide the sequence of residues and they click on search, and then their engine will give them these results. For example, in this case, I looked in particular for a protein corresponding to a class 1 aldolase superfamily, which is generally included within what is the Timbar fold. And what you see here are several graphs. For example, this graph at the gen ontology level of the possible functions that it would be making. Approximately 900 are functions. And even at the level of enzymatic diversity, through their enzymatic commission numbers or S number, we even find a capacity of up to 160 different catalytic activities. And here you can see various statistics, such as the number of domains found in this CAD database. Also, when clustered at different sequence similarity limits, how many proteins in the PDB to this fold have been deposited, among other aspects. And perhaps one of the most interesting graphs where you can have a greater degree of resolution of the sequence and structure diversity of your protein in question would be this one, where, for example, they highlight in red what would be the protein that you used as a query regarding the totality of foldings that they integrate into their database. And in this case, what would be this class 1 aldolase superfamily seems quite well represented in the databases. And that we can even find here some quite extreme points, which are precisely folds recognized as superfolds, such as the Rossmann folds, the Philip folds, the hydrolase folds, among others. So let's say, if your protein falls within this margin on the right, you might consider it to be fairly well represented, or perhaps if it falls within these margins. Well, they would need to zoom in precisely on this specific region to assess how much structural diversity and how much sequence diversity this CAD system suggests to them. And so you can have greater certainty that perhaps your proteins are being modeled better or worse by protein language models. But it turns out that not only is there a bias at the level of protein folding, but there are also biases at the level of the species that contain these foldings. So that this bias is now at the taxonomic level, since it is also represented in the databases. In this case, let's say that we want to use protein language models in sequence design processes. In this case, what we do is compute this fitness or these probabilities, verisimilitudes, to be able to show a space of mutations, and thus enrich it in such a way that every time we show a mutation, this probability assigned by some language model is maximized. And so what we assume is that we are exploring this fitness landscape in such a way that we are precisely arriving at variants that are maximizing the fitness or functionality of our specific protein. Something that we assume that this probability assigned by the protein language models really approximates the fitness of the protein, something that we now know. This obviously depends on the physiology of the host, but we make this premise to be able to really operate in bioinformatic terms. And then it turns out that when we guide our designs based on the likelihood or probabilities assigned by this type of protein language models, mutations are introduced that they maximize the phenotype towards some species that is overrepresented in the databases. And we see this, for example, in this case, where, for example, we start from a protein that is thermoresistant, for example, from these proteins that are used in PCRs, which turn out to be quite good because obviously they have a widely used biotechnological use due to their thermostability. And then let's say that we are dealing with this polymerase, which is quite thermoresistant and which corresponds precisely to a specific set of archaea. Archaea in particular would obviously be poorly represented in relation to the proteins of model organisms, such as the mouse, human, cow, chicken, among others. And if we apply these design campaigns based on the verisimilitudes assigned by protein language models, it turns out that when we observe the trajectories that are generated during this sampling of mutations, they cause what would be a protein similar to archaea to be lost, while maximizing phenotypes that are similar to proteins from model organisms or models overrepresented in the databases. In the case, for example, of archaea, of bacteria, for example, it is recognized that there are also considerably more bacteria of the protobacteria type, which in this case may not be of interest to us, perhaps because we are working with some cyanobacteria or some bacteria from another phylum. So now the level of bias also has to be considered not only at the folding level, but at the taxonomic level of the databases. And this not only turns out to be true at the species level, but also at the level of kinship in terms of their evolutionary neighborhoods, in such a way that even the probabilities or verisimilitudes that this type of protein language models assign are also influenced by this evolutionary neighborhood, something that we see now with these new diagrams, where, for example, here we have several species represented by blue, green, and orange, where it turns out that the proximity between what would be the blue species with respect to the green species, it is causing the model to assign even a greater probability to the proteins coming from this green species, and not to the orange proteins, because, as we can see, the blue proteins would be associated with a species that is overrepresented in the databases. Therefore, during this process of observation and probabilistic assignment, the model is giving greater preference to what would be the proteins of this blue species. And this is even transferred to its neighbor, which, for example, could be a closely related species. For example, let's say this is about Homo sapiens, and this is about some primate like the orangutan or some other primate in general. And perhaps this is extrapolating us to this other species that could perhaps be a banana or a rabbit, which would be evolutionarily more distant from this family of primates. So it is quite necessary that when you are developing design or fitness maximization campaigns in general, you take into consideration these two sources of bias at the protein folding level, but also now at the taxonomic level. And this is something that has even happened in machine learning algorithms in general terms, such as this example, with this algorithm called phase depixeler, which what it does is, well, we give it a pixelated image as input, and it must be able to depixelate it and give us the face that would correspond to said image. However, as models that have also been trained with your respective data, you are perhaps much more likely to recognize who this face belongs to, that perhaps it would be quite sensible for you to agree with me that it is one of the former presidents of the United States, that would be Barack Obama. However, when the face is depixelated, it turns out that it does not correspond to Barack Obama's face, but rather the model is generating several specific pixels that distort the reality of the face that would correspond. And this is precisely what protein language models are doing, but at the level of sequences and not at the level of faces. And this is something that is represented quite well by a tweet by Jan Lekin, a fellow researcher in artificial intelligence, of the pioneers in this area, where he says that any system based on machine learning will be biased by the data that is being used for its training. And in this case, well, models like Face Pixeler or FlickFace HQ are obviously trained with data that is intrinsically biased. And in the event that, for example, we had only used data from people from Senegal, where there is a much more marked African descent compared to white test people, well, obviously, this algorithm would tend to generate faces of people of African descent. We can even see it in other biases, such as these models called CLIP, which understand text-level descriptions of a figure or even the figure itself. And then we can assign some probability when we give it some specific text or image, which we are seeing in these cases, where what we do is provide the image of an astronaut, and then now we observe what is the similarity that the model assigns in terms of the text strings. And in this case, it is quite interesting how the model assigns a lower similarity when it says that she really is an astronaut, and assigns a greater similarity, saying that she would be a housewife dressed in an orange suit. And this is obviously wrong. Well, it does not mean that she is also a housewife, but mainly this is a photograph of her as a doctor, which would be the astronaut Aileen Collins, who in this case the model has been trained with a lot of data, and most of them are detecting that a female face is associated with a housewife, since it will assign a greater similarity or probability precisely to this type of responses, and not recognizing these nuances as it could be Ellen's characteristic face, the fact that there is a rocket, or the fact that this is not a typical costume, but rather an astronaut's costume, which together obviously recognize the total composition of the image as one corresponding to Dr. Ellen Collins, and not to a housewife dressed in an orange suit. And so there are quite a few biases recognized throughout the machine learning literature. And it is quite interesting how we as a community at the protein science research level are barely recognizing these types of defects in our algorithms. Something that, at least in my opinion, is quite well represented by this other opinion from Nolia Ferris, which really tells us that the easiest way to predict what we are going to be doing in the biology and machine learning community is simply to look at what our colleagues in the area of chemistry and in the area of machine learning are doing. So what they are investigating right now, we are going to see reflected in approximately 1 or 2 years of research, as this could be the case, where, as you can see, in approximately 2020, 2015, these explorations of how machine learning algorithms are biased were still being made, and only in 2022, 2023, we are reaching the same conclusions that our colleagues in these other areas had previously made. However, I do not want to leave protein language models in a bad light. Obviously, their capabilities at the protein design level are also quite interesting, as long as they consider their biases and know how to mitigate them using various design strategies, which we are going to explore below in the case that you are precisely designing proteins, using this type of protein language models to be able to predict fitness, using them as oracles. In this case, we are talking about an early protein language model, which was not even trained with a transformer architecture, but was trained with an LSTM-type architecture, similar to recurrent neural networks, which is called in particular UNIREP. And in this case, we are seeing that it was precisely trained in an unsupervised way, learning statistical patterns of all the proteins deposited in the UNIREP database, which is a database that takes the UniProt sequences and clusters them at 50% sequence identity. So now we are going to compare the performance of this general version of the language model with respect to a version of UniRep that has been finely tuned on a specific family that we want to improve, which in this case is a fluorescent protein. However, as in most cases in biology, there simply isn't much data, because data costs and obviously costs both money and experimental work. So it is normal to have really only a few hundred, tens, and perhaps in the best of cases, thousands of measurements of the functionalities in experiments. So it really is what it is. It is the data that we have, and we have to have certain cautious methods to be able to model them in a good way, and that are useful to predict the fitness of the proteins. This is a regime known as the low data regime, or low-end protein engineering, which simply means being able to maximize what we can extract in informational terms from said dozens or hundreds of data. So we start from the fact that we have very little data, and that we obviously have their corresponding functional annotations on a small scale. So what we do now is generate their embeddings using a fine-tuned language model for the protein we want, which in this case, as we previously reviewed, is maximizing the likelihood for said protein family, and therefore is better modeling its statistical patterns. And now we couple a model on top of our protein language model, which in this case can be a random forest, it can be a logistic regression, or it can be a two-layer, multi-layer perceptron. And then, once we have this architecture, we generate precisely a perspective design, where what we do is we take our sequence, we pass it through our oracle, which is composed of both our fine-tuned model with the apothic family that we want to model, along with what would be a supervised model, which in this case again can be one that adapts well to its computational or experimental conditions. And then we explore a mutational space with different protocols, such as Gibbs sampling or exploration using Monte Carlo methods, and we choose a set of mutations that equally maximize the probability. We then generate new sequences, pass them through our oracle again to predict the fitness, and repeat this step for several steps until we maximize the fitness of our proteins. And then this allows us to detect which variants would be more functional, again, multiplying. Performing the observation in this diagram, where we train on this regime and generalize to new sequence spaces. And this is precisely what is called the task of exploring in this publication, where what they do is explore in terms of fitness, what this flowering green protein is also flourishing in comparison to the designs that are being generated. With respect to a protein that could be the wild type, or the one that was used as a seed during these cycles of mutational exploration. And then here what we observe on the y-axis is that the model is also being able to distinguish how functional it is with respect to this protein used, such as Seville or the Vol type, with respect to how many sequences were used during the supervised training, which would be this module or head, which precisely helps us predict fitness in a supervised manner. And what we observe is the performance of the non-fine-tuned model and the fine-tuned model for the specific protein family. And we found quite interesting behaviors, where we found that even approximately 24 sequences or 48 sequences would be enough with a finely tuned model for the specific family that we want to model to be able to discriminate between the variants that do have good fitness and variants that do not have good fitness, as we can see when comparing with what would be the generalist version trained on this unsupervised scheme, compared to the fine-tuned version in this sense. So here what we observe in general terms is the fact that in these screening and mutational exploration campaigns, it would be highly advisable to perform these fine-tuning tests in order to maximize the likelihood that this type of models assigned to the proteins. And obviously, this was quite striking for the community, and they obviously took on the task of developing increasingly complex algorithms that have even reached what is known as effortful learning, which is what is explored in this new slide, where what we observe is precisely the way in which protein design was actually done in a traditional way with respect to the diversity that we have access to through exploration based on protein language models, where we found that locally, through, for example, directed evolution experiments, we only explored of the entire sequence space, a fairly local neighborhood. But thanks to the generalization capabilities of protein language models, we can explore a hypothesis space of this sequence space that is much more diverse with respect to the results of directed evolution. However, it must be recognized that these protein language models are really limited to showing regions that are more densely populated, because they are regions that also have more data. In what was the training? For this, precisely, these reinforcement learning regimes are used in particular. Here the authors took on the task of developing an algorithm called DPOPLM, which helps precisely to be able to generate sequences that, although we do not have data, we can apply this agent approach where our agent in this case is being represented by a protein model, which in this case is enzyme control. And then the environment will be given by what is the maximization of some specific property, such as the TM score, the PLDDT, the functional activity measured in the laboratory, or its catalytic activity predicted by other algorithms, such as this algorithm called CLEAN. And then we enter under this dynamic of people, where if during the training obviously people take a good path at the level of what mutation they are selecting, obviously the reward is maximized, and then the SAM control weights are reconfigured to be able to generate particular sequences or enzymes, because this is the behavior of this model that maximize these interactions with the environment. And this is precisely what we observe with this example, which in particular involves carbonic anhydrases, a set of enzymes that are represented by up to 8 different types, represented by the letters of the Greek alphabet, gamma, etc., where it turns out that when we see that they are also represented in the databases, only the beta-type carbonic anhydrases are the ones that are overpopulated. And these carbonic anhydrases are quite interesting because it is a case of evolutionary convergence, where it turns out that they are completely different folds. However, what would be their catalytic sites converge to be able to perform this specific enzymatic function. And then in this case, Through what would be the application of the DPOPLM algorithm, what is attempted is to maximize two properties. One is measured by the folding similarity provided by the TM score calculated by Foldseek, and the other is that the structures of the proteins are also being predicted, measured by the PLDDT provided by ESMFold. And then we see precisely on the y-axis, in this case. This folding measured by TM score is also being reflected throughout the iteration cycles of this reinforcement learning-based algorithm, where we observe that the beta-type carbonic anidrases are obviously the most abundant at the beginning, and as these reinforcement learning steps are taken, their probability decreases while precisely maximizing what would be the desired objective, which would be the alpha-type carbonic anidrases. So this is also a way to be able to guide and control the generations of our protein language models, without having to give them more data from which we can simply calculate from their sequences or structures, and thus obviously have better performances of the generations that we intend to model. This has even been taken to other more interesting aspects where, as you will remember, the models really GPT type are obviously more widely used to calculate this type of fitness. However, they are not limited to BERT type models. And in this case, this is what is explored in this preprint, where it is from the rather striking title, BERT has a mouth and can talk. That is, masked models can also be used as generative models, using various mathematical approaches, such as sampling using GIFs, which are ways of sampling and parameterizing various functions where we are not going to give so much detail. But in summary, what it tells us is that we can convert these BERT type models into GPT type models in terms of their generative capabilities, and we can even do something known as fixed backbone design, where we are conditioning the generative capabilities, where again in this case we start from a sequence. And so we want to maximize what sequences can exist based on a given structure. And this is obviously reflected as a contact map or the histogram, which is precisely what we want to maximize during this mutation sampling procedure, which in this case is based on Monte Carlo-based methods, in this sense a set of algorithms that perform this mutational exploration through several steps. And vice versa, we can also start from what would be a histogram, a specific structure, and now find what possible sequences are capable of folding into said specific pattern. Something that also helps us maximize or explore diversity now at the level of protein sequences. Or even during these regimes, what we can do is leave this two-dimensional contact pattern fixed, which is obviously reflected in three-dimensional coordinates, and then only design sequences that maximize the probability that this type of contacts are recovered, and that they are reflected in three-dimensional structures. And this type of protocols is precisely what is used in this other project that I highly recommend you check out, which is also quite amazing regarding the generative capabilities that these types of protein language models have. Where in this case what the authors do is use SM2 to generate proteins, despite the fact that SM2 is a BERT type model and not a GPT type model. But by precisely coupling these mutational exploration mechanisms based on Monte Carlo sampling methods, we can explore a space of possible mutations. And this is precisely what we are observing in this graph, where we find on the x-axis the number of mutational exploration steps by Monte Carlo, and on the y-axis we find two metrics. One is energy, which is a property associated with what these Monte Carlo sampling mechanisms are, which in short, energy also suggests to us how plausible our generations are being over time. And in this other part of the diagram we find the TM predicted by AlphaFold. And what we found is that during these early facets of mutational exploration, obviously the generations at the level of the structures that we look at are pretty bad in terms of PLDDT. But throughout this mutational exploration, until the last phases of cycles that maximize the verisimilitude that can be obtained from the patterns of the histogram. We find that precisely the capacity for folding similar to a conditioned folding that we have used as a seed to actually be generated is being maximized. So that in the last iteration of this sampling process, which is approximately step 170,000, the model is now capable of generating proteins that have a much higher PLDT and that are also being reflected as the structure that we used previously to seed or condition during the fixed backbone generation process. And this is quite interesting because obviously these generative capabilities by models that are not GPT type, but are BERT type, also have experimental success. And this is precisely what we observe in these last diagrams, where what we find is some designs that were carried out through what would be the implementation based on protein language models. Again, in the case of SM2, its version of 650 million parameters, with respect to designs that were not achieved through an approach based on Monte Carlo sampling and protein language models, but were based on another algorithm that in fact uses AlphaFold 2, which is called collab design. And what we find is their comparison in terms of different experimental trials. For example, that they did manage to pass several filters to be evaluated, if they turned out to be soluble, and if they really present patterns at the level of, for example, chromatography, among other aspects that are suggesting to us that they are the structures that we predict at the bioinformatics level. And it turns out that the designs obtained through language models, coupled to Monte Carlo sampling methods, they are successful in approximately 95% of the cases, or 19 of the 20 designs that they tested, and where 9 of them do present the desired structure in this sense. But when we use other protocols, such as this one based on AlphaFold 2, it turns out that it does not seem to be enough, because only one of 20 proteins tested in the laboratory actually managed to have experimental successes, but it does not reflect the structure that was designed quite well. So this only serves to illustrate that BERT-type models can also be used in generative approaches, but it is necessary to couple more sophisticated mechanisms such as Monte Carlo sampling. So let's say, general conclusion of this part is that you have to be quite careful when using protein language models as oracles. And obviously, you already reviewed that now protein language models are blinded at the folding level, at the taxonomic level, even at the fitness level that they can predict, so you have to consider all of these inherent biases, both in the training data and in the architecture, based obviously on your proteins that you are modeling in. Obviously, the task that you want to do with them. And well, once having explored, this is the first facet of generative models based at the sequence level, which as we saw usually occurs thanks to protein language models. We move on to explore the second facet of generative models, which on this occasion corresponds to the inverse task of structural prediction. In this case, what we do is start from a structure, and then we will look for the side chains of the residues, or specifically what the possible sequences would be that could be folded for said three-dimensional structure. A task that is known as inverse folding, or also has a second name known as fixed backbone design, because obviously we are fixing the backbone of our folding, and then we have to find all the possible diversity of sequences that can satisfy said three-dimensional folding. In this sense, we must recognize what were the seminal works for what would be this type of models, such as the structured transformer, which basically what it does is increase the capabilities of the classic transformer, but this time establishing new connections and above all, representing the proteins as graphs, and obviously encoding the three-dimensional aspects of the proteins. In that sense, as you can see, we obviously have the typical encoder-decoder architecture, where first the encoder has to look at the structure of the proteins to generate information-rich representations, and then the decoder also uses the structural information represented here by the blue arrows, but it also uses sequence information to then be able to predict which residues could be the most probable under this autoregressive paradigm. And specifically, what this transformer does is obviously not to train only with protein sequences, but now to use the proteins in a way represented in graphs, where we are obviously injecting several geometric paths so that the transformer understands and recapitulates what the structure of the proteins would be, in such a way that now we represent the proteins by nodes, which would be their residues, and also by their connections or edges or links between said nodes that would represent various properties related to what are, for example, peptide bonds among other aspects. And as you can see, this type of information passes through what would be a transformer architecture. Let's say a little classic, where, for example, we find the encoder module where the classic voltage also has to be computed. Only on this occasion, several inductive biases are also included so that it learns to interpret the local environment of each of the protein structures. For example, computing the tension only for what would be the closest residues, also including various aspects such as positional coding, since we also inject their information from the contacts 8 amps away, as well as the hydrogen bonds. In the way of representing the nodes, we do so, for example, by including information on the ihedral angles, or even in the way of now representing the links. We also provide what this model would be, with information about its distance, its direction, and its orientation. Something similar to what happens, say, in AlphaFold 2, with what would be this idea of local frames or residue frames, which provide the architecture with this possibility of being able to reason in geometric or three-dimensional terms. And as you can see in what this part of the decoder is, now the information coming from what the links or chemical bonds are, as well as the information of the sequence, is also being injected as a type of feedback, so that then it is the decoder, in conjunction with the rich representations learned by the encoder, that becomes capable of generating these tokens related to, say, what is the next most probable residue based on this feedback that I now have, both sequence and protein structures. However, this model was evidently, let's say, a kind of proof of concept, because it was obviously the first to be able to explore these types of facets to be able to generate this type of inverse folding protocols, and we arrived at what would be approximately 2022, where this model called ProteinMPNN is presented, which has practically been the most widely adopted by the scientific community, as it has demonstrated great robustness even at the level of experimental validation. And ProteinMPNN, which in summary from now on I will only say MPNN, is mainly based on a type of neural networks that we also already reviewed in the deep learning class, which is precisely these graph-based networks, where what would be a state of a particular graph corresponds to a specific encoding, and this encoding is obviously fed back with an increasingly larger neighborhood represented by these circles, where then we add the information of a neighborhood. In the following layers, we add more information increasing the radius of neighbors that we can include, and each of them obviously, as you see, also have their respective embeddings that represent various physicochemical properties of the proteins in this case, or even more specific, waste. So we can see this as feedback at scale. In a simpler way to say, it is like a kind of molecular gossip, where each of the residues communicates to its direct neighbors what type of information its particular microenvironment is representing. And this idea is precisely what the MPNN algorithm adopts, where in particular two main inputs are used to train this architecture. As we can see here, we have a small diagram that represents two chains. In this case, we have the L chain, the H chain. Obviously, there are two proteins, but each one represented by residues, and obviously their interactions with each other. In the case, for example, of what would be the information coming from the links, its atomic coordinates will mainly be taken. Only in this case, we are going to turn to see what the Bachmann atoms are. As you can see, here is the nitrogen of the amino part, the alpha carbon, the carbon, as well as the oxygen associated with the carboxyl group, and what would also be a virtual center that in this case would correspond to the beta carbons. We also have, for example, orientation and orientation information picked up from the connections between alpha carbon triads. Also similar to what happens, for example, with local alpha-fold frames. We also take what would be precisely the dihedral angles of practically the entire chain. We also take or measure the pairwise distance between the 48 closest residues, as well as a token that indicates that we are working with a specific chain. And as a second input, we mainly have, at least during the initialization of the training, a vector that was intentionally left with 0 values, since in this case, what we intend to inject into the model is this ability to learn local chemical environments, what the configurations of these side chains or microenvironment of the interactions are like, so that it then becomes capable of being able to say which is the most probable residue in a sequence. So in this case, we are not biasing the identity of the amino acid so much, but rather we are simply defining the residues based on their closest atomic interactions. So that is why we use a vector that is practically initialized to zeros, and throughout the layers of its architecture, these representations will have to be updated, which obviously will already have more information depending on how the functions are minimized. And in particular, this type of information is injected into another classic architecture that curiously is not based on transformers, but as we saw is based on these graph neural networks that we can also adapt to an encoder and decoder type architecture. Obviously, what would be the encoder is going to take all this information from the backbone and generate representations rich in information at the level of what the chemical bonds are, as well as the residues or nodes. And then this information will be provided to the decoder to be able to precisely assign what would be the most likely residue to satisfy said specific structure. And this becomes several cycles until the entire sequence is obviously predicted. However, here is a very interesting application of the MPNN algorithm, which is that compared to other similar inverse folding algorithms, which used a decoding that was from the amino end to the carboxyl end. In particular, ProteinMPNN does a decoding that is order agnostic, that is, it does it independently of the positions of the residues, which is all specifically what we see in this diagram, where, for example, here we see a paradigm that is autoregressive, where we have position 1, 2, 3, 4, 5 of the protein. And to be able to say which is the most likely amino acid to follow, we only have to take the information of what would be position 3, 4, to be able to predict the information of position 5. This obviously occurs under autoregressive schemes, but as we said, MPNN is order agnostic, so it does not necessarily have to take the information from all those that precede it, but it can obviously do so in a way, let's put it this way, disordered, but it becomes capable of satisfying which residue would correspond to said sequence. And this is quite important because even when it is done in what would be multimers, such as in this protein, it allows what would be this algorithm the ability to choose which amino acids would be the most likely to satisfy some particular geometric environment for all the chains. That is, it is already optimizing at the level of multimedical complexes, so obviously it is not only about monomers, but now it can also even predict sequences for multimers that obviously consider their interactions simultaneously. And this is obviously quite relevant, because we also want to know sequences corresponding to multimers and not only monomers. However, you might ask, because only the atomic interactions of the 48 nearest neighbors are being calculated. And this was obviously an engineering process, as also happened with AlphaFold 2, where the authors obviously took on the task of exploring multiple configurations of their architecture and found the most optimal versions in terms of various statistics, which in this case, for example, we see precisely on the axis of the how many neighbors we obviously use to calculate what the predictions would be, and a quality metric that in this case is the sequence recovery, which is practically what we are also recovering a sequence with which we have obviously trained the algorithm. The more we recover that sequence, it is telling us that the algorithm is almost even memorizing what the identity of said structures would be, so perhaps it would not be desirable, since we also want the algorithm to generate diversity. So then we have this as a reference to be able to evaluate that our algorithm is also really generating sequences. And in this case, we have two evaluations, both for monomers and multimers. And what we see is that the performance in terms of sequence recovery is really coping with approximately 32, or more specifically, to 48 atoms in the local neighborhood, right? So it was then chosen by default to be able to work on this architecture in what would be its final versions with 48 nearest neighbors. Also, another of the aspects that they explored in MPNN is the idea of being able to obviously dirty the predictions, so that thanks to obviously dirtying them, the algorithm is forced to learn representations richer in information similar. How the dropout regularization mechanism occurs. In the neural network, so that, in this case, what we do that we see represented in this diagram, which are the coordinates of the backbone, as you can see, each of these frames represents a particular residue with its corresponding alpha. Carbons, amino carbons, and carboxyl carbons, and then MPNN was also trained with the addition of what is known as Gaussian noise, where in summary we can vary the noise because at various scales there can be quite small noise or quite large noise, which will obviously dirty the atomic coordinates that the model is learning, and obviously forces it to be able to generate representations richer in information capable of dealing with said noise, which is obviously also desirable in terms of training, so that it generalizes to new sequences in this sense. And this is precisely relevant because this type of data is what was included in the nomenclature of their models. For when you access the repository on GitHub and then download and use the one you want. In this case, when you access the repository, you will find several models that usually come with this, from V48002. 48 is obviously the number of neighbors that were considered during training, and these three numbers that follow 48 are precisely the amount of noise that was used to train said version of MPNN. And obviously you have to choose a noise range. In this case, it ranges from 0.02, 0.1, 0.2, and 0.3 of Gaussian noise added during training. And this specifically is due to the fact that, again, we see on the y-axis, the one where we are recovering the sequences on the. And we also see it in terms of the experimental, or rather insipid, success of the AlphaVod algorithm, based on the fact that the proteins are also being predicted, for which there would be two groups with predictions greater than PLDT of 90 or 95. And what we observe is that, obviously, as we add noise, the recovery capacity of the sequences decreases, but there is a point where, obviously, even. Well, it is, let's say, pathological for the predictions provided by ESMFold at a noise level of approximately 0.3 Armstrong as standard deviations during training. And then it is precisely that you're going to find this nomenclature in the models deposited in the repositories, because that is the number of noise or the amount of added noise that was beneficial in terms of training, before what would be its predictive capabilities in terms of the quality of ESMFold's models declined. Obviously, you will also be able to choose, for example, which model you want in particular, let's say, depending on whether you want more diverse approaches or more strict approaches with respect to the diversity of sequences that you intend to model. Because, for example, larger amounts of noise have been associated with a mapping of the structures that is more robust in that sense. Sometimes they are also considerations that have to be kept in mind when using, for example, the MPNN algorithm to maximize diversity or properly control those designs in a more specific way. And obviously, not only does it remain a bioinformatics algorithm, but they took on the task of being able to demonstrate the success of their designs in experimental terms. Where you can see that there are two protocols where 150 proteins were synthesized, in this case using a hallucination protocol based on AlphaFold 2, and what would be another protocol that is properly the generations based on MPNN, where only 96 proteins were experimentally tested. And obviously here we see on the y-axis what these two groups would be, and on the x-axis, the solubility they had in the laboratory in experimental terms. And quite impressively, it turns out that of the 96 designs that were tested in the lab by MPNN, 73 turned out to be expressed in a soluble form, as we can see in these stark differences between the distributions of the two algorithms. But it also turns out that these generations provided by MPNN, they are quite thermostable, an aspect that we really all want in terms of protein design, so that our protein does not denature so easily, but rather remains stable over time. So not only is the difference marked in terms of the healthiness of the designs, but they are also thermostable, which is quite interesting regarding the behavior of this algorithm. And not only in terms of what the solubility provided by this type of designs would be, but also in type. Well, actually, the quality of the proteins that are being generated through their design, which is also what is corroborated with what would be a crystallographic structure based on X-rays, where we see the difference between the design in terms of AlphaFold with respect to the real structure obtained through crystallography, where we see that there is even a coincidence of the electronic densities of the side chains that is quite good, and that in general terms results in a difference in terms of RMCD of approximately 2.3 amps. So let's say that they could be quite successful computational designs, where they really do not deviate too much from reality. And this is quite interesting, also remembering how to interpret this type of algorithms trained with data from the PDB, similar to AlphaFold, where, as you will remember, we really do not have to see AlphaFold as a predictor of what the physiological conditions of the structure would be, a structure folded in its negative form, but rather they are precisely reflecting the biases of the type of proteins that are deposited in the PDB. That in this case, we are talking about proteins that maximize the probability of being able to be expressed, because obviously all those proteins that could not be expressed, which were perhaps even completely failed doctoral projects, are simply not represented in this type of databases. So there is obviously a bias towards proteins that tend to crystallize more easily and obviously the set of their conditions. For example, solution of temperature, among other aspects, are biases that this type of algorithm such as MPNN, which was also trained with the PDB, are learning. So we are learning that really the type of designs that MPNN is optimizing would be reflecting this bias towards structures that precisely have a greater bias towards being crystallizable, which is quite ironic and even. Well, we can implement these biases to be able to optimize our sequences and increase, for example, their solubility or even their thermostability. And obviously, this is something quite desired in protein design, in such a way that it is established as a kind of duo in what would be the interaction between MPNN, which designs sequences. And then we used AlphaFood 2 in its single sequence version to be able to assess whether this protein could really be experimentally successful. And if it meets several quality criteria, then obviously we test it in the laboratory, or we start with more iteration cycles between MPNN and AlphaFood 2. And this is precisely what we see in this other diagram, particularly for a protein that helps a colleague optimize in general terms. And what we observe are 7 cycles of this type, of MPNN and AlphaFol, which would be the protein of approximately 300 residues, as well as PLDDT. What we see is that, with respect to what would be the initial protein in gray, well, there are really some valleys in terms of PLDDT, where we observe that it could be quite bad in terms of its solubility, in experimental terms, and over 7 cycles of this, we see that the PLDT was raised globally to 95. But precisely, these regions that are even below a PLDT of 90 are eliminated, which could suggest that perhaps it could have greater experimental success in terms of its solubility or thermostability. And obviously this is reflected in terms of sequence, as you can see, which would be the original protein used as a seed, since it has some PLDT regions of medium to low confidence, or well, rather, from 70 to 90, and obviously what would be the latest design provided by MPNN, since it already optimizes the side chains quite well, such that the majority already have PLDT-type residues greater than 90. So this could even help us explore different configurations of the side chains to better characterize what the three-dimensional folds would be. And even this has also been applied in campaigns to improve stability, improve expression, and even function using MPNN, now including explicit information because thanks to its agnostic decoding. Well, we can set which residues we want to remain completely intact, and then on these the algorithm must be able to decode all the remaining residues. This is something that is precisely explored in this other project, where we take a protein from the tobacco virus, and then specifically we know some aspects such as which residues are in a location interacting with the ligands, with their specific substrates, as well as perhaps also information related to what a multiple alignment of sequences that are perhaps more conserved is telling us. So perhaps then by doing this type of analysis, we can choose which waste to leave fixed because perhaps they are 90% preserved, 80% or 50%. This will be up to each person regarding what type of waste to dispose of. For example, we have at least two different sets of waste that may be carrying out a relevant activity in structural terms or directly in functional terms. So obviously, once we fix these types of residuals, what we give to the algorithm is this ability to be able to explore different sequences that could satisfy the folding, but using this context of the protein to be able to design, well, several sequences we do not have design 1, design 2, design n, and then when they prove in particular that this strategy of being able to fix residues based on their interactions at the level of structure as well as at the level of what evolution marks us, they find that of a total of 144 sequences that were designed, 129 even managed to show, because its expression levels are much higher than what would be the protein used as a seed for the experiment. Something that we can finally see in this chromatography experiment, which in simple terms what it is telling us is that the stability of the protein in the solution is being improved, avoiding some aspects such as aggregation, among other aspects. So then the protein is being improved precisely in general terms. And specifically, well, obviously at the level of expression, stability, or perhaps functionality. But this obviously has to be based on assays that precisely measure its specific activity. Even other more interesting behaviors related to MPNN have actually been that they can even be fine-tuned for some specific purposes. That is, datasets specially curated for some property, such as optimizing solubility, optimize thermostability, specific expression in some host, for example, taking sequences that only correspond, well, structures that only correspond to Pseudomonas aeruginosa, perhaps another species other than E. coli, and then, well, MPNN is able to identify which local atomic environments could maximize, well, the idea that we find precisely sequences with greater experimental success. And this is precisely what is explored in this other project from approximately 2024. In this case, we have a problem, let's say, of the type of diversity that we have characterized at the level of the monomers deposited in the PDB, in such a way that the proteins that are usually associated with the membrane represent less than 3% of the total of this database. And this is precisely what we observe when being able to evaluate the database provided by scope, that is, at the level of the three-dimensional folds that this classification classifies, rather than redundancy. For example, those folds that are soluble, well in the intercellular medium, in the cytoplasm, well there are approximately 31,000 folds. And what would be the folds that have a more hydrophobic environment, because they are obviously inserted in the membrane, such as this claudin, this protease, or this G protein coupled to the membrane, since we obviously find different configurations at the level of the type of folding that we find in this type of environments. And it has been quite complicated to characterize this type of folding, since anyone who has worked with membrane proteins will find that it is really difficult to express them, manipulate them, because many of our tools are based on what would be foldings that are soluble in the cytoplasm in general. So then we need some way to be able to create what are known as analogs, which are not directly the proteins that are embedded in the membrane. By replicating an analog that presents a structural similarity and several properties similar to what the protein would be, we can precisely characterize it more easily with our experimental protocols. And this is precisely the task that is explored in this project, where we obviously consider MPNN is used to create this type of analogs, where in particular what they do is retrain a version of MPNN, but only using a dataset that maximizes the capacity to generate soluble proteins in this case. Something that obviously did not happen when MPNN was trained as the first project, since practically all the PLB proteins were integrated together with all their biases in order to represent a more general universe of proteins. And then the protocol to be able to generate these structural analogs is to first use a sequence generation algorithm based on AlphaFold 2 hallucinations. And then, since we have this feedback both at the sequence and structure level, we give the structure to this soluble version of MPNN so that it maximizes the probability of having these structural analogs. And finally then, we carry out a set of evaluations in bioinformatics metrics to be able to discard the worst proteins and preserve the best candidates to express in the laboratory and crystallize them. Which is what we see in this case, where we again find the structural analogs, in this case of Claudin, which would be this protease, and in the case of GPCR. And as you can see, this is the GPCR protein. Regarding what the design would be, as well as the experimental result in terms of crystallography, which differ by approximately 3.63 Armstrongs of RMSD. And obviously we observe that what would be the membrane structure, which is the original, since it obviously presents this behavior of its surface residues, which obviously have a more hydrophobic behavior. And what would be the structural analog, as you can see, because obviously MPNN in its soluble version was capable of assigning residues on the surface that are obviously hydrophilic and that are more easily expressed with our experimental protocols. And the same thing happens with these other membrane designs. In this case, the RMCD is 5.67. In this other one, it is 5.31. And we observe the same behavior, where what would be the native structure is presented in hydrophobicity patterns on the surface, and in its analogous version, precisely hydrophilic residues are found, which is signaling to us that this protocol for generating structural analogs is serving us well. And we can even, well, let's say that we care enough about fixing the behavior that is not soluble because it is associated with membrane. Well, simply thanks to the agnostic decoding stability of MPNN, we leave all these residues fixed, assigned to what would be the interaction with a membrane, for example, and solubility is optimized only for a specific region. And so let's say that we can more easily characterize what perhaps would be happening in this section that is completely hydrophobic compared to this other one that is hydrophilic, and that is tentatively improving the experimental successes that we can implement through the protocols. And so then, you can even fine-tune versions of MPNN to, let's say, comply with some specific property of the dataset that you seek to represent and thus have greater experimental successes. Even as we reviewed at the beginning of this class, the capabilities of MPNN to generate sequences have been extended, but now they explicitly consider what the information of those ligands would be. In terms of, for example, metals, including their interaction with nucleic acids or small molecules. So when MPNN was trained in its original version, this type of information was not really considered during training. But in its version that is now known as ligand MPNN, as you can see, the resistances are calculated again. In this case, it was reduced to 32 nearest neighbors, obviously because now we are also including the ligand information, where let's say in general terms, what is done is to represent this type of small molecules, both small nucleotide molecules and metals, through a representation generated by an encoder, where we take the atomic coordinates of this type of molecules and then feed it back to the classic MPNN encoder-decoder. And then by injecting this latent information, well, now the model is obviously being offered this ability to reason, not only in geometric terms of the proteins, but also now in even psychochemical terms of the interactions that it is establishing with the ligands. And as you will also remember at the beginning of this class, this type of algorithm, such as ligand MPNN, developed in David Baker's laboratory, has really caused these types of laboratories to adopt it mainly as their workhorse, their default method, to be able to generate designs that have these types of interactions with ligands. Which is quite interesting, since these types of laboratories are adopted in a way left and right and quite interesting or quite optimistic. These types of algorithms based on deep learning and perhaps some other groups, perhaps due to a phobia of algorithms based on artificial intelligence, are more reluctant to adopt them. Well, now we explore what would be the third facet of generative algorithms, where this time we are going to focus on the ability to generate structures. Only in this case, within all the possibilities, we are going to especially cover what would be the diffusion algorithms, which are the ones that have been most successful with respect to this task. You can actually observe that a generation can be done both free and conditioned to an interaction, which in this case was with a specific ligand. Obviously, for this type of structure generation applications, there are several architectures. For example, we find generative adversarial networks, a type of architecture that we did not really explore in the deep learning class. But in summary, two similar sequences compete as if they were an encoder-decoder architecture. Only in this case, we have an architecture that generates sequences and another that learns to discriminate them. And then during this process, they feed each other to generate designs that are more plausible. We also have, for example, architectures based on flow samples, such as this idea from AlphaFlow, where vector representations are learned, and then they make linear interpolations along this vector space to sample. We have the classic autoencoder. Obviously, we have the encoder-decoder part to be able to learn latent representations, and obviously from them, reconstruct the inputs in the most similar way to the real ones. And obviously, the architectures based on diffusion models that we also already explored. In this sense, we are not going to go into much detail because we have already explained this type of architecture in what is the AlphaFold 2 class. But at least here it is enough to understand that what this type of architecture does is a process of adding noise to what would be the three-dimensional coordinates of the protein atoms in such a way that we start from a completely structured structure. And we carry it out of order through this addition of noise until we have a completely random normal distribution. And then the algorithm must use this information from a latent variable that, as we saw, is usually a Gaussian to be able to reconstruct the three-dimensional structure. And simply during this mechanism, it is precisely during this mechanism that the model learns to generate structures, and obviously the diffusion models as well as the generative models at the structural level have had multiple applications. For example, to be able to generate treatments that are agnostic in this sense regarding various properties, or even that are specific to satisfy some type of interaction. And they have also been applied at the level of structure prediction, as in the case of AlphaFold 3 or even other algorithms such as Boltz or the other AlphaFold 3 predictions that we reviewed. To be able to generate sequences, something that is much more complicated, because we are adapting the generation of sequences to models that are not as easy to encode in an autoregressive way as sequences. And what would be the design of backbones itself, which can be with a fairly conditioned context, such as the presence of a ligand or another protein chain, or they can be completely free. And in general, this type of diffusion models or backbone generation protocols can be used in the following way, which is practically the archetype of protein design campaigns, where we have to define our problem in geometric terms in the first instance. In this case, we would care about what would be the specific microenvironment that perhaps occurs in an enzyme from the interactions that exist in the catalytic site. Whether, for example, our protein is interacting with another to be able to establish interactions and activate some signaling cascade or something specific, or what would be the formation of homoligomers, which in this case we see that. It is a protein complex of 6 units to establish complexes larger than its monomers. So we have to, obviously, in each of these aspects, think about the type of interactions that are being established and which ones would be most relevant to our protocols. And that, for example, we could condition the type of generations by this type of diffusion models so that they satisfy these interactions. Later in step number 2, we move on to the generation of backbones, where we obviously explore several facets. Let's say also using these parameters similar to protein language models such as temperature or TOPK, top sampling, others, in order to generate greater diversity of our backbones. And we are obviously going to have to filter these backbones using various quality metrics, such as the PLDT predicted by AlphaFold, among other aspects. And obviously once we have a backbone that we say is designable, since most of our builders have passed at least in silico form, well, even now we can apply these inverse folding protocols to be able to generate sequences that fold to said folding and then further explore what would be a possible space of conformations that may optimize some particular task that we intend to solve. To be able to really address how these types of diffusion models work when applied to the generation of backbones, it is also quite relevant to consider what their classification would be, and what helps us is to know, or to be able to deal in a greater way with the type of information that is being modeled as well as its possible biases, which is quite relevant at the moment for us to propose some computer protocol for protein design in this case. The diffusion models that obviously generate backbones are, let's say, in 4 broad categories. Those who train with what would be the information of a histogram, those who train with a 3-dimensional cloud of their atomic coordinates, or who even enrich these types of representations of atomic coordinates with what would be noise, in such a way that this idea of local frames is being added to them. And also, well, another version that instead of taking these 3-dimensional coordinates or histograms is taking latent representations learned by some other models, where obviously an encoder-decoder type architecture has to be purchased to be able to guide this entire process of adding noise and removing noise during the diffusion process. We also have other diffusion models that are capable of generating sequences. In this case, you are simply masking the particular residues, and then during the addition and removal of noise you have to learn which residues would be most likely to satisfy that sequence. And obviously at the sequence level, you can also use their latent representations or, in other words, their embeddings of some protein language model, in such a way that it is again coupled to an encoder-decoder architecture to be able to inform the noising and denoising process of the diffusion models. And finally, we have another category of diffusion models that in this case take into account what would be the backbone information along with the sequence. So obviously the latent space that is learning these types of models, it is clear that it will be different from the latent spaces trained with other types of paradigms or even specific types of modalities. In this case, we're obviously considering both the 3-dimensional coordinates of the atoms, along with what would be the sequence that in this case is, let's put it this way, completely masked. And during the noising and denoising process, obviously these 2 modalities of protein representation are being fed back so that obviously we learn to decode which structural state could most likely correspond. Obviously these are many diffusion models, so obviously I am going to make a recommendation of those that are most used in the scientific community because they have demonstrated greater experimental success and in general have been more widely adopted, which is precisely the models of the Rosetta-Fold diffusion family, which in this case, as you can see, is Rosetta-Fold diffusion, but in its version to the atom, which obviously tokenizes not only at the level of residues, but at the level of the small localities of all the atoms, for example, interactions with ligands. And then we have the most recent version of ESMFold diffusion, which is ESMFold diffusion 2, where even sensitivity is improved to even be able to generate active sites of enzymes, which, as you will remember, this type of design requires precision at the level of those MAMS, which is precisely what we see in this type of animations, where obviously the diffusion process is taking place, and then conditional on what would be an active site, since the model has to be able to generate a general structure. And as the second most widely used model, there is also what would be the Chroma model, which we also reviewed in the AlphaFold class, where, as you can see, Chroma is really also distinguished by its great conditioning capabilities, where we can condition at the level of symmetry, at the level of structural motifs, volumetrics, or even the productive context, generating designs that do have experimental success, such as designing proteins that resemble a letter of the DNA alphabet, which, as you can see in what these microphotographs would be, it turns out that they have been expressed in experimental terms. And perhaps more relevant to our protein design campaigns is also to consider the type of licenses associated with these types of algorithms. So when you also find a new diffusion or generation algorithm of any type, you have to keep in mind the type of license for when, let's put it this way, you intend to patent a design, because during what would be the development of your project, obviously use some algorithm that perhaps had intellectual property, and then the assignment of ownership of your patent is going to be a little more conflictive. And in this sense, fortunately, what would be the models of the ESMFold diffusion family, as well as Chroma, we find licenses that, let's say, are open source, which even allow this ability to be able to market the products of said algorithms, only that they will have to review in particular what type of, let's say, nuances there are in each of these types of licenses. But in general, these two versions, which is the BSD as well as the Apache 2 license, are considered open source in general terms. And not only is this a suggestion at personal discretion, but it is even reflected in what would be several comparison campaigns of this type of backbone generation models, where we find precisely this type of project called the dance of atoms, where evaluations of various diffusion models are made. Where again we are only highlighting what would be the Chroma or ESMFold diffusion algorithms, which is quite interesting that it is these two that exhibit a more balanced behavior compared to others such as Genie, Protiv, Protein SGM, among others. And this is precisely what we observe in this diagram, in what would be various design metrics, such as its computational efficiency, which is how designable it is in terms of the bioinformatics metrics that help us filter the plausibility, the nature of this type of proteins, how new they are compared to what we know in the databases, as well as how diverse. So you can really see the behavior of these two algorithms, which would be ESMFold diffusion and Chroma. For example, when we see ESMFold diffusion here, it turns out that this is the one that has, let's say, greater designability compared to the rest of the algorithms. And obviously, depending on the type of protocol that you are investigating, if for example you are interested in exploring a family, but in simple terms, perhaps not do X-ray crystallography experiments, because you can choose some type of metrics that obviously maximize the criteria of your research project. But if, for example, you are interested in exploring what would be experimental validations, then you would have to choose another set of metrics that are obviously more desirable for your specific project. And you can learn this in more detail by consulting this publication, where you will have more details on under what context you can choose what type of diffusion model to generate backbones. This other project is also quite interesting in the sense that comparisons are made of the diversity generated from protein language models with respect to diffusion models, where in general terms it turns out that these diffusion models they generate designs that are more plausible in terms of the confidence metrics that we can obtain from their structures. However, they are not as diverse compared to the designs generated by large language models, which obviously maximize diversity. However, their predictions at the level of structural metrics are perhaps of lower quality compared to the designs generated by diffusion models. And this is quite well illustrated in what would be these two-dimensional representations generated by reducing the dimensionality of the SM2 embeddings, which, as you remember, is a protein language model. Only in this case, we generate many sequences using this type of algorithms, for example with PROGEN2, with PROGPT, with Chroma, and with Rosetta-Fold diffusion. And now we return to their respective sequences. We represent them at the embedding level, and then project them to two dimensions using reduction algorithms, which in this case is Disney. And then, well, more algorithms are included here. Only in allotatory terms I only present four, which in this case are two protein language models, and in this case they are two diffusion models, and this is the totality of sequences, designed by various generative algorithms. And obviously, we observe that the sequences designed by large language models cover a much broader latent space compared to the designs generated based on diffusion models, where you can see that they concentrate more on specific regions. So it is reflecting this observation that obviously language models explore more diverse spaces compared to what would be the spaces explored by diffusion models. However, these would have greater success at the level of structural prediction. Obviously, there are several criteria that you can consider, such as the type of output that this type of generative algorithms give, the type of data that was used in training, which is quite critical because you have to look at precisely this type of information to be able to consider the biases that the generative algorithm is including, and therefore its designs. Even, for example, if some type of post-processing is required, such as being able to generate sequences using MPNN, or being able to validate using some type of structural prediction algorithm, such as ESMFold, among other aspects. And in this case, this is their last column in this particular publication, since they evaluate an experimental success by being able to evaluate what the design of a protease would be. However, I strongly recommend that you obviously explore this publication again so that you can learn more details about what contexts, select which generative model, and then maximize the probabilities of success for your specific research. Protocol. What we see here is even how we can apply this type of diffusion model to be able to explore issues that were previously much more complicated. In this case, what we have a protein that we want to know or think presents some type of interactions with some other protein during some pathology, something specific, but perhaps a priori we do not know what type of interactions can be established with respect to these two types of objectives. So in this case, we first start from our sequence that we think could be important. We identify which residues could be much more conserved. This is done, for example, in research in the literature that tells us that these are relevant binding sites or those sites that are perhaps conserved in a multiple element of sequences. And then we generate structures that satisfy these residues using backbone generation algorithms such as Rosetta-Fold diffusion. So that now we have a structure that suggests precisely this type of generative algorithms. And now with this structure, we perform a structural blasting with tools like Foldseek against the proteome of, for example, only corn, only mouse, only the biological model that we are investigating. And thanks to what would obviously be these structural similarity metrics. So now we can precisely find which candidate is perhaps establishing protein-protein interactions with our effector protein, which previously we did not know with whom it would be interacting, but which perhaps we hypothesized was quite relevant. And so now we can characterize the space of interactions between proteins. Well, obviously we don't know a priori, but thanks to what would be the information provided by Rosetta-Fold diffusion and Foldseek, we found the best candidates. As you can see, in what would be the Rosetta-Fold diffusion design, here represents a small section of a domain, and that perhaps obviously during Foldseek we find proteins that are perhaps composed of more domains. In this case, it would be 2, and that obviously this is the one that is establishing the interactions between proteins. We also find even much more complex design protocols, which what they do is couple other generative tools in order to maximize experimental success, which are increasingly complicated protein designs. Something that is explored precisely in this project, where they present a design protocol called alpha full dose cycler, where in particular they use the chroma model, along with its conditioning capabilities, which in this case we are conditioning on the ability to be able to fulfill a volume similar to the letter F, and then we generate what would be the structure using chroma, which, as you can see, really has an F shape. Then the information coming from this structure is given as a sequence to AlphaFold 2 to be able to have the histogram information, but also the result of this first prediction made by chroma. Well, we derive it with another protocol that allows us to obtain the sequence, which is a protocol based on hallucinations called chroma design, to be able to have, let's say, two modalities of our protein, both the structure and the sequence. And as you can see, again, through this diversification of the sequence, since we inject it again with AlphaFold 2, we obtain a histogram again. And then we compute several interaction matrices, which in this case we are not going to go into much detail about what they mean, but quite relevant in recognizing that we are now obtaining the feedback provided by, let's say, this ability to optimize proteins in terms of substitutability created by MPN protein, along with the ability to add noise so that the diversity of the generations is maximized. And so we generate a new sequence that now includes multiple modalities, including information provided by MPNN, information provided by histograms, and by chroma-based hallucination protocols that maximize sequence diversity. Finally, we then arrive at a candidate sequence that we again pass through AlphaFold 2. I know this is quite tedious, but the results we obtain through this type of protocol are quite interesting. To arrive at what would be the F-shaped protein that is most likely to have experimental success. That finally we optimize it for a last cycle using the soluble version of MPNN and discard those sequences using ESMFold in terms of PLDT, among other metrics. So when we have the final protein, we compare it in terms of TMSCORE so that it obviously improves, adapting more to what would be the original design folding. And as you can see, this whole entanglement of various algorithms, what allows us to really have successes, let's put it this way, quite interesting or complex that had not previously been able to be designed. Here what we observe is, let's say, the design capabilities of several protocols that were investigated in this specific publication, where, for example, if we use chroma, let's say it like this in its crude version, well, let's say as the size of the sequences increases, it becomes more complicated to be able to have results that are really satisfying the foldings that we want in terms of LTM score. But by making this type of sophistication, by feeding back through various generative algorithms, what we do is that through the interaction of chroma and this protocol called alpha bull cycler, since we maximize the probability that the fold that we intend to design is quite probable in bioinformatic terms, even at lengths of approximately 1,000 residues in total. Something that obviously when we are going to explore it in terms of experimental validation in the laboratory, we even find the ability to be able to design proteins with approximately 1,500 residues in total. This is a tremendous monster, which they precisely took on the task of being able to express in the laboratory, and it is previously observed that this little figure that has a triangular projection, actually in the photographs of the type of proteins that are observed, well, they do show, let's say, this similar morphology. And being able to express proteins of 1,500 residues, this is terribly complicated, since previously what would be our abilities to design proteins based on methods that have biophysical and biochemical bases in general, well, as you can see in this specific histogram, they simply did not go beyond 300 residues in general. Even these other proteins that do exceed these residues are usually binders. That is, we are crystallizing two complexes in what would be the protein-protein interaction, where usually the design of, well, the protein design work involves a mini binder that usually represents proteins of between 50 to 30 residues, and the other part would correspond precisely to the protein to which we want to add said binder. So the vast majority of our designs with our tools that we had prior to this type of protocols based on deep learning, since usually we could only obtain designs of no more than 300 residues. And now, thanks to this type of feedback between various generative algorithms, we can even design 1,500 residue monsters, something that was almost unthinkable before. Obviously, we also have to have several criteria in order to rule out which designs would be less likely to have experimental success. Something that is explored in this other project, where they propose an idea called COMPS, which is precisely a set of metrics that would allow us to select, in the particular case of enzymes, which would be the sequences with the greatest functional success. And in this case, it is quite clear that we have quality metrics for different facets of proteins. For example, we have quality metrics at the structure level, such as the energies provided through what would be the Rosetta suite, also the plausibility assigned by various inverse holding algorithms, such as MPNN, SM inverse holding or this other one, as well as, for example, the predictions of AlphaFold 2 in terms of the PLDDT. At the level of what would be a multiple alignment of sequences, we also have, for example, the likelihoods assigned by transformers that have been trained with MSAs, such as the MSA transformer. We also have, for example, the identity of its nearest neighbor deposited in the databases, which we can reflect as well as natural sequences. Or also statistics that we can calculate from the substitution matrices, BLOSSOM, for example, to be able to precisely evaluate what would be its most plausible context in terms of these matrices. And finally, we also have sequences, metrics that are derived from sequence, such as the likelihoods assigned by protein language models, such as specific protein language models for fitness prediction, such as SM1V, COW-V, or this other architecture based on convolutional neural networks, but which is a language model called CARP, in its version with 640 million parameters. We can also, for example, identify if part of the sequence corresponds to what would be a transmembrane section, which would obviously have little experimental success, because those are obviously membrane proteins. As well as other filtering criteria, such as finding sequences that actually exist. Well, they start as methionine, like all proteins, or they do not have many amino acid repetitions. All this type of information is obviously more detailed in what this publication would be, so it is still highly recommended that you review it. But it is also important to see how much they correlate so that, for example, the type of calculations that you are evaluating are not redundant, considering to be able to select which would be the most successful proteins to test in the laboratory. For example, this is a map, a heat map in particular, where what we observe is the comparison in terms of the Spearman correlations provided by these quality metrics calculated by various algorithms. For example, we found that the verisimilitudes given by the MPNN algorithm correlate at 8.9 with Spearman correlation with another algorithm that is also inverse folding, which is precisely SM1F. So then, in the evaluation of what would be several protocols, you could, let's say, save the calculation of this type of truth and minutiae, perhaps choosing only the MPNN one or only the SMIF one so as not to be redundant, since you would perhaps be providing the same information, and you can save the necessary computation to be able to evaluate their proteins, especially in cases where they are evaluating at levels of 1 million candidates or 10 billion candidates, where they would obviously need a lot of computing power. However, within all the criteria of being able to filter and select this type of proteins with greater experimental success, this protocol stands out, called the self-consistency test, which is practically an idea of the degree of designability that our sequence or our protein would have, which we can say take as the potential for it to be obviously stable, functional, and expressible in laboratory terms. So in this case, it is already done at the structure level or at the sequence level. Let's say that we generate a sequence with a full diffusion recipe. A structure with a full diffusion recipe, and obviously we have its structure that we can easily recover its sequence. Or we generate sequences with PROGEN or with ProGen 3, and we also obviously have the information at the sequence level. So once we have our sequences, we give it to these structural prediction oracles, such as AlphaFold 2 in its single sequence version, or also to SGMFold or MegaFold, but also recognizing the biases that we already reviewed previously in this class. In this case, they will also remember. In what would be the AlphaFold 2 class, we saw how AlphaFold seems to have learned a statistical potential that allows it to discriminate which structures would have a greater probability of being able to fold correctly without being able to give it information from the templates as well as the MSA. So in its single sequence version, what it is telling us is how likely it is that this sequence will actually fold. So that is why we usually choose to run AlphaFold 2 in its single sequence version, so as not to bias this type of analysis. And so, now we do analyze the type of structures that have been generated from our designs at the sequence level, where we graph what would be the PLDT of this type of structural predictors, with respect to the RMSD of our structure used as seed, where we will observe that usually with criteria of PLDT greater than 8 and RMSD values less than 2, we find proteins that are folding with respect to what would be the protein that we use as a seed. That this reflects precisely this idea of self-consistency, that if a predicted structure really resembles another structure, we are seeing how designable said folding is. And then, obviously, you could rule out these types of structures that obviously don't look anything like your particular design, which, as we can see here, if it's folded correctly. And here, although it perhaps has high PLDT values, we observe here the presence of an alpha helix that has nothing to do with our structure used as a seed. However, they also have to have. Let's say there is some caution regarding how to apply this type of self-consistency test, because when we actually generate sequences or structures that have been highly conditioned, for example, in their terms of interactions, it turns out that this type of algorithm such as AlphaFold 2 or ESMFold, because they tend to predict them as correct, even though even in experimental protocol campaigns, perhaps they are not being folded and expressed correctly. So also when they are working with protocols that especially include proteins that are again, they also have to be more careful and obviously design what set of to be used as a filter, as well as obviously turning to see classic aspects of protein design, such as the fact that it presents a hydrophobic core with respect to a hydrophilic surface, and thus obviously making this filtering even more refined of its candidates. Obviously, this also depends on the type of design you are doing as well, as even if you intend to train a specific generation algorithm, or perhaps more specifically, the fine-tuning where perhaps in this case, they obviously have to do the evaluations in benchmark, where they could be, for example, the set of tests provided by this other benchmark called Protein Bench, where they can also explore in more detail the publication where for tasks of inverse folding backbone design sequence design, or for example doing infilling or conditioning based on motifs, even specifically for the design of antibodies, we find a set of metrics that at least the authors suggest to us to be able to precisely evaluate them from multiple facets. For example, when using sequence design protocols, we can evaluate them in terms of their quality using alpha FOT2 PLDT predictions. In terms of how new they are, for example using the TM score, against what the PDB would be using FOLSIC. Or in terms of their diversity, well, we can also calculate what the GM score would be between pairs of all the proteins, as well as other statistics from FOLSIC, such as Max Glosser, where here you can see that there are different facets of these protein design protocols, and in each of them, we can make several evaluation criteria to which different sets of metrics correspond that would be giving us a greater signal, of which would have greater experimental success. You might also ask yourself, well then, how many sequences do I need to generate in order to have experimental success? something that was quite well reflected by this tweet from a colleague named Diego Del Alamo, who explores many articles and recommends it to the community. So I also highly recommend being able to follow him on the social networks he has, which is both X or Twitter, as well as Blue Sky, and where, for example, we can find mentions of the literature, such as in this specific article, where approximately 8,000 and 3,000 variants were selected to be experimentally successful, which perhaps would make us think that the experimental success perhaps comes from the fact of being able to make this bioinformatic analysis massive, and being able to consider up to 10,000 sequences, well, perhaps only a handful of them of perhaps a few hundred would have experimental success. And this is quite ironic because there has been a lot of progress on this specific topic in such a way that there are even new algorithms such as this protocol presented in this specific publication, where we no longer have to scale to the scale of thousands of proteins, but perhaps even a few hundred would suffice. And obviously in this case exemplified with only 96 sequences, where it turns out that 94 of these 96 were actually expressed in the laboratory, and perhaps some with greater functionality compared to others. But it turns out that the designs that are coming from this type of generative models, since it is no longer necessary to scale them to these dimensions of 10,000 or millions, but simply with a few hundred, or perhaps a few tens, depending on the filters that are applied, would be enough to be able to find designs with experimental success. And this also gives us the idea or guideline to be able to think about. For example, if we could use synthetic data to be able to train future models, something that we also reviewed in the Transformers and Language Models class, which is perhaps more related to these distillation paradigms, where what would be a large model that we consider as an oracle, for example in this case at the protein level, could be ProteinMPNN, because it obviously explores a greater diversity of the sequence space in terms of its capabilities of being able to generate sequences, and that this type of enormous models in terms of its parameters, since they can precisely distill information to much smaller models of the order of perhaps 3 billion or 15 billion parameters that are more easily executed on domestic range GPUs, and thus improve our predictive capabilities. And we also observed a similar behavior with what would be the training schemes used with AlphaFold 2, as well as AlphaFold 3, through their implementations of self-distillation or cross-distillation, where, as you will remember, the PDB actually contains few three-dimensional structures. So in the case of AlphaFold 2, they took the proteins from a subset of the UniProt to enrich the total of 170,000 proteins deposited in the PDB. Now, join them with 350,000, and thus be able to train the final version of AlphaFold 2 using practically 500,000 structures. Or for example, in the case of AlphaFold 3, in order to mimic the desired behavior, for example, that disordered structures are well predicted, that interactions with nucleic acids or folding of nucleic acids such as RNA are well predicted, since obviously different datasets were used that were weighed to a greater or lesser extent throughout the training, along with its three facets of fine-tuning. So then one might think that precisely those types of distillation mechanisms are occurring. However, when we think about it a little more coldly, it turns out that AlphaFold 2 is not actually directly using synthetic data to train, but rather using data that was already real, but simply not labeled, or labeled, for example, at the structure level. However, this does not remain that. This ability to be able to include synthetic data in the training of various generative models has not been explored, such as the publication of the SM3 algorithm, where when you review in detail what would be the methods section as well as the supplementary material, you will find that what would be the training of its largest version actually used predicted structures from the AlphaFold database, where in addition to using these structures predicted by AlphaFold, they also generated synthetic sequences using an inverse folding model to be able to enrich their set at the sequence level, where in particular in what would be the sequences coming from both the AlphaFold database and the ESDM Atlas database. Using the inverse folding model, they generated 5 sequences for each of the structures. And in the case of the proteins deposited in the PDB, they generated up to 64 sequences for each of the structures, which obviously allowed them to scale their dataset to, for example, 3,143 million sequences, or specifically, obviously, 236 million structures. Where, for example, thanks to the implementation of these inverse folding protocols, we see that of this total of 3,143 million sequences, approximately 12% would correspond to what would be these sequences that have been generated through its inverse folding algorithm, which is representing a fairly important percentage within the sequence dataset. And obviously, at a structural level, we're talking about 236 million, which has nothing to do with the approximately 250,000 structures deposited in the PDB. And obviously, this determines the type of generative behaviors by the SM3 protein language model in its version of 98 billion parameters. Something that, for example, we can see in this graph where the proportion of its proteins is precisely graphed in terms of folded betas as well as their proportion. In terms of alpha helices, where we can see in comparison what the distribution of the proteins deposited in the PDB would be with respect to the distribution of approximately 100,000 unconditioned designs, and we observe that obviously the distributions are different. That is, another structural space is being explored by the SM3 algorithm that is not recapitulating the statistics represented in the PDB quite well. The PDB also obviously has its biases. It does not mean that the PDB is, let's say, the gold dataset or the one that is completely representative of the universe of proteins. However, it is a fairly important reference to note when we are training our generative algorithms. This has not only been explored by this publication, but also, for example, we find several projects, such as this one called the DHOF Atlas, where we go into more detail about the success that perhaps being able to train with synthetic data would be providing us. And in particular, they do something quite interesting, which is to enrich what would be their training datasets, using in particular the Rosetta-Fold diffusion algorithm, in such a way that they again allow this algorithm to generate up to approximately 240,000 backbones or structures, from which we will now be able to derive sequences using ProteinMPNN of approximately 10 sequences for each of the backbones. And now, we have a dataset of approximately 2.4 million sequences, which obviously have to be discarded using this type of self-consistency protocols based on AlphaFold 2, OmegaFold or ESMFold, which in this case was OmegaFold, to finally have a dataset that contains its 240,000 sequences. Let's also have an associated sequence, but obviously also even a surplus already measured in millions of sequences. And now what is presented in this project is being able to train an architecture that they call DIHOP, obviously inspired by Margaret DIHOP, who would be the mother of computing, where various dimensions of her models are presented. Some models were trained, for example, using information from the UniProt clustered at 50 or 90% identity. Another model trained only with data from metagenomic sequences. Or, for example, this model, which is the one that matters to us because it is trained in addition to the UniProt data clustered at 50% sequence similarity. It now also includes the sequences coming from this synthetic data generation protocol, called the Backbone Ref, or BR in its abbreviations. We also found that they scaled the architecture from 100 million parameters to 3 billion parameters, in this case specifically training their configuration to what would be a 3 billion parameter model with the UniProt clustered at 90% sequence similarity, and 2 other models that, in addition to being able to be trained with UniProt, now also have an approach of being able to train with multiple sequence alignments. And obviously, all of these models were given the task of being able to generate their respective candidates and express them in the laboratory to see which one would be giving us greater experimental success. Which is precisely what we see in this bar graph, where in general 200 unconditioned sequences are generated for each one. And only 29 were selected for each one, between 60 to 1,500 residues, to be able to explore them in the laboratory. And it turns out that of the total of 144, only one generated 28 and not 29. Only of these 144 total, 40 were able to be expressed, reflecting approximately a 27 or 28% experimental success rate. However, the behavior is quite different from the models, where we can observe that the one that is most successful in terms of expressible designs in the laboratory turns out to be the model that was trained with the synthetic data that comes from this structural input protocol in such a way that what they at least suggest in their research project is that this ability to be able to increase the training data based on this structure generation protocol with Rosetta-Fold diffusion. What allows the model is this distillation behavior, to be able to inject structural information into what would be the sequence space, and then, obviously, be able to discriminate which sequences, let's say, could be more likely to be able to meet said structural criteria and be reflected in experimental success. This is quite interesting in the fact that it really seems that the injection of synthetic data is successful at the level of not only various criteria of bioinformatics metrics, but also at the level of experimental success. However, we also have to recognize that there are several dangers associated with training models using synthetic data. Something that is explored in particular in this other publication, where the risks of training this type of models using data that are not real, but rather synthetic, are explored in its statistical mathematical version, where we really found some problems that are reflected in the degradations of the model. For example, in the possibility of losing information associated with events that are rare or very infrequent in the datasets with which they were trained. And vice versa, obviously maximizing the probabilities of those events or data that are most frequent in our datasets, which obviously amplifies statistical errors. And this is quite complex when we approach it, at least in mathematical terms, but it is quite intuitive when we explain it with this doggy chart that we all like, where we can observe that, for example, we have here a diversity set of various dog breeds, which would obviously represent a natural or real data distribution. However, once we have trained our models on this relatively diverse data set featuring dogs, well, it is natural that some distribution that we intend to model turns out that the distributions that learn this type of generative models, as we previously reviewed, do not repeat the entirety of the original distribution with which it was trained, but rather a fairly plausible one. However, you can see that the diversity of, for example, dog breeds is reduced in this case, where, for example, we are now losing the statistical information associated with bulldogs, and yet the data that is perhaps more present in this dog dataset perhaps corresponds more to this other breed of golden retriever. And then, if we train our models with the generative data from this type of models, we see that the signal is amplified towards the data that presents the greatest bias in the training, which would be the data associated with the golden retriever. So that even as we train more and more and more and more with more data, these types of generative models completely lose the statistical capabilities associated with the distributions that we understand as a golden retriever, but that you can represent as, for example, structures of the beta-lactamase type, structures of the chinchoflactoreductase type, among any other protein, or obviously the proteins that are most widely represented in the databases, such as TIMBARs, such as Rossmann folds, such as immunoglobulins, will obviously present a greater tendency to be able to be incorporated in statistical terms by these models, with respect to other proteins that are underrepresented, such as perhaps thermoaquatic polymerases, which is precisely a protein that interests us in terms of biotechnology, because it helps us in the polymerase chain reaction, or PCR. So you can see that what these types of models trained with synthetic data are really doing is misinterpreting reality in terms of their distributions, and obviously biasing themselves in terms of being able to precisely model rare events that do represent a statistical reality, which tend to disappear throughout these training phases, and skewing their statistical behaviors even more obviously, which is obviously not at all desirable when it comes to generating some object, images, texts, videos, or proteins in our case. And obviously, we have to explore the type of biases present in databases such as the PDB or the AlphaFold 2 database, something that is precisely explored in this other project, where they demonstrate that the PDB really does represent a natural variation that is much broader than the AlphaFold database, which tends to be quite grouped in regions that, let's say, are more permissible. And we get this from what would be the information of the Ramachandran graphs, which are obviously plotting these ihedral angles on the x-axis and on the y-axis. And what we observe in particular here in blue is precisely the distribution of these angles of the PDB that we observe are much more dispersed, but that reflect a greater naturalness or diversity precisely than what is possible with respect to the space provided by the proteins deposited in the food database in red, where we can see how the set of possible combinations of angles and ivy is quite limited to regions that are, let's say, more permitted. The distribution of angles and hedgerows is much more constrained and is not reflecting, let's say, the real distribution that the AlphaFold 2 algorithm actually intended to make. Here again, we are observing how the type of distributions learned by our models really do recapitulate most of the distribution of our real data set, but not its entirety, because obviously we are not able to represent it in computational terms with any algorithm. And this is not only reflected with what would be the Ramachandran graphs, but also with other physicochemical properties, such as obviously the distances measured in terms of what would be the carboxyl carbon and the alpha carbon, between the amino and the alpha carbon, or between the carboxyl carbon and the amino group, where again we observe how there is a bias in the distributions between what would be the PDB reference with respect to the distributions of the AlphaFold database, which, as we can see, obviously this is also being reflected in terms of what would be the Ramachandra graphs, in such a way that it is quite clear that there is a bias in the proteins deposited in the AlphaFold database. And obviously, the PDB captures a physical realism that is much more desirable in terms of the generalization that we can obtain when training generative models or structural predictors from this type of data. Obviously, this is an aspect that is quite relevant to know when being able to train algorithms, as well as being able to implement them in our protein design campaigns. Although that corresponds more to the topic of what would be our last class of the course, where we obviously talk about where to get data, as well as the biases associated with them in more detail. However, obviously we are going to explore other examples a little so that you recognize that these generative algorithms are really biased depending on the data with which they were trained, as well as the architecture and training procedure. Here there is a behavior that is quite characteristic of what would be the majority of generative models, such as the model, not even a protein model, but at the genomic level, that is, it works with nucleotide sequences, which is called IBO. We also have other generation models based on diffusion, such as PLATE, PROTPARDIL, or protein generator, where it is quite curious to observe that the freely generated or unconditioned models, whatever you want to call, turns out that they have a higher proportion of alpha helices. As you can see, the proteins that are generating these types of models are practically pure spaghetti. And then we can ask the question, why is it that there is no presence of folded betas or even regions that are made up of spins to a greater extent? We see proteins that have a tendency to be globular and mainly composed of alpha helix, and that are not reflecting this diversity. Deposited in the PDB database, we obviously found proteins that are even completely beta-folded. Why is it that these generative algorithms only seem to be unconditionally designing alpha helix proteins? Well, there are multiple answers to that question. For example, when we explore this project where the architecture of AlphaFold 2 is retrained in detail, as well as what is learned throughout the training, we found that alpha helices are learned first with respect to the other secondary structure regions, which would correspond to their relative frequency deposited in the databases. This is precisely what we see in this diagram, where we find that they are also being able to be classified throughout the training, obviously depending on their training times, and obviously we find here that what would be in gray, green, yellowish, golden perhaps, obviously during the first stages of AlphaFold 2 training, or in this case specifically OpenFold, the regions associated with alpha helices are obviously being predicted in a better way. Later, the folded veins arrive, and then other regions of secondary structure. And when we look at what is the proportion, the count, the frequency of this type of secondary structures deposited in the PDB, we found again that what would be the alpha helix regions are the ones found most frequently. In second place would be precisely the folded beta regions, and in other places, the other forms of secondary regions. This could be a possible answer to why these biases are obviously occurring on the part of the generative algorithms to only attend to alpha helix regions. However, it is not the only explanation. There is also this other one, provided by another colleague, called Anastasia Borovieva, which in particular what it tells us is that it really seems that alpha helices are simpler, more designable, in terms of their hydrogen bond patterns, which obviously complicates a little more the design windows that this type of generative algorithms have to make when they are trying to model distributions that correspond. As you can see, in the case of alpha helices, we obviously have some hydrogen bonding patterns that practically correspond in a slightly easier way. However, when we find the configurations of the folded veins, we find that they can be both parallel and anti-parallel, and also the specific angle at which these hydrogen bond interactions have to occur. It turns out that it also has to meet several properties in terms of how large they measure in three-dimensional space. Something that is even reflected when we see these Ramachandran graphs, where we find that there are obviously regions with greater density, in particular those associated with regions of alpha helices. But when we show the space of what would be the IV angles for what would be the IVs, configurations of folded betas, there is only a set of river angles that is much more specific for those types of molecular interaction, such as hydrogen bonds, to be established. Perhaps we can look at this other map that precisely represents the densities in an easier way, and we really see how the folded betas are much more difficult to be modeled by this type of algorithms, because they have to consider a geometric environment that is much more complicated, both in three-dimensional and physical chemical terms, in such a way that these folded betas really seem like they would not be forming favorably in their latent spaces, and therefore they would not be generated under unconditioned criteria. So as a general, we see that it is more difficult to make this type of models generate precisely folded betas, because physically, chemically, and geometrically, they are more complicated to satisfy compared to what the alpha helix regions would be. So with this, we already cover the three facets that we had pending of the generative capabilities of protein models. And so now, we are going to move on to a last, brief part of the class, where we are going to present a summary of everything we saw in this section, where I would like to return to this phrase where it is really suggested that protein engineering really seems that we are not conditioned or tied to what would be the statistics that we see in nature. Using, for example, these algorithms as a recipe that had these strong physical-chemical bases, and that perhaps did not allow us to explore multiple hypothetical spaces both at the sequence level and at the structure level, but thanks now to the implementation of algorithms based on deep learning especially, as well as even more in particular what would be the large language models, both therefore only trained in sequence or in multimodal versions, as well as diffusion. Models or perhaps even flow sample models. What they do is allow us to enter a new phase of this world of protein design, where we can really make designs that are already completely articulated to a purpose that we intend to design in a much easier way. However, you also recognize that the implementation of this type of algorithms has to be quite cautious, because we have to look, obviously, at the biases they have. And obviously not only at the architectural level, but also with the data that was trained based on, obviously, the design task that we intend to model. So let's say, in order to summarize the entire class of protein design, we're talking about that just as there is a central dogma of molecular biology, well, let's say, there would also be its equivalent at the level of protein design based on artificial intelligence, where in general terms these design campaigns are based on the generation of backbones that are designable. We then maximize the number of sequences that may be meeting aspects such as expressibility, stability, or even functionality using inverse folding algorithms such as ProteinMPNN. And then we optimize the side chains that could satisfy a specific sequence that we will now have to validate using structural prediction algorithms such as ESMFold 2 in its single sequence version, as well as other algorithms that operate in a similar way, such as ESMFold and OMEGFold. Once we have generated this set of structures, now it is time to validate them in silico, using various quality metrics, such as those associated with this type of structural prediction algorithms, such as the PLDT, the pi, the predicted, TMSCORE, or their versions, in the case that we are using TMSCORE modeling. Multimeric complexes, as well as other sets of metrics that are also in silico, such as the pseudo-perplexity provided by the large language models, the degree of packing, the Rosetta energy, the surface area exposed to the solvent, among other metrics that you can consider. In this school, there are several groups. There are some who say that even the quality predictions provided by Rosetta are enough. And there are others who say that we can even perhaps extend the filtering criteria, obviously using these other types of metrics that have another, more physical-chemical perspective. This will also depend, let's say, on you and what criteria you are using to filter your best candidates. And even once we have predicted the structures of our designs, even using, for example, these self-consistency tests, we can even implement what would be a data augmentation, as we saw, through what would be the AlphaFold all-cycler protocol, to be able to even maximize properties such as solubility, thermostability, and thus even generate designs of up to approximately 1,500 residues in total. Also, when you find a new algorithm, well, in general terms, it helps us quite well to consider that they can be classified in 7 major categories. And this is a suggestion coming from this review article, which I also highly recommend reading, because it covers the entire world of protein design based on artificial intelligence quite well, since its title is quite explicit, where we also find phrases like this, where they really suggest that traditional methods seem to have reached their limit in terms of protein design, and that it is thanks to artificial intelligence that has emerged as a transformative force in our analysis capabilities, since it is capable of dealing with these challenges at the design level that were previously considered impossible, such as generating proteins with 1,500 residues, and that we can now do thanks to these algorithms based on deep learning. And in general, when you see a new tool, they can classify it into 3 main categories, and then into 7 more specific groups. There will be some tools that help us understand proteins better, and these can be seen related to capability of being able to search for both sequences and structures in databases. For example, it could be Foldseek, which opened a new panorama to be able to do research at the level of analysis like blasting, but on a scale of millions of structures, something that was impossible before. We also have structural prediction algorithms, such as AlphaFold 2, which opens up a new way to validate our experimental designs. In this case, in the particular course, we didn't talk much about what function prediction tools would be. However, there are quite a few tools that have obviously been trained using, for example, information from commission numbers in semantics, gen ontology, among other aspects. Obviously, all of them using, for example, neural networks based on transformer graph neural networks, among others. We have a second large category, which are those models that are more generative in nature. For example, it could be the idea of being able to generate sequences, such as ProtGPT, ProtGPT, or ProGen, which can even be conditioned to a specific motif. We also have algorithms that generate backbones, such as Rosetta-Full Diffusion or Chroma, which again can also be conditioned at the level of a symmetry, at the level of a volume, among other aspects. Finally, we have a third large category, which is precisely the algorithms that help us optimize our design campaigns, such as this screening capacity, to see what interactions our protein could be establishing with other proteins. That is, if it is a binder, proteins that can interact with nucleic acids, small molecules or post-translational modifications, and even also protocols based on deep learning that what they do is maximize the protein in terms of the preferential bias in codons at the time of being able to express our protein. Because we obviously have to encode every protein as a gene, and when we are expressing the protein in a specific species, such as Escherichia coli, it could be the mouse, the rabbit, or even in human cells, obviously each of these species, depending on, for example, its specific strand. They have some preference for the use of codons that they use at the time of this entire process of protein replication and translation. So obviously, there is a set of algorithms that we do not review in this course, which are precisely aimed at being able to resolve, given a protein, which would be its most probable gene based on a specific host. Obviously, there are many tools that you can choose from that obviously allow you to explore at the level of sequence, structure, and function of the protein. That in this case, for example, when they want to go from sequence to structure, it is quite clear that we have algorithms like AlphaFold 2 or 3, Bolds, ESGFold, Rosetta-Fold, among others. In order to go from structure to sequence, well, we have these inverse folding protocols like MPNN, MPNN in its soluble version, SMIF, among others. Or even at the level of exploring the possible conformations that a structure can adopt, we also reviewed that the alignment generated during AlphaFold 2 can even be subsampled, and thus find different conformational states. And so you can, let's say, select, well, what type of information do they require, and thus choose the tool they need. For example, in the event that you are starting from a function, obviously you would use algorithms such as enzyme control, ProGen, or Evotif, which allow us, starting from a specific reason or a commission number in semantics, to generate a sequence that is more likely to fulfill said function. This is just a scheme. Obviously, there are even more sophisticated schemes such as this other one, where we can also explore proteins in terms of their backbone, in terms of side chains, in terms of sequence, and you can go from modality to modality depending on the question that you intend to model during your research projects. So now they also know what type of tools to choose. They must also be able to look at the databases with which they were trained and reflect a little more about the biases and how they would obviously impact their research protocols. So thank God that I have lost my voice. We move on to the conclusion stage. The first is that the success of protein designs can really be defined in terms of their activity, their specificity, their stability, as well as the development capacity of said protein. For example, when scaling it to bioreactors or even complete factories. All of these are aspects that have to be considered simultaneously to obviously design a successful protein. As point number 2, we have that what artificial intelligence has allowed us to do is to be able to design protein complexes that have been much larger compared to those designs that we previously generated based on traditional methods that are strongly inspired by the biophysics of proteins. As a third point, we have the integration of multiple modalities as well as inductive biases mainly inspired by biology, such as the fact of equivalence. The links, obviously respecting their lengths and other aspects, seems to be a robust way to precisely model this paradigm of the relationships of the sequence, structure, and function of proteins. As a fourth point, we have that protein language models do not learn biophysics. And I repeat this, protein language models do not learn biophysics. But what they are doing is learning statistical patterns that are quite complex, and that perhaps can correlate with various facets of what we know as the biology of proteins, but that the sequences can actually be associated to a greater or lesser degree with respect to this statistical distribution, learned by this type of protein language models. As a fifth conclusion, we have that the largest protein language models, they're not necessarily going to be the best, as we previously explored with this behavior, where it turns out that fitness predictions deteriorate at a threshold of about 3 billion parameters, and that even these 100 billion parameter scale protein language models are pretty bad relative to these 650 million parameter versions. So it is because these protein language models have been trained on data that is biased. Obviously, they tend to recapitulate said biases in such a way that there are families of proteins that are overrepresented, and said models obviously overfit towards these families that have greater representability in the databases. As conclusion number 6, we have that generating new proteins really implies that the generative models are interpolating in this latent space that they have learned. And obviously this latent space, or this distribution, is really completely biased with respect to the distribution of natural proteins, which intrinsically is also already biased. So then we are learning the bias of the bias, and this also requires us to be quite cautious during our design protocols. And as the seventh and last conclusion, we have to, despite all these biases that we explored in this entire class of approximately 8 hours, it turns out that tools based on artificial intelligence have already reached a state where we can consider them as a technology that is already mature. And obviously, its integration into what would be our protein design frameworks is not only done as an intellectual or biotechnological curiosity, but it is already necessary. This is obviously, in my opinion, and obviously those laboratories that are reluctant to adopt this type of technology that has consistently demonstrated its success compared to our previous methods, past in physical chemistry. Well, it would perhaps correspond to a much more particular opinion of each person's interests, and not to the great results that these types of tools are giving us, and that obviously allow us to explore not only at the level of protein design, but at the level of biology in general. So at least on my part, and perhaps because of your interest in this class, it is quite clear that the adoption of this type of tools in our work is now necessary. So, with this, we come to the end of this class. Thank you very much for having accompanied me for approximately 7 hours. I don't know how long it lasted. And remember, we really did this not because it was easy, but because we thought it was going to be easy. No protein design protocol is going to be easy. You are going to encounter multiple problems, and you are going to have to be able to reflect on them, to their tools regarding their data and their objectives, and obviously explore possible answers. They have to be able to address their problems. So this has been it. Thank you very much, and see you in the next class.